नमस्ते नमस्ते देवीजी लगायत नित्य डाक्टर साहब सबै साथीहरूलाई नमस्ते।
नमस्ते नमस्ते नमस्ते।
सुरु गरौँ है त आजको सेसन।
निशान भाइ त्यो भिडियो बजाउने भने नि त
निशानजीले स्टार्ट गर्नु।
ओके यो भिडियो सिद्धिने बित्तिकै हेरोसन जी स्टार्ट गर्नुभएको छ। हामी अनलाइनमा छौँ अहिले खास चाहिँ। किन साउन्ड भइरहेको थियो?
Namaste. Good afternoon to those who are watching us from Nepal, and good morning to those who are in the Western Hemisphere of the Earth. First of all, on behalf of the coordination committee of this session, I would like to extend very warm welcome to all the participants in Zoom platform and also the viewers in Facebook Live. I'm Roshanlal Shrestha, currently living in Washington, DC, USA. By training, I am a cancer biologist working as a research fellow at National Cancer Institute, NIH in the USA. I received my PhD degree in genetics from Cambridge University and completed my postdoctoral training from NIH. I'm also the founder of Center for Health and Disease Studies, Nepal. It's an honor for me to moderate this session today. Before starting, I'd like to give you a quick overview of this session. So this session will be divided into 3 subsections, followed by a panel discussion. So each scientific session will have 4 speakers from various fields in biomedical sciences. We will introduce the speakers as we move on with the talks. Each invited talk will be 20 minutes and contributed talk will be 15 minutes, followed by 5 minutes of question and answer session. We will take the questions after each talk. So if you have any questions, you can either post them in the chat in the Zoom, or or post them in the Facebook comment section. To speakers, it's my humble request to stick within the allocated time, and to moderators, please monitor the timing so that we can be on schedule. Before 5 minutes of ending the session, we will give you a signal. For example, for invited speakers, we'll give you a signal on 15th minute and then on 20th minute. The first session will be moderated by myself and Dr. Devi Basnet. Dr. Basnet is also the chief coordinator and chair of this session. To introduce Dr. Basnet briefly, he has been working as a senior scientist for a global pharmaceutical company called Meditox in South Korea. Dr. Basnet completed his postdoctoral training from Iowa Women University, and his research interest is genetic engineering and protein therapeutics development, including antibody production and processing. Dr. Basnet was the director of and current president of BRIC Investment Private Limited in Nepal, and was also the board of directors of Research Institute of Bioscience and Biotechnology. Currently, he is involved in various research activities in RIB. For the second session, our moderators are Dr. Sushila Maharjan and Dr. Nitesh Aryal. Dr. Maharjan is a research scholar in Brigham and Women's Hospital in Harvard Medical School. She received her PhD degree in biochemistry from Sun Moon University, Korea. Dr. Maharjan's research interest is on developing organ-on-chip system and 3D bioprinted tissue models to mimic human organs in vitro. She is also the founder of Research Institute of Bioscience and Biotechnology. Another moderator, Dr. Aryal, is a senior physician based in Doha, Qatar. Without further ado, I'd like to start the first session, and to start the session, I'd like to invite Dr. Devi Basnet to chair the session and also give a brief overview of the biomedical technologies. Dr. Basnet, it's over to you.
Thank you. Namaskar. Thank you very much for starting the sessions. Before I go through further, I want to share my slide, and I want to quickly go into the introduction. Well, as we already know that biotechnology, biomedical technology. Can you see this, my slide? Okay. As we know that we are having a session of biomedical technologies. Actually, this session deals with the kind of a technology, especially using for the engineering and technology principle to the domain of living or biological system. And as we know, we have known so many things before, but the recent advancement in technology make us very easy way in all aspects of life. Particularly today, we are talking about the technologies we are using for the advancement of the health and disease. How we can make our health more efficient, and how we can cure the disease by using several kind of technology, particularly engineering of several technologies. We will be talking about these kind of things. So if you see here, briefly, there are some kind of technology which we would not have imagined before a few years ago. For example, we are using some bionic hands that we can feel. And also there are some kind of technologies for the cancers which were not thought of curing the cancers by using some technology like a CAR T-cell treatment. Or there are some kind of augmented reality which helps in medicine to go for the several kind of operation or to understand this biology, so that the people can understand and can treat it directly. There are also some information that we can see like a direct to consumer genomics. As we know, nowadays we can have a sequence of all the genome within a very short time and with a very cost-effective way. If you have known the genomic sequence, then we can have a lot of information about the personal treatment. So all this happens because of the technology that we have. And similarly, there are some kind of 3D printing system, like now we can have a print up to kidney to hands, and we are thinking of rudimentary organs replacement. On the other hand, there are some kind of nanotechnology helping us a lot of in health and disease cure and understanding, like nano robots, which can be used in living in our bloodstream. And not only this high advanced technology, By using all the technology, we can develop very simple but effective technology. As we know, this dye biotechnology, which is a kind of a technology that can be used for the test of pancreatic cancer by using simple method. And there are so many other robotic and surgical methodology have already been developed. So in short, there are so many technologies, particularly making our curing of disease and making our life very efficient are being developed. So in this session, we are talking about some of them in detail, and we will be discussing onward. So this is all about our session. And with this, I would like to invite with our first um, speaker in our session. So our first speaker is Professor Ramesh Giri. Professor Ramesh Giri is Weinberg Early Career Professor at the Department of Chemistry in Pennsylvania State University. He received his MPhil from University of Cambridge and PhD in chemistry from Scripps Institute. Professor Giri's research group is focused on developing and understanding sustainable chemical processes for the manufacturing of pharmaceuticals. He conducted mechanistic study on key chemical technologies for the biopharmaceutical, and he was rewarded various kind of fund and rewarded. So Professor Giri will be talking something in line with his research team on title Sustainable Chemical Approach to the Pharmaceuticals. So onward, this is for Professor Giri, please. Maybe you can share your slide from there. All right, you are here.
You are on mute, Dr. Giri.
Oh, yeah. You are muted.
Yes, all right. I was trying to find my window. Thank you, Dr. Basnet, for very kind introduction. And also I want to thank the NRN Second Global Knowledge Convention for the invitation. As Dr. Basnet mentioned, my research group works on developing sustainable chemical transformations that we hope eventually will be applied to provide sustainable approach to pharmaceuticals. So a lot of people talk about sustainability depending on the process they use. And in our area, a lot of people talk about sustainability with regard to using cheaper metals, which I'm showing here on the periodic table, which may not make sense to a lot of people. But the most common metals like iron, cobalt, nickel, and copper, those are sustainable metals and that are available in nature in large quantities. My group not only works those cheaper metals to try to make the pharmaceutical process sustainable, but we are trying to develop a new chemical transformation that will eventually make the process to make pharmaceuticals sustainable in the long term. As human beings, right, we come across a lot of different types of drugs in our lifetime. We use them, our family use them, and our friends use them. And most of the time we are familiar with the disease and drugs. For example, I'm showing here is some of the examples of drugs, lowering for blood pressure, and also for diabetes, right? So a lot of us use these drugs, but we don't know where they actually come from, right? Most of the time we go to the pharmaceutical, the pharmacy and then get them. So for us, the origin of these drugs is pharmacy or doctor, right? But what we don't realize is, all the time, and I would say most of the time, these drugs are actually synthesized in organic lab, right? Which, I'm showing here the picture of the organic lab. These drugs are originated, or I would say born in synthetic labs, right? So a lot of people won't realize that these drugs actually come from the synthetic lab, right? And whether the synthetic lab is in the academia or the pharmaceutical industry. So today I'm going to talk about a little bit about what we do in our lab at Penn State, trying to develop a new chemical process that we hope eventually make the access to drug convenient and faster and cheaper. So that's the process we are working on. But before I go in there, right, I want to give you an overview of like why do we even want to do that, right? Because chemistry has been developed for over 100 years, especially organic synthetic chemistry. But what's out there that's gonna make the drug synthesis in the future sustainable? So if you look at the population of the world, right? The population is exploding. Now it's at 7.8 billion as of October 2020. Along with that population explosion also comes the public health spending, right? The public health cost is also rising, dramatically over the years. And I've shown you this picture that predicts as of to 2060, right? So in order to make, in order to fulfill the demand that comes from 7.8 billion people for drugs, we have to be able to access them rapidly and cheaper, right? And with the COVID situation, now we already know that we're trying to expedite the drug discovery process, and it's going to take time before we finally can get a drug, right? That's because the process for developing a drug is a long and tedious process, and with an average of 10 years and a price tag of 2.8 billions, roughly, depending on which drug and which company it makes, right? So that process, let me just get the pointer here. That process starts with discovery and development, just like in organic synthetic lab, where research begins, trying to get some chemicals that, after you find a lead compound, that goes to clinical trials. And then if you pass through the clinical trials, and most of the time those chemicals fail in clinical trials, and they go back to discovery and development phase. And if you pass through that, then you go through the governmental, the approval process, and then you go to market, and then the government starts monitoring their efficacy or effectiveness in public. But most of the time is spent on the drug discovery and development process, because that itself is a very tedious process, right? And here I'm showing you the drug discovery cycle. That is, you can start with in a collection of large number of chemicals that you're going to test for the drug, for the type of disease you want to find the effectiveness for. And, so once you find some compounds, lead compounds that have some effectiveness, then you do a structure activity relationship that also requires synthesis of new chemicals. Then you go and design based on that structure activity relationship, new chemicals, and synthesize them, and go through this cycle over and over again until you actually get a clinical trial that can then go through the clinical trial processes. So now, most of these drug discovery cycle steps involve synthesis of new chemicals, and that actually happens in organic lab, right? So even if you synthesize, the compound, and then it becomes a clinical candidate and goes through all the approval process and to the market, then to bring it to the market, you do need to make them in large quantities, hundreds of thousands of kilograms, to feed the 7.8 billion people, and hopefully not everybody is sick, right? But we can expect a large fraction of people to be sick, different from different diseases, right? So even after R&D, that spends a lot of time in synthesis and trying to make new chemicals, the cost distribution for manufacturing a drug also has several factors that contribute to it. So I don't want to go through all the details of this, the factors that contribute to the manufacturing of a drug. But if you look at the volume time output, that contributes about 40% of the cost toward the expenses toward the cost of the drug, right? So if you look at this volume time output, what that basically means is that how big a pot you need to make that drug, and how many synthetic steps you have to apply. So the more the number of synthetic steps, the more the volume and longer the steps, and the volume time output would be larger, right? So that means if you can discover a new chemical process that actually shortens the synthetic routes, you will be contributing to a large fraction of the cost of the drug manufacturing. And if you can decrease the volume time output, it also has consequence on material cost, because you will be decreasing the material cost, you will be generating less waste, atom economy will go up, and yield will also go up. So you can actually impact the cost of the drug by about 75% by developing a new chemical transformation that will eventually shorten the synthetic process, right? So unfortunately, the current technology is such that it requires a large number of synthetic steps to synthesize a drug molecule. And in the long term, we don't think that's going to be a sustainable process that we already saw in this COVID situation, that most of the time we try to figure out what drug actually works for COVID-19, right? So that's a long, lengthy process, and with the ever-growing global population and demand for drugs, so we don't think that it's going to be sustainable in the long term. So this is where my research group is involved. We're trying to reinvent the way people make not only the drug molecules, but any lead compounds or bioactive compounds or any complex molecules that has any potential for developing into pharmaceuticals. So here we're trying to develop a new process, new chemical transformation that will eventually shorten the synthetic routes for making drug molecules, not just by 1 or 2 steps, by a large number. So just to give you an idea about how the synthesis works in making drug molecules, here I'm showing you a very schematic diagram where you have a final drug. And let's say, for example, that final drug is actually derived from 5 different pharmaceutical precursors. Then it takes several steps to put them together in order to make this final drug. So with the current technology, the way people do in synthesis, making the drug molecule, is that then you start one precursor, and then you require several steps to prime this precursor so that it can react with a second precursor. So you have to go through several steps before you can make the first precursor be able to react with the second one. And then you react with the second precursor, and then you get this intermediate. And with this intermediate, you also go through the same priming steps, through several steps before you can make this first intermediate, reactive with the third intermediate. And you react with that through several steps. And you go through this iteration of the process, not necessarily the same synthetic process, but different chemical transformations. Eventually, going through priming and reacting and priming and reacting to actually make the drug molecule in large, number of synthetic steps. So those priming steps basically mean that you have to do protections, deep protections of functional groups, and a lot of functional group interchange processes, functional group interconversion processes. So what we want to do eventually, in future, as a future technology in chemical synthesis toward pharmaceutical, is that we want to take the precursor here, just I'm showing you the exact same precursor to give you an example. And we want to use as fewer steps as possible to prime it. And we want to react this with the precursors that are here, not only 1, actually 2. Or if we can do it 3, that would even be better, right? But at the moment, once we prime this intermediate, we're going to react with 2 of the precursors to arrive at this intermediate rapidly, where this intermediate was obtained through several steps in the current technology. And we're going to use another couple of steps to prime this intermediate and react with 2 more the pharmaceutical precursors to arrive at the drug much faster. So if we can develop any chemical technology that we can go through this shorter iteration process, that it will not only cut the synthetic steps to making a drug molecule by 1 or 2 steps, but more than half, right? That will provide us faster access to drugs, will generate less waste, and will be environmentally friendly. And more importantly, it will be sustainable in the long term. Like just imagine the COVID-19 situation, we're trying to get drug molecules, right? And most of the time, these chemicals don't work as drugs, right? You have to go back to the lab, synthesize a lot of compounds, do the testing, and again, do the structure activity relationship, come back, and then make more molecules, right? Eventually, hopefully we can get something for COVID-19 as well. So, and to be more specific, right, what we are trying to do, right, based on what I showed you as the schematic in the schematic diagram, is that we're trying to take this alkene in an unsaturated in a compound, right? So it's kind of acts as the first precursor I showed you, and it is already primed for reaction, and then we'll react that with organic halides and organometallic reagents, two of the most common chemical reagents in organic synthesis, using a transition metal catalyst that I showed you already in the periodic table, using cheaper metals like iron, cobalt, nickel, or copper, and form a new complex molecule that will have two bonds installed in the same step. And with the current technology, most of the steps that we use in making pharmaceutical molecules is it requires one step at a time. So here we're trying to make these compounds with two steps two bonds in one step. So just to give you an example of what we have so far done in our lab toward this goal is that this is one of these examples that we have developed. So where you take an alkene, there's one component, and take an organometallic reagent, that's a second component, and there is an organic halide, that's a third component. So this basically are three precursors toward a drug molecule. For example, if you want to use this process toward synthesis of the drug, and you make this complex molecule, that will eventually look like a drug molecule, not necessarily the compound that we are making at the moment, but you can use this technology to make a wide varieties of chemicals that will, you can either elaborate them to make drug molecules as natural products that have bioactivity. And just using this method, just to showcase what we've been doing toward making drug molecules, this is a potential flap inhibitor that Mark synthesized and tested in 12 synthetic steps, right? If you look at this molecule, this doesn't look that complicated to any organic chemist, but it requires 12 synthetic steps to make it. And you can look at who made it. Mark made it, right? That's a pharmaceutical company that everybody knows. Now, we recently developed a chemical technology that actually makes this potential flap inhibitor just in 5 steps, cutting down not only by half, actually more than half the steps are cut out. It's coming down from 12 synthetic steps down to 5 synthetic steps. So this is what we are imagining the way we're going to make pharmaceuticals in the future, right? So there is another direction that we are also taking in terms of more specific transformations, is that rather than having 3 reagents in 3 separate components, we can also attach one of them into for example, to one precursor here that is an alkene, and may do the reaction in a 2-component fashion, again with the transition metal. So what will this allow us to do is make complex carbocycles and heterocycles that are widely available in natural products, right? And natural products and pharmaceuticals.
5 minutes to wrap up, please.
Sure, on time, thank you. So and then we recently used this technology, right, to develop. We developed this technology to make a large number of the heterocyclic backbones that you can now imagine using this kind of backbones here I'm showing in red that we can easily access through our chemical technology, forming 2 carbon-carbon bonds shown in blue. So these kind of structures have a wide range of biological properties, including antibiotic, fungicidal, antiviral, anti-HIV, and so on and so forth. So, and these kind of compounds also occur in natural products, and the lignin natural products can have a wide range of biological properties. And here you can see more simpler form, and you can also convert this there's a ready structure in here to more complex natural products that have biological activity, and eventually some of them can lead to forming a drug lead compound. So in this business, we recently synthesized a dimethyl rhododendron. Is a natural product derivative that actually comes from pine trees. That this kind of molecule have been synthesized in the literature in 8 to 12 synthetic steps. We've been recently been able to make that just in 2 synthetic steps using our chemical transformation here by using simpler precursors, doing our chemistry, reach this intermediate much faster, and then the next step we make the natural product. So we've also done the same with another natural product, codenicin, which turned out to be the first synthesis in the literature. And looking at the structure of this complex, you can easily see that it might take 8 to 12 synthetic steps, but we just made it technically in 2 synthetic steps. But if you look at the synthesis of one precursor in another step, technically in 3 steps. So with that, what I want to tell you today as a summary is that we've been able to put together 2 or 3 precursors at the same time that will eventually be able to lead to the synthesis of drug candidates or drug molecules. And we've also been able to make some natural products and bioactive molecules using our chemical technology. So with that, I want to thank my research group that does most of the group, past group members and current group members, our collaborators on the side, and more importantly, the funding agency, the National Institute of Health, National Science Foundation, Penn State, and so forth. And again, I want to thank you guys, from the NRN Knowledge Global Knowledge Convention for the invitation, and Dr. Basnis Sali for reaching out to me. And I'll be more than happy to take questions. Thank you.
Thank you. Thank you, Professor Ramesh. It's a very kind, it's very good to see you in this presentation. Actually, he is also my colleagues in Tribhuvan University. We had the masters and we did the same organic chemistry major. And he is still doing the same things and in a high advanced level. That's really good. And I think your presentation would be really helpful to the people who are working on the same field with a very fast and rapid way of synthesis. So I want to open the floor for the questions. Any of us, if you have questions, please go ahead. We have a few minutes left.
Hi, Ramesh. Can I ask?
Are we looking at the Facebook Live? Well, you can just type the questions, or you can.
I'm monitoring Facebook Live. We don't have any questions in Facebook. I think Dr. Raju has a question here.
Yeah.
Okay, please.
Yeah, thank you. That's an excellent talk, Ramesh ji. This kind of chemical transformation is the way forward technology. I was wondering that can you do a sort of a parallel synthesis, high throughput synthesis, this kind of chemistry in more robotic way rather than,
So yeah, I mean, that's a great question. Thank you, Raju, for bringing that up. I was actually just asked the same question that I had to give this talk, not the exact same talk, but a different one to the chemistry department, just actually on Thursday. So yes, I think eventually we should be able to do these things in high throughput, platforms, 96 well plates or more that a lot of people are doing. We're inching closer to doing that in our own synthetic lab. But this transformation is only 3 or 4 years old that we developed and along with other people in the field. It's probably going to take a little bit longer time in terms of logistics and everything to do in a high throughput screening fashion. But these are the reagents that we use, very simple ones that are all, and one of the difficulties in the high throughput screening is like, how are we going to put all those reagents? Like some of them are solid, they don't dissolve and all those things, right? So But these reagents are, we're trying to minimize the number of reagents we use and, focus on putting together multiple precursors at the same time. So I think eventually it will be, but as far as, I know that nobody has used high throughput screening for this kind of transformations.
Excellent work. Thank you.
Any questions further? Well, if we do not have question, then I have one question. Dr. Giri, as I see the presentation slides that you are working on some metallo catalyst, right? So to shorten the synthetic pathway, there is another synthetic pathway. I mean, another way the people are trying to reduce the synthetic pathway by using some genetic engineering, like particularly those steps to reduce the dosage steps, which has a very complicated stereochemistry, are difficult to synthesize and having a very low yield, even we can synthesize. So are you thinking of doing or finding some alternatives in the case if you have some issue in synthesizing stereochemically pure compound?
So yes, that's a great question, Devi. Thank you for bringing that, right? And always more is better, right? So if you have more technologies coming in to make this drug molecule, is always always great. And genetic engineering has its own strength to make this kind of molecules. And the last year the Nobel Prize was in chemistry was given to that area. But that area still has a long way to go. And I think, what I think, and I may be really biased being an organic chemist, is that it will take a long time before that field matures to a point of making actually drug and supplying to the world population in large quantities. And even if it really does, right? And we know how nature does. These things gonna do exactly the way the nature is doing, right? For hundreds of years, but we are trying to tune it to our own, making it to our own chemicals, right? And first of all, these, the genetically engineered things will be more specific to one type of chemicals. The making large libraries is going to be always difficult. You have to go back and through these iterations of optimizing it. And eventually, right, it's not going to do any better than the nature does, right? So it's always going to do what nature does. It's just a modification. And if you look at nature, like penicillin, we used to get penicillin from bugs, right? Is that enough to supply the demand, meet the demand of the world? Right? It's always, I think, comes down to large scale synthesis that you have to make in as I said, volume time output, right? So yes, that would be a great technology. More is always better, but we have to make a synthesis in a short, specific way. The organic synthesis is always going to come on the top.
Thank you.
Hello. Yeah, I have a question,
hello.
Doctor, for Ramesh Giri. Do we have a minute of time? Just
Yes,
a short, just for a short question.
Yeah.
Yeah. While we are talking about the natural products from different sources and or microbial sources, so have you ever thought of using the compounds, the natural products, just to modify them to increase their potency for whichever application?
I'm looking for people who can take our chemicals and use them. I'm trying to find some collaboration. I'm in touch with one of my colleagues here, but if you have anybody, I would be more than happy to collaborate. We are making these heterocycles. We know heterocycles are very critical in pharmaceuticals and bioactive molecules, lead compounds, and all these things. We just shelve them at the moment because their lives, is totally different from what people use in testing, right? So we're looking for collaborations, and if you have anybody there, we would be more than happy to do so. That's a great question, and this is where organic people waste a lot of compounds, and I'm an organic chemist, right? So there's a disconnect there, right? It's always, and it goes with all these the other political stuff that comes with the university and research centers, right? So, but yes, we are more than happy to collaborate with anybody who can actually use and test for any diseases or any bioactivities. Thank you.
Excuse me, I have one question, please.
We have
Hello.
Hi. Oh, Ganadai, namaste.
Namaste, namaste.
Please,
Hello, organizer.
Please
please
go
go
ahead.
ahead.
Please go ahead.
Okay. Okay. First of all, congratulations to Dr. Giri for
Thank you.
tremendous success, and he's doing actually very good. Just simple question. So in your slides, you have shown that some substrates that is used in your reactions, you have used either the amines or you have used some substrates that is already constituted of the metal substrates.
Yes.
So I think that preparation of these substrates is also tedious. Then why you choose this type of substrates in your two carbon-carbon bond formation reactions?
That's a great question, Ganaji. Thank you so much for bringing that up. I guess I didn't choose the right substrate for my presentation. We have solved that problem. We have other examples that we published recently that you don't have to functionalize them, or you don't have to even take any activated alkenes. You can just use simple ketones or esters or nitrites, just like you would get it from Mark or Sigma Aldrich, and functionalize them. So, and we have, done some work on completely unactivated alkenes that you wouldn't even need anything to direct the metal. So yes, that's a great question. I should have chosen my example there more specifically, knowing that there will be people who can look at these slides more closely and scrutinize. But yes, we have solved that problem. Currently we have one manuscript under review at JAX, and the other one is submitted to Nature Chemistry. Yes, so we are solving this issue. Thank you.
I found one question from Facebook, Dr. Roshan. Just 1 minute, please. Let it clear in 1 minute. The question is, how expensive does it get compared to natural product isolation? Is from Neeraj Arial questions from the Facebook, please.
Yes. So natural product isolation, I've done natural product isolation when I was in TU, right, for my master's thesis. So, so it depends on how you look at it, right? If you're trying to get a small scale and you know exactly where that compound is, then early on, if you just can collect their compounds and isolate, like for example, Penn State is well known for developing the sex hormones synthetically going into Mexico and isolating some intermediates from plant materials, some steroids, and in one or two steps making that actually beat the Merck process at the time. But in the grand scheme of the whole world and the population, right, you want to be able to make them in a set of labs rapidly, and then you can. Nature has its own way to make. It has its own pace, so that you can increase that pace, right, to make more chemicals. But in synthetic lab, you can develop more process, you optimize, and you can make them faster. So I think eventually it will be the synthetic lab that's going to provide. But again, more is better. If you can get access to from chemicals from natures early on, that's great.
Thank you.
All right. So we'll move on with our second speaker. Our second speaker is Dr. Indira Tiwari. Dr. Tiwari is a postdoctoral fellow at Department of Infection Biology, Wong Kang University School of Medicine. She received her PhD degree in molecular virology from Pusan National University. Dr. Tiwari has also received the fellowship in clinical embryology from Creation and Love Women Hospital, Gwangju, Korea. Her title of today's talk is not about embryology, but she'll be talking about the Toxoplasma gondii, its advances, challenges, current status, and public health significance. Dr. Tiwari, over to you. You are mute. Please unmute yourself.
Thank you, Dr. Roshan.
I will interrupt on 15th minute, and then
okay,
you can
sure.
finish.
Okay.
Thank you.
Thank you, Dr. Roshan, for the elaborative introduction. So I would just like to go with my presentation directly. So can you see my slide?
Not yet.
Can you see it now?
Not yet.
Really?
Yep.
I can see.
Can you just select the PowerPoint once you open it?
Yes, just a minute. I think. Can you see it now?
Yes. Yes.
Yeah, go ahead.
Yeah, very good.
So are you able to see my slides?
Yes.
Yeah. Okay, thank you for the time. So today I will be explaining about my ongoing present work that I received the fellowship from the government of Korea. So this time my topic is Toxoplasma gondii, advances, challenges, status, and public health significance. So Toxoplasma gondii is a parasite, and it was first isolated in 1908. And it is estimated to infect upon 1/3 of global population. And this parasite especially love to grow and multiply in the muscles and the brain cells. So if we see this parasite, it has 3 different forms. So tachyzoid, bradyzoids, and the cyst. So these are the pictures that I am presenting here from my own research work. So these 3 different form of the parasites, they can be converted from one stage to another according to the host. So these are the very clear pictures. And if we see how this parasite get transmissions, so this parasite is infected by the oral ingestion of the contaminated food product like the meat and the other kind of salads. So when the parasite is ingested, it get penetrate to the epithelial cell, and then it enter the interstitial cell. So once it complete its sexual life cycle in human or the cats, it is released from the host after the 3 or 18 days of the infections. And now if the cat or any cat or livestock is infected by the cyst, the species of the livestock is now very dangerous because it contains the Toxoplasma gondii. So if we see the epidemiology, it is foodborne. It is foodborne disease. So we will have different kind of outcomes. So we will have congenital transmission, zoonotic transmission, and other rare instances are apart from the congenital transmission. So we will have the Toxoplasma gondii, the organ donor also might get the toxoplasmosis, or after the blood transfusion, or the laboratory worker who are working in the lab, they might get infected by the Toxoplasma gondii, and the normal person get contaminated or infected by consumption of the food. So if we see the clinical features, these are the more significant clinical features. First is the congenital toxoplasmosis, toxoplasma retinochoroides, and acquired toxoplasmosis. So if we see the global status of Toxoplasma gondii, all the 5 continents, they are infected or they have some seroprevalence of the Toxoplasma gondii. And if we see here in context to Nepal, we have around 40 to 60% of the presence of the Toxoplasma gondii. And if we talk about the Toxoplasma gondii and economic burden, so this Toxoplasma gondii is not only very dangerous to human, but it is also very dangerous for the livestock like the sheep, horse, goat, and cow. And in the US, among the 15 foodborne pathogens, Toxoplasma gondii is the second most common, and US only spend around 3.3 billion each year in the treatment of the Toxoplasma gondii. And it is estimated around 60 million people in the US are infected with the Toxoplasma gondii. So, and what are the recent diagnostic techniques? There are the different type of Toxoplasma diagnostic method, and these are the diagnostic methods here. And in most common, we use the ELISA technique because it is very easy and it doesn't require the trained manpower. So if we see what are the recent advances in the Toxoplasma gondii. So the Toxoplasma gondii is mainly very contagious to immunocompromised patient like the organ donors or the HIV patient or the pregnant woman and the normal patient. So in the immunocompromised patient, we do, especially in the serological test, we do IgG and IgM test by the ELISA. So these are the different type of test and the form how we test. So I will not go on detail in this one. So this is just the flow chart how we determine the toxoplasmosis in the different patients case. And if we have the diagnostic criteria for that one. So we check the IgG and IgM and IgG avidity. So we basically check what is the state of the toxoplasmosis, whether it is acute or chronic or subacute. So depending on the value of IgG and IgM, we determine the patient state of infections. So how can we prevent the toxoplasmosis? This is the CDC. I take this picture from the CDC, and it has they have clearly shown how can we prevent the foodborne diseases along with the toxoplasmosis. So for very simplification of this chart, what I want to say is, if we have a farm sanitation, if our livestock has a very good sanitation, we have very less chances of toxoplasma contamination. And the second is the general people, they need to freeze their food in minus 20 degrees. That is, the freezing of the food is very important because these days we are consuming a lot of meat. So before preparation of the food, it should be stored at minus 20 for longer time, and it is recommended more than 48 hours. And the other thing is, if we are eating the vegetables which cannot be stored for a long time, the proper cooking is recommended. Now, my study is mostly focused on the diagnosis of toxoplasma in the pregnant woman. So I would like to discuss what are the cases and why this study is more important. So as I told, the pregnant women are more vulnerable to toxoplasmosis. Why is during the pregnancy test, we do the TORS test, and TORS, the T means the Toxoplasma gondii. So if the pregnant woman is found to be IgG or IgM positive, or IgG positive or IgM positive, so it determines what is the status of her Toxoplasma gondii. If she has the IgG, then we can preclude that she has the infection prior to her pregnancy. If it is not missed, we can say that she got infection during her pregnancy time. So in this case, we need to do continuous testing and get the medications here. And the second one is, since she's pregnant, it is not only affecting the mother, but it is also affecting the child inside her womb. So we need to determine whether the child is infected or not. So in these cases, we determine the infections by the detection by testing the amniotic fluid. And if the amniotic fluid seems to be positive in ultrasound, and we need to do continuous screening of the mother. And if the mother is found to be positive and the amniotic fluid is also found to be positive, then in that case, the clinician need to determine whether to continue with the pregnancy or stop the pregnancy. And if it is not positive, we still need to go for the postnatal follow-up. Now, after the child is born and after the seroconversion, the child still need to go for screening test. And if in the first year of his life, if the child is found to be IgG positive, then what we say is the child is having the congenital toxoplasmosis. And if his IgG level in his blood is negative, then we confirm that the child is without congenital toxoplasmosis. So apart from this kind of multiple complication, now recently different studies shows Toxoplasma gondii is not related to only clinical disease, but what the research has found is Toxoplasma gondii increase the risk of self-directed violence. So, and in Korea also there are multiple reports saying that the seropositive, T. gondii seropositive patient, they have found to be more violent, and then there is a higher risk of suicide rate in the T. gondii positive patients, because the T. gondii is multiplying in the brain. So there is neuronal defect. So there is a very high chance of the suicide rate. And there are multiple cases that above 45 years old patients here in Korea, they have found to be doing the suicide here. Now, I just want to talk about the toxoplasma. What is the status of toxoplasma in Nepal? And I went through some papers, but I couldn't find much paper related to toxoplasma gondii. I could find some papers, and in some paper they have found that I found one paper that described the 12 pregnant ladies. They were tested for toxoplasma gondii, and among them 50% they were positive. And in the slaughterhouse in the Kathmandu Valley also, they found that the toxoplasma gondii is around 50% positive in the slaughterhouse. Similarly, one study saw that the sheep also found, in sheep also they found that toxoplasma gondii was 36% of the tested samples were positive. That means we are at the high risk of toxoplasma gondii in Nepal. Why? And from this, what I can conclude is 50% of the Nepalese pregnant women are seropositive, and then the T. gondii seropositivity is the indicator of hygienic status of the livestock in stock production. That means we don't have the hygienic style of the lifestyle production. That means we are still in the danger zone. So now, what are the limitations of the diagnostic approach in the toxoplasmosis? At present, the diagnostic kit what we have, or the diagnostic test ELISA kit what we have, they use the total parasite lysate, and that doesn't show the accurate result whether the we can just confirm that the patient is seropositive or negative, but we cannot know at which stage of the infection is the patient. Why? Is because we are using the total lysate of the parasite and it doesn't indicate the stage of the infection. And apart from this, we are using the crude lysates, so it doesn't indicate, there is not a standard level of the test, because there is the difference in the products under the parasite here. So what is needed is we need to have a world standard in the diagnosis of toxoplasma gondii. From America to Nepal, we need to have a standard toxoplasma diagnostic kit. So what is the next things to be done in this field is we need a recombinant antigenic protein of toxoplasma gondii, so we can establish a standard gold standard for detection of the toxoplasma gondii. And now at present, I am doing the detection of the toxoplasma gondii. We are designing a way to detect the toxoplasma gondii on the stage, like the tachyzoid stage, bradyzoid, and the oocyst. So what we are using is, now we are using the DNA recombinant DNA technology. So what is this? In this step, what we are doing is we are isolating the antigens from the tachyzoid and the bradyzoid stage of the parasite, and we are going to check the broad spectrum of the antigens expressed, and we will be selecting the different antigens that are seropositive, that are positive with the serum, and then we will identify the antigens which are positive, and we will be developing the antigen, or we will be preparing the antigen in the lab. So we will be using the cloning technique. This cloning technique, we will purify the antigen and we will be developing the monoclonal antibodies. So why we are doing is we will use this antigen and the serum developed in vitro developed serum to make the immune assay systems to confirm the stage specific detection of the toxoplasma gondii. So if you see what is our approach is, we are trying to develop a diagnostic kit or the strip that will be very accurate in determining the acute or the chronic phase of toxoplasmosis depending on the antigen. So what is our approach is, in the previous we used the whole human serum to detect the parasite, and that the ELISA plate there was the total parasite lysate, and this method, it couldn't specify what is the stage of the toxoplasma, and this stage was very less sensitive. Now, what is our proposed program is here? If we use the recombinant technology and have a recombinant antigen, we will have a standard sample every time. So we can easily differentiate between the acute and chronic phase. And apart from this, the parasite culture is very difficult in the mouse model. So in the in vitro model, we will be able to generate lots of parasites, and that is very easy and cheap way, and that is giving the very sensitivity and specificity is also very high.
You have 5 minutes to wrap up, please.
Yeah. So in this slide, what I want to conclude is toxoplasma diagnostics present and future. So what is the limitations of the present method is we use there are different companies, they use the random antigens. Like one company is using one antigen and the other is the another company is using the another one. So there is the absence of kit that can distinguish the acute and chronic phase. Now, what is our innovations, or what we are trying to do in our lab is that we are trying to identify the stage-specific antigens. So this is the method how we are going to use and develop a diagnostic kit. We are not only doing the molecular work, but our final goal is to develop the kit that can be used worldwide. So what will be the outcome of our project is we are trying to develop a kit that is rapid, accurate to diagnose the disease, and that is very time and effective treatment of the patient. And the second thing is recombinant antigen. They are very effective in terms of cost and quantity. And also what we can do from this is we will be able to create the gold standard across the globe. So what is the take-home message from my presentation is toxoplasma cause the toxoplasmosis is a disease caused by Toxoplasma gondii, and this parasite, it successfully infect about 1/3 of the world population, and it is not only its primary infection is usually subclinical, but in immunocompromised patient it is life-threatening. And women's and animals, not only the women's, animals also who get infected by T. gondii during the pregnancy, there is a chances to have the stillborn, abortion, or the neonatal abnormalities. If we take a look at about the US, about 10% of the patients in the US and 30% in the Europe are estimated to die from the toxoplasmosis-related complications. So what is the thing is, what is the recommendations for the developing country like ours is we lack the modern farming, and we have open air market that increase the risk of domestic and imported meat. That means we have the chances of transforming the different strains of Toxoplasma gondii and then developing the toxoplasmosis. So there should be a very strict monitoring system by the government to intervene the disease transmission. And the recombinant DNA technology can be used to generate the very cost-effective antigens that can be used for the detection of the T. gondii. Thank you. And I will be happy to have questions from all of you.
Thank you, Dr. Tiwari. Now we will open the floor for the questions. If we have any questions from the participants in Zoom, and also we'll monitor the Facebook comments.
Yeah, Dr. Tiwari, it's a nice talk, and something about that we are not very aware of. So my curiosity is, what is the prevalence of this disease, toxoplasmosis in men? Because most of your talk was focused on women.
How can we prevent?
What is the prevalence in men?
Men, usually there are also almost, if we see the infections rate in men and women, there is 90% chances in the women's, but 10% in the men. But what is the recent finding is, one of the paper is reported from India in the goat. They found that the Toxoplasma gondii is also found in the semen of the goat. So much of the study is much more focused on the women's because of the pregnancy related issues, but it is not related to men. But if we see the studies in Korea, the men who are residing in the area nearby the forest, they are more prone to do suicide because of the Toxoplasma gondii infections, because they eat the raw meat. So it is not, the disease is not related to age or sex. It is related to the exposed to the Toxoplasma gondii.
And what are the very visible symptoms of this disease? Is there any?
Actually, it is a very asymptomatic. Asymptomatic. Because only in the immunocompromised patient only we can find some symptoms, but in the normal it is very asymptomatic. So it is very, very difficult. Usually the women's also have more complications, but they only came to know during their early pregnancy torch. During the examination of the torch examination only they comes to know, and that is the very late stage of the toxoplasmosis.
Right. Okay. Yeah. Thank you.
We have another question in Zoom. This is from Nirmal Marasini. Just wondering about the role of immune system in toxoplasmosis infection. Does some population has inherent immunity against it? Any updates on vaccine development?
Actually, there is no, actually the person who are much immunity, if the immunity is good, the parasite is going to remain silent. But if you are immunocompromised, in that case, the parasite is going to be reactivated. That is why I have shown you the 3 forms of the parasite. So the tachyzoid is the most infective phase. So if the person is very immunically active, the parasite remains in the dormant state. It will be remaining in the body for a long time, for like 30 to 40 years. Once you are immunocompromised, the parasite, which is the bradyzoid or the cyst form, it will be converted to the tachyzoid and it will started to infect you. And most of the time the parasite is found in the eye and the brain and the muscles. So we have the complications related to neuronal systems and the retina and the muscles. Most of the cases are related to the brain and the eye.
From Nirmal Marasini ji again. So does your idea of diagnosis of toxoplasmosis covers all serotypes?
No.
Okay. And one more question from Dr. Ajit Karna. It will be an ambitious goal to have modern farming in Nepal. What do you see practical solutions that farmers in Nepal can do to prevent it?
In case of Nepal, toxoplasma can be prevented if the water system is very good. So if we are using clear or good quality of water for the farming, there is very low chances. And if we have wild animals coming to the agriculture area also, there is the chances that your crops will be infected by the T. gondii, because the wild cat, they are the carrier of the T. gondii.
Okay.
And we cannot control the cat to roam here from your farming area. So if we are using the good quality of the water, which is not contaminated by the feces of the wild cat, then there is a chance we have. And the other thing is we don't have a good slaughtering system in Nepal, and the water quality is bad. And we have a culture of eating the raw meat.
Right.
Yeah, we have the culture of eating raw meat without freezing. So that is the reason why we are seropositive in the toxoplasmosis.
I'll take one
prevent
question.
This. Yeah.
I'll take one question from Facebook and then I'll come to Dr. Giri. So in Facebook, we have a question from Neeraj Aryal. Why is someone having suicidal tendencies if it's infected with toxoplasma?
Because there is a defect in the neuronal signal. I think it is there is defect in the neuronal signals that will have the patient will have a tendency to have a suicide, because it disrupts the mental system.
Right. Thank you. Dr. Ramesh, you have.
It was a nice talk, Indiraji, and very enlightening, information on that. So when I look at a chart, right, it shows the mostly it is in the developing countries. Is that correct?
Yes.
So is it mainly related to sanitary, as you mentioned, and is also the occurrence of that T. gondii is less in developing countries versus natural occurrence of these bug?
I would like to give the reason why it is more in developing countries is if you see the trend of rearing the pet animals like cat is more common in developed countries. And the second thing is they are eating more protein, the animal products. And I told freezing is the best way, but still freezing cannot control the cannot control completely kill the toxoplasma gondii, because when I have my own personal experience. When I freeze the sample T. gondii parasite in -80 and still the parasite, some of the parasite, they are alive. That means still the freezing is not enough. So when we cook the meat or when we boil the sample for 10 to 15 minutes, what we found is the parasite is lysed, or some of the parasite, they are inactivated.
Thank you, Indiraji, for your nice presentation. We have 5-10 minutes delay. So thank you for your presentation. Rosanji, next.
Yes. So if you have any questions, we can discuss that in the break time as well, actually.
Okay, yeah. Let me introduce another speaker. We have another speaker, Dr. Asim Dhakkal. Dr. Dhakkal is an engineer by training with PhD degree in photonic engineering from University of Ghent, Belgium. Dr. Dhakkal is a co-founder and chief scientist at Photon Research Institute. Today, he will be talking about the application of photonic technologies in biomedicine, and his title is Nanophonic Technology Being Developed in Nepal. So we are in the session. Of approach to the biomedicine. So I want to go for Dr. Asim Dhakkal. I think IT guys should help in here. Please go ahead. Thank you. Nisanji?
Yes, sir.
Yeah,
you
please,
play my video? I think it will be easier to play the video because I have a call and my internet is not very stable. So.
And photonic technologies being developed in Nepal. So, I thank the organizers for this opportunity. In this talk, I will discuss three things. First, I will try to explain why we need to focus on the cutting edge or advanced research, contrary to the common assumption that we should not, because we cannot afford to do that. Second, I will discuss a new type of nanophotonics device called on-chip hexagon-like lens, which we are developing in Nepal. Third, I will discuss a systems-level implementation of fluorescence spectroscopy to detect harmful contaminants in drinking water. I will not discuss challenges and opportunities, as I expect this has been or will be discussed adequately in other sessions. Let me start with so-called Global Innovation Index. So this index is based on several indicators to measure the health of the research, development, and innovation ecosystem in different countries. According to this index, not surprisingly, Nepal is in the red zone in the globe, indicating its poor innovation ecosystem compared to the rest of the world. After being ranked 108th, 109th in the previous years, Nepal has jumped to 95th this year. So this was touted as a major achievement by several people involved in this area. However, the reality is quite different, as seen in this graph, where all indicators except for the business, market, and investment environment are quite poor. Our rank seems to be clearly biased by the business environment rather than the innovation outputs. So nevertheless, all of this indicates potential investment opportunity should we innovate, which is good for, which may be good for innovation. As clearly indicated by our very poor rank in the knowledge impact metric, as shown in this table, there is no room for satisfaction for the scientific community. Also, it is important to note that R&D output indicators seem to be poorer compared to the R&D input. So this does not bode well for the long-term development of the innovation sector, as it discourages investment, even though we have a good investment indicators. So therefore, clearly, we need to work on knowledge impact, which can only be enhanced by research at the cutting edge. So this is the main reason why we at the Futon Research Institute focus on research at the cutting edge, in spite of the common pessimism in this concerning advanced research. At Futon Research Institute, we mainly focus on advanced nanophotonics and optical technologies. We have Nepal's only nanophotonics and photonics lab. The lab is world-class in the sense that we have state-of-the-art infrastructure for design, simulation, and measurement. So here, so these are the chips we sometimes brag for being Nepal's first sets of advanced nanophotonics chips. I will discuss about one particular component of that is of the chip. So in the slide, you can also see the chips when compared with a 1 rupee coin. So you may ask, what exactly is nanophotonics integration, and what is the technology we have been developing? I assume that you are aware of the electronics integration, integrated electronics. Basically, all of our mobile phones, computers, they have these electronic chips. So the photonic chip is similar. So in an electronic chip, electrons or current runs through the structures in the chip. The idea is similar in nanophotonic integration or photonic chips, where, however, light runs through the structures instead of the current. Using the integrated photonics technology, we are trying to miniaturize an optical coherence tomography system, which is currently a benchtop system. So we are trying to miniaturize the OCT system into a very small size of 1 by 3 millimeters. So the benefits of this integration, like in the case of electronics, is that it will have a smaller size, it will hopefully be more efficient, low cost, and it will be portable so that the system can be taken to the point of care, for example, in remote villages of Nepal. So now I will discuss a key component of the on-chip OCT that was there in the chip I showed before. So in a traditional free space setup, so-called hexagon should be used. The hexagon has a conical surface on the one side and the planar surface on the other side. So when the light beam impinges from the planar side, the light refracts to the axis. So the light refracts to the axis, creating a focus of long length on the axis due to interference effects. So the resulting long depth of focus allows for scanning deeper tissue structures. So in the case of a photonic chip, where the light is largely confined within the chip, however, this traditional approach based on free space light cannot be used. Cannot be used, and also the delivery of light to the tissue and collecting the light back from the tissue to the chip. So delivery of light from the chip to the tissue and back from tissue to the chip is a very challenging problem in integrated photonics. So to address this problem simultaneously, we have developed an on-chip or chip-based hexagon lens, which works on the principle of diffraction of light, like in the gratings. So here we saw our design, the microscope image of an actual lens and the expected behavior on top of the photonics chip. So the details, I'm not going to discuss quite a lot of details about this particular work because it's available in the archive. So if you are interested, you can look at this. And this work is currently, the manuscript is under review. Here is the measured behavior of our chip, our exicon, which we measured in Nepal, of course. So we have a central lobe of 5.7 micrometer diameter, which spans over about 1 millimeter. So which should be sufficient to penetrate 1 millimeter depth of the tissue, hopefully. So in this slide, I'm sort of trying to give a glimpse of how the design and measurement are interlinked. I hope you have appreciated that we have developed a nanophotonic design and a measurement ecosystem in Nepal. But I have to tell you that we do not have the facility to fabricate the chip itself. So this is largely done by our partner in the UK by Cornerstone Project. The fabrication of chip is a very expensive enterprise, and it needs a long-term big investment from the government, most likely. But rest of the things we can do it in Nepal. We have been doing this in Nepal. All right, so now I move on to discussing another project where we have been developing a fluorometer to detect contaminants in drinking water. So when exposed with a UV light, almost all the minerals and biochemicals exhibit fluorescence in different colors depending on their molecular composition, as you can see in the figure there in the left. So the key idea of this project is that the specific impurities in water can be detected by measurement of the light of the fluorescent light at some specific fluorescent band. So this is a schematic of our basic setup. So what we essentially do is we focus the light from the UV LED into the water sample, and the fluorescence is then collected and then detected by a photomultiplier tube or a spectrometer using a series of lenses, optical filters, gratings, and mirrors. So this is schematic just to give you a flavor of the optical design, not exactly what we have done. It's a bit complicated than what I've shown here. So this slide shows the measured calibration curve of our setup. The calibration is done using tryptophan amino acid, which is indicative of the biological activity in the water sample. So in a natural state, if we detect some tryptophan, it indicates that there is some bacteria, there is some biological activity going on there. That's why we choose tryptophan. So as you can see, our setup can measure as low as 100 parts per trillion in water. So this detection limit is better than most of the commercial fluorometers available in the world. So tryptophan, so these are the fluorescence spectra as measured with our system. As you can see, we can measure the spectra of tryptophan as low as 1.25 parts per billion in water. So here we have attempted to to measure these fluorescence from the actual groundwater samples taken from different locations in Kathmandu Valley. The yellow bar labeled as drinking water, which exhibits the lowest fluorescence, is from our reverse osmosis filtration system. So water from the groundwater from several places seem to exhibit fluorescence corresponding to tryptophan of concentration 7 to 28 ppb, while that of the RO system is lower than 1 ppb. So we are currently working on correlating these findings with microbial studies. So this is an ongoing work, and hopefully in the next conference I can discuss more about it. So with this, I would like to end by acknowledging our research partners, our funding agencies, and our sponsors. So without whom, of course, this whole research would not have been possible. So with this, I thank you, the audience, also for listening, and I'll be very happy to answer any questions you may have. So thank you very much. Thank you very much. And I would like to end this video.
Thank you. Thank you for the presentation. Now let's have questions from the floor. Yeah, please go ahead.
Can I ask a quick question?
Yeah, please.
Thanks for a very nice talk. I guess I just have a small question toward your, the second talk, the detection of tryptophan. So I know you can detect down to 100 parts per trillion, right? But is that with a specific chemical, or like if you have similar compounds, right? For example, let's say, this is hypothetically tryptophan and 5-HT5-hydroxy tryptophan. So how well can you differentiate these two? Because you might have different types of similar chemicals, right? All these proteins coming from proteins, all the amino acids, right? So what is the how specific is it? How well can you differentiate?
So tryptophan. So we use tryptophan as just a calibration chemical, but the idea is to detect not tryptophan itself, but the tryptophan like fluorescence. So this is called TLF in the standard. Yeah. So it may contain, for example, indole or some other derivatives of indole. Tryptophan is one of the derivatives, and the idea is so there has been a lot of studies which correlate tryptophan TLF fluorescence with the number of E. coli bacteria, for example. So there is a very strong correlation between this, and this may come from the TLF, may come from tryptophan itself, indole, or several other derivatives of indole. So it's not very specific. It's specific in the context of excitation wavelength and emission wavelength. But the rest of the things, there might be some other disturbances from, for example, from plastic bottles. So in our study, we have seen there is some interference from plastics, for example. So this is something we are trying to optimize.
Thank you. That's a really nice talk. Yeah. Nice work.
Thank you very much.
We don't have questions now.
There's one question in Zoom. Nirman Marasini asks, how is tryptophan fluorescence could be correlated on water contamination?
Yeah, so this is something already been doing in context of groundwater in Africa. They are also trying to do similar work and have found a very strong correlation between E. coli and TLF. Not tryptophan itself, but the TLF. I should tell you that actually, so whatever I have shown, another big project. Basically, we will not be going, with the setup that we have, saying, this is something. Actually, the idea is to miniaturize all of this fluorescence setup into a chip. So this is something I have not talked about in this talk, but so the idea is to have everything on chip so that it can be sold, it can be used by everybody at a consumer level.
Okay, we are running a little bit late. I think we should move on to our next speaker. Okay, our next speaker is Dr. Deepak Upreti. Dr. Upreti is an immunologist by training with the PhD degree in immunology from University of Manitoba. Currently, he is working as a senior scientist at the Century Therapeutics and McMaster University. Today, Dr. Upreti will be talking about one of the most exciting fields in cancer biology, and that's also one of my favorite areas, that is cancer immunotherapy. So his title of the talk is Application of Chimeric Antigen Receptor with CAR T cells in cancer. Yeah, please go ahead.
Can you all share my slide?
It's
Yes.
here now.
Yes.
Yes.
Okay. Thank you to all moderator and chair, especially David and Susla Didi for providing this wonderful opportunity to present our work in this stimulating conference. And before I begin, my thoughts and prayers to all the people who are living in Nepal and abroad who are dealing with this pandemic from multiple levels. So the topic my today's presentation is the applications of chimeric antigen receptors in cancer. So before I start, so due to the confidential reason, I'm only presenting the data that our team has recently published. So this is actually my favorite slides from back from PhD days. So how the cancer cells and immune cells, they interact, I know is very important to understand before we develop or design any therapy against the cancer. So so the interaction between the cancer and immune cells actually says that it has a 3 stages. So the first stage of the interaction between the immune cells and cancer is called the elimination phase, where the immune cells, they can detect the newly transformed cancer cells and they can eliminate these cancer cells. But not always these immune cells are capable of eliminating these cancer cells, because what happens is that sometimes these, because of the mutational pressure, or these cancers are very smart, they can utilize multiple strategy to hide or get away from these immune cells, and they can the equilibrium phase. So this equilibrium phase is the phase which exists for the longest time in the body. So where the immune cells and it's a war between the immune cells and the cancer cells, and it takes significantly longer time, and it depend on the which side will win. That will determine the next stage. So the next stage is called the escape stage, and this is the, so when we hear of cancer or when we see people having cancer, so this is the, okay, I think towards the cancers further growing. यो चाहिँ कस्तो नि, यो स्केप फेज भनेको चाहिँ यो चाहिँ त्यो नारायण गोपालको गीत छ नि, त्यो मन्दिरमा छ कि मूर्तिमा छ माया भन्ने गीत छ नि, त्यो माया कहाँ छ भन्ने कुरालाई चाहिँ एकदम इम्पोर्टेन्ट रोल प्ले गर्छ। किनभने चाहिँ त्यो माया काँडा जस्तो भयो भने, जस्तो यहाँ टिरेकले चाहिँ यो क्यान्सर सेल्सलाई चाहिँ बढ्नलाई मद्दत गर्छ, किनभने चाहिँ यसले CD8 सेल्सलाई रोकिराखेको हुन्छ। तर यदि माया फुल जस्तो भयो भने, जस्तो CD8 T सेल्स जस्तो भयो भने यसले फेरि क्यान्सरलाई चाहिँ रोकथाम गर्छ। So to understand the main concept of the interaction between the cancer cells and immunity. So then why is immunotherapy better than other standard therapy such as the chemotherapy? So the interest, actually this immunotherapy is a biological therapy where we your own patient's own immune systems is given power to control or eliminate the cancer cells. It has the less side effects as compared to the chemotherapy. It's very specific, and importantly, it forms the memory. So that's why it can train the immune system to identify the cancer cells to destroy even if it comes back in later stage or later time. So our body has the white blood cells, and among the white blood cells, one of the important cells that we have is the T cells. And the T cells based approach, what this T cells does is it tries to eliminate the newly formed cancer cells or infected cells. So there are a number of strategies that can be utilized for the T cells, such as bone marrow transplantation. We can extract these T cells from the tumor infiltrating lymphocytes, which is from the tumor itself, or making the cytotoxic T cells like CD8 T cells. Or another strategy which is also going in parallel is the engineered T cell therapy, which is called TCR-based therapy. And today's my talk is on the chimeric antigen receptor T cells, CAR Ts. So what is CAR and why it is required? त्यो भारतीय मित्रहरूले नाम में क्या रखा है भन्छ नि, यो वाइ हामीले किन कार चाहिँ नाम दिएको भन्दाखेरि, so if you think about this, look at this T cells and cancer cells on the side. The T cells to act, it requires two major signals, which is the signal one, which happens through this in context of the MSC molecule, which is presented by antigen presenting cells, such as dendritic cells and macrophage. So it has to be in the T cells should recognize that peptide or cancer's new antigens in terms of this APC. And you have this other signal which is delivered through this, for example, the co-stimulatory domain, which is the CD28 or 41BB in T cells. So if you think about it, to perform, or to detect the cancer cells by the T cells in our body, multiple things must go right. For example, the cancer cells must express these antigens in context of MSC, express this co-stimulatory domain. As that's why the signal 1 and signal 2 both are required. But unfortunately, what happens is that, as I said, the cancer cells are very smart. They can either downregulate their MSC molecules, or they can suppress the immune cells such as T cells, or sometimes the T cells even do not express this co-stimulatory molecule. That's why this T cell therapy, these endogenous T cells may not work. So to bypass this whole system, त्यो नेपालमा हिजो आज त सुरुङ मार्ग बनाइदिएछ नि, त्यो जताततै भिडहरूमा तल बाइपास गरेर बनाइदिएछ। यो काटी पनि एक प्रकारले भने त्यस्तै बाइपास गरेको हो। यो मेन टी सेल्सका जति पनि फङ्सनहरू छन्, यी सबै फङ्सनहरूलाई चाहिँ बाइपास गरेर। So here, if you look at my pointer, now this is the CAR. So what is CAR? CAR is a fusion of proteins. So on the one side, you have this antigen detection, antigen binding domain, which we call single chain variable fragment, which is derived from either you can derive from the antibody or you can derive from other ligands. And these single chain variable fragment will specifically recognize the CD19, for example, or any other cancer antigen that are present in the cancer cells. So if I'm talking about the B cells, cancer cells, then it is one of the antigen marker is the CD19, which is expressed by the cancer cells, and also sometimes also by the normal cells. And then you have this co-stimulated domain, and you have the intracellular signaling domain. So all these domains and the single chain variable domain will help not only recognize the T cells, but upon recognition it will proliferate and it can persist in the tumor microenvironment. That's why it will go and recognize these tumor cells, and using this killing machinery, which is through by the granzymes, perforin, or by the fast mediated killing, these T cells will go and kill these tumor cells. So this is about the basic of the how CAR T cells works. And when I say about CAR T, so all these fused molecules, if I transduce in the T cells, it's called CAR T. If I use the same system in the macrophage, it's called CAR macrophage. Or if I use the same system in the NK cells, it will call CAR NK cells. So CAR is the chimeric antigen receptor. It's a fused protein. So depending upon which cells you use, you get the number. You get the name. So as I said before, that these T cells, so they are engineered to express the tumor reactive receptors. So Dr. Ramesh Giri was saying about this synthetic chemistry before. So this is a synthetic biology in a way. So this is a CAR. So chimeric antigen receptor is a synthetic molecule. So it is comprised of the antigen recognition domain fused to other more T cell signaling domains via the transmembrane region. And one of the major advancement, which is that I call as the bypass, because these now CAR Ts are not the HLA restricted, which means now CAR T cells do not need to depend on the cancer cells which express these antigens based on this HLA. So these are non-restricted HLA based therapy. So when I talk about this history of the CAR T cells, and the reason I put this slide is that, so the research and development, it takes a number of years, before we reach to the clinic or before we see the success. So, there should be a continuous investment and continuous, curiosity to make this therapy or any other treatment. So, if you think about these CAR T cells, for example, in 1998, these preclinical studies of the second generation CAR T cells were started, and in 2006, the first human studies of first generation CAR T cells. So when I say first generation CAR T cells, that didn't have the co-stimulated domain. So that's why that first generation CAR T didn't work in the clinic, because that CAR T cells do not have the signals that can help them to proliferate when it reaches to the tumor site. But the scientists quickly learned that there is something missing. That's why they developed the second generation CAR, which is now have this signaling domain. And whatever we see in the clinic today is the second generation CAR T cells. So as we are all are familiar now, at least with the clinical trial world, with all these ongoing phase 3 and phase 1 clinical trial with the virus. So for these CAR T cells also, it took like almost 6-7 years to do these clinical trials from 2010 to 2017. And in 2017, interestingly, there are two major outbreaks. One is in the technology, one is the SCARTA, and one is the Chimera. So which is the approval of these CD19 CAR T cells against the cancer cells. And because of that, it get a really huge momentum in this field. And there is one recent publications on why this CAR T therapy actually gained the momentum all over the world is because it showed the response in the clinic. So if you look at this patient, for example, out of the 43 patients, almost 25, which is 58% response, that 25 people got this complete response. So, and this when I say the complete response means these patients which were non-responder, which couldn't respond with the standard therapy like chemotherapy or surgery, radiation, and those and it worked in the patients. That's why, because of this imaging performance with these CAR T cells. So this is in terms of the CD19, which is the B-cell malignancy CAR T cell cancer. So because of this imaging result, now people are looking, applying these CAR T cells in different other cancer cells. So this is the cartoon, which is published by the National Cancer Institute. So it shows the overall process of the CAR T cell. So what we do is that we take the, so when I say here, this is in terms of the autologous CAR T cells. So we took these T cells like a blood from the patient, and from there we purify the T cells. And once we purify the T cells, we do this, some genetic engineering for these T cells in the plate, in the outside, in the lab. And once we confirm that, okay, these T cells, now they express the chimeric antigen receptors, and now we expand these CAR T cells using this, expansion method, and we grow millions and billions of the CAR T cells in the plate. And once we have do this, grow these CAR T cells, which is sufficient to infuse the patient, then we go and give back these CAR T cells to the patient. And this, once it goes inside, these CAR T cells, now it will go after the tumor antigen, and it will seek these tumor cells and destroy these cancer cells. But one of the problem, I would say, well, it's not problem because we are in the early phase. But if you look at this cost of the CAR T cells, which I developed by the Novartis or even the another company, which is called Gilead or Kite. So the cost is almost like 475 USD, which is almost, I think, 5 crore or more than that, Nepalese rupees. So it's very expensive. Even in the US, it's not all people can afford, or all people are getting that. Very few percentage of the people are getting to this, getting this therapy. But the but what we learned from these autologous CAR T is that it actually works in the patient. So that's where, like many other companies, they are developing this allogenic CAR T, and which we believe that it will reduce the cost significantly. Companies such as Cellectis, now they are developing these universal CAR T cells, and we think that the cost will be reduced to significantly between $5,000 to $25,000. But now many companies are involved that, and there are many technologies which are developing, and I believe that that will be even way cheaper than what this company is proposed. So as I said, that there are a number of clinical trials going on. That CD19, which is the B cells malignancies, this is one of the mostly many companies are actually competing against this against this little space. But in addition to this B-cell malignancy, there are a number of other targets, for example, multiple myeloma, BCMA, and many other cancers. And also not only the CD19, but also keeping these two like multiple antigens together, like CD19, CD28, and many other antigens are going on. And also in the solid cancer, so which is way difficult to treat than the blood cancer. In our group, maybe I'll present some of the data that we have generated. So we work in the solid cancer field, and it's very, very difficult to treat the solid cancer as compared to the blood cancer. And there are several
wrap up, sorry.
Oh my God. So so since I said that we work in the glioblastoma, so which is one of the most aggressive cancer in the and patients have only less than 15 months if they are diagnosed with the glioblastoma, and work in this patient-derived model, which is so we take the tumor cells from the glioblastoma patients and make this mouse model, and then we develop this therapy CAR-T and BiTEs, and then give it to the mouse to see whether it works. So recently we published this paper in Cell Stem Cells, where we develop these 3 immune modalities, which is we develop this antibody against the CD133, which is one of the target for the glioblastoma, and DATE, which we call the dual antigen engager. On the one side, it will bind to the cancer, and other side, it will bind to the immune cells, such as T cells. And most importantly, we also develop the CAR-T based on that CD133. So what we, since using these 3 modality, we saw that the CAR-T cells is most efficacious, and also in terms of the safety, it's very safety against the CD133 CAR-T cells. So then we saw that look as compared to the between the antigen antibody and the CAR-T cells. So the CAR-T cells significantly improved the survival of this of the glioblastoma orthotopic model as compared to the antibody like DATE, dual antigen engager. So the Carl John, which is one of the leader in our field, he beautifully summarized that there are multiple problems in the solid tumor, which is the trafficking, tumor recognition, proliferation, microenvironment control. I could have gone detail into this, but due to the time, I'm just going fast. And so there are multiple approaches that for at least for the glioblastoma. So since if you give this CAR-T from the in vivo, then the patient has to deal with this blood-brain barrier. So the CAR-T cells may never reach to the tumor site. So what we are developing is we are thinking to deliver, actually we are planning already to develop and deliver these CAR-T cells directly into the brain using this catheter. And so there are other collaborators like in City of Hope, Dr. Kristen Brown is already giving this IL-13 receptor alpha to CAR-T, and they are trying in the directly giving to the brain. So we are also like maybe next 2 to 3 years, we are going through the clinical trial, and we are also proposing giving these CAR-T cells in the brain itself. So there are a number of strategies which we can make this CAR-T cells effective by not only making a different other approaches such as the SynNotch approach, or making these CAR-T cells effective even in immunosuppressive context by deleting this PD-1 or CTLA-4, or making universal CAR-T cells. So so as most of you heard that these 2 ladies who recently got the CRISPR. So this is where we are, our CAR-T cells meets the CRISPR in terms of making this therapy widely, and using the genetic scissors to do a lot of modifications in the CAR-T cells. So, and for the safety switch, we are developing that either we can go by making this soluble molecule-based safety switch, or antibody-based safety switch. So what I'm saying is safety switch is like, if your CAR-T cells goes into the body, and let's say if it destroys the normal cells, and if it gets crazy, then that's the time we want to eliminate those CAR-T cells from the body. So for that reason, we can either give the antibody, so that this antibody will go and bind to those CAR-T cells and eliminate from the body, or we can base from that small molecules inhibitors. So as I said, that this autologous CAR-T cells is very, very expensive. It costs like almost $475K in US. So there are multiple approaches to reduce this cost, and one of the ways to go with this of the cell CAR-T cells, which is making these allogenic donor CAR-T cells, selections of the non-reactive T cells, or engineer other effector cells like NK cells, macrophage. And so our company mainly makes the CAR-T cell, make these T cells or immune cells in vitro. So we develop these immune cells from the pluripotent stem cells. So, and we believe that that will even significantly reduce the cost. So, and people, especially if there are students who are listening this talk, then this is exciting time for making career in the therapy field. It's not only the CAR-T which is going on. You can see the BiTEs. There are a lot of these antibody drug conjugate, checkpoint inhibitors. And before previously, when I worked at Mayo Clinic, I was working in oncolytic virus and giving this conducting the phase 1 clinical trials. So this is exciting time for the immuno-oncology. And so this is my final slide. And we have a very close, we work with a very close collaborations with the academic lab. Dr. Sheila Singh Groups is one of the top neurosurgeon here in Canada. And so she received this funding from the different organizations. And with that, I would like to conclude my talk. And if you have any questions, feel free to ask. Thank you.
Thank you, Dr. Upreti. We have a couple of questions here. One question, Jivan Sake has asked, even with the CAR T cells, they are to be migrated into the tumor microenvironment to interact with the tumor cells to eliminate them. How do you think the highly immunosuppressive microenvironment contribute towards CAR T cell therapy?
This is $1 million question. This is a great question that it's a challenge in the field, and I'm glad that somebody asked this question. So, so dealing with this immunotherapy, like immunosuppressive microenvironment, especially in the solid cancer, is even more challenging as in the blood cancer. So what we are doing is we are trying to modulate the CAR T cell itself, which can go and sustain in the tumor microenvironment. For example, so if you delete one of the immunosuppressive molecules, PD-1. For example, if you delete that PD-1 molecules from the CAR T cells, this is just one of the example, but there are a number of other factors. So if you delete that molecules and now we give it to the immunosuppressive context, like in the glioblastoma, it can persist there. So there are, but again, I said that that's the number. So mostly we don't know about this, like how we can get success with that, but it's just the approach that we are taking in, and many other people are taking, not only in the PD-1 context, but also in the many other suppressive factors. For example, in many solid tumors, macrophage is one of the major challenge, and it is like I said that these immune cells not always inhibit the growth, but sometimes it also supports the growth. For example, if you can tune in or repolarize the macrophage, then that's one of the strategies. So either by secreting the different molecules using the CAR T cells, or making the CAR T cell itself that can go and sustain in those tumor microenvironment, maybe the way looking forward.
Okay, there's one more question. This is the last question we'll take because we are running out of time. So Nirmal Marasini asks, is there any chance cancer cells escape CAR T cells therapies? How effective they are in patient? Is CAR T cells tumor specific?
Yes, very good question. So yes, like in both questions, yes. So in one way that even in the clinical trials that we showed, so the patient who come back later on, they have this antigen escape. For example, if you deliver these CAR T cells against the CD19 molecule, the patient may develop the cancer cells which are which are CD19 negative. So now the challenge is that's why we are thinking to design these CAR T cells which can recognize not only one antigen but also the multiple antigens. So there is possibility. That's why we are overcoming. We are coming with this different strategy to deal with that. Sorry, and what was the second question?
How effective are they in patient, and whether the CAR T therapy is specific to any tumor?
Yes. So, and regarding the specificity as compared to, yes, it's specific. That's why the CAR T is all about. But when I say about specific, it also, in the different, at a multiple contexts. For example, if your CD19 CAR T goes against these, B cells, it can kill the B cells also, the normal B cells, which we call on target, but off tumor toxicity, But even if the CAR T cells, kills all your B cells in the body, patient is still going to survive. That's not the, because with this corona, maybe that's challenging, because if you don't have a B cells in the body, then you may have a, higher chances of infection. But that can be dealt with, giving this immunoglobulin. But in the cancer, in the solid tumor, like in the brain cancer, so there are, not only CD133, for example, is expressed by this glioblastoma, but there are other normal cells that can be also expressed by these, immune cells. So it also that, but to deal with that, so either your cancer cells should express very high, antigen tumor burden, so that we can deliver this therapy, or we have to, like I said, if you give these CAR T cells just in the brain, so that it shouldn't go to your system. So there are multiple strategies we can deal with that again. But it's a very good question.
Thank you. So with this, I'd like to conclude the first session of our first section of our session, Biomedical Technologies. I would like to thank all the speakers who presented in this session and also all the participants. So in this session, we had Dr. Ramesh Giri, who discussed about the newer technologies that can be used to minimize the steps for synthesizing the drugs. And then we had Dr. Indira Tiwari, who gave us the oversight about the Toxoplasma gondii, its diagnostic approaches, and then importance of Toxoplasma gondii, especially in the pregnant woman. We had Asim Dakal, who gave very nice introduction about how the physics can be used in biomedicine, especially the nanophotonics for, he showed the data on how they use the nanophotonics for determination of microbes in the water. And then finally, we had Dr. Deepak Upreti, who gave very good insights upon CAR T therapy, which is the very, I would say, hot cake in cancer research field and cancer immunotherapy. Okay, so because we don't have much time, so we are not going to take break, and we are continuing with the second session. To continue with second session, I'd like to request Dr. Nitesh Aryal to take over. Thank you.
Thank you, Dr. Roshan, for moderating very beautiful session and with very important topics that included various subjects, which is good for us and all the viewers in Nepal, and those who are interested in research can go through these topics. So today we have another session which is about modern technologies in biomedicine, which myself, I'm Dr. Nitesh, working in Qatar as a senior physician since 9 years after I completed my master's in internal medicine from Teaching Hospital, IOM. And with me, Dr. Susila Maharjan. She is a postdoctoral fellow in Harvard Medical School, Cambridge, USA. So we would not like to delay to start at another very interesting session with excellent speakers from around the world with important topics. So I would like Dr. Susila to take over and start the session.
Yeah. Thank you, Dr. Nitesh Aryal, for opening our second section of the biomedical technology session. Without any delay, we would like to move forward with our next speaker, Dr. Wang Di, who is a plastic surgeon by practice at Plastic Surgery Hospital of Chinese Academy of Medical Sciences and Peking Union Medical College in China. He has been practicing ear reconstruction surgeries and microscopic vascular surgeries over the year. And recently he has joined Harvard Medical School to research on tissue engineering, specifically on 3D bioprinting of vascular tissues and external ear regeneration and engineering. Dr. Wang Di, over to you. Please go ahead.
Okay, thank you, Sushila. Can you hear me?
Yes.
Yeah.
Please, would you please share your screen?
Okay. Okay, can you see my screen?
Yeah.
Yes.
Okay. Hello, everyone. This is a great opportunity to give a speech and talk with you guys. Thanks for my good friends, Dr. Sushila's invitation. And in my presentation, I will try to cover the most representative research and the clinical works on this topic. Most of them are from the researching achievements and the clinical experience of the plastic surgery hospital. Two departments of the hospital are responsible for external ear abnormalities, and we finished thousands of surgeries about this kind of disorder each year. And I think most guys have no idea about what kind of this disease is. And the external ear abnormalities differ from the acquired and congenital diseases. The acquired outer ear malformations usually generate from trauma or burn, while congenital diseases result from gene disorders or some social factors during pregnancy. And there are hundreds of different kinds of congenital auricular diseases. Therefore, they are very complicated, which beyond most people's imagination. We can see the examples. The upper right The upper right figures are auricular deformities, is usually without tissue loss, a distortion of skin or cartilage. By contrast, the malformation such as the figure shown in the lower right corner. And there are macrotia and anotia, which were defined as the absence of tissue of the external ear. They are often combined with hemifacial macrosomia, just like these two figures. Okay, so maybe most people thought they are just the rare diseases. However, there are a large number of patients all over the world. And unfortunately, Native American and we Asian guys are the most susceptible population to this malformation. There are around 9.8 to 14.3 million macrotia and anotia patients in Asia suffering from it. So we cannot ignore it and have to do something to help these poor patients. Therapies differ from the severities and the type of the auricular abnormalities. For example, to cure some less serious auricular deformities, we used a device system called the ear at the early age of the kid. This system was developed by us and have amazing results for those patients. And in this way, the slight deformities can be corrected without surgery. For those who cannot be cured by this system, we may choose a mini-invasive surgical method. In this case, just the regional repair would be performed. However, for most macrotia and anotia patients, non-surgical method and the mini-invasive, we use this. So due to the lack of blood supply, elastic cartilage has very limited capacity to regenerate or be repaired compared to the other tissues. Autologous costal cartilage graft, a multiple stage and complicated reconstructive surgery is the golden standard therapy for this macrotia and anotia patients. And the surgical method is shown in the slide named the Badachu method. It is named by the title of our hospital, and nowadays it is one of the most famous surgeries for this malformation. And we can see that before the surgery, we used a 3D scanner to get the digital data of a patient health ear. This is me who is doing the scanning. And after data analysis, we print it with the common resin to get an ear model. It will be used as a template in the second stage surgery. And in the first stage of the surgery, a tissue expander is implanted under a patient's mastoid skin, and we inject saline water twice a week after the surgery to make it expanded to get enough skin, just like the procedure of pregnancy. In the second stage, we harvest the rib cartilage from the patient's chest and curve it with a surgical scalpel to form a ear-shaped framework, according to the 3D ear-shaped template. And then remove the tissue expander and suture the framework under the skin. A year later, the patient will receive a modification surgery to get a better appearance. This method is nice with a high rate of success and perfect experience. We can see the reconstructed ear. Nonetheless, the reconstructed ear is overstiff because the rib cartilage is the hyaline cartilage and has distinctively different mechanical properties with the native auricular cartilage. So we know that we have to harvest the rib cartilage from the chest, so the other donor size deformities might be left. And also the other surgical complications make patients feel very hurt and uncomfortable. Now, this is the worldwide problem. We have to find a better way to solve that. So to solve this problem, tissue engineering as a potential alternative strategy was developed and applied in this field during the past decades. And more than 20 years ago, Dr. Cao developed the first human ear-shaped tissue engineered cartilage in mice when he was a researching scholar at Harvard Medical School. That was called a milestone in the tissue engineering field. He used the PGA fibers to fabricate ear-shaped framework and then seed chondrocytes on it. After implantation in the mouse bags, the cartilage had formed. After that, he came back to China and being the president of my hospital. Then in the past decades, researchers and surgeons of my hospital dedicated to this researching work related to the auricular cartilage engineering. And after years' work, we finished the stage 1 clinical study and published that in the subjournal of the license. In that paper, we published the results of 5 patients with tissue engineered auricle. We can see a flowchart of this study on the left. Generally, the ear scaffold used a PCR mesh as an inner core, which was wrapped with PGA fibers and coated with PLA. For cell parts, macrosal cartilages were harvested to isolate chondrocytes. Passage 2 cells were seated on the scaffolds. 12 weeks later, in vitro characterization were performed. As shown in the middle, cartilages had been initially formed. And then the cartilages were implanted in the patient. This video is a follow-up after 3 years of the implantation. We can see very nice elasticity of the cartilage and the excellent shape of the auricle. According to the follow-up figures, we can see a dynamic changing of the tissue engineering auricles. The pre-operated macrosal ear showing peanut-like structure, and post-implantation assessment at the different time points shows the signs of gradual shape recovery, and MRI examination reveals an obvious sandwich structure of the ear framework with cartilage. And however, we are still facing some tough challenges. For instance, the inflammation brought from the materials, macrosal environments for cell viability and differentiations, and degradation. So for this part, 3D bioprinting allows us to print the scaffolds with cells for direct fabrication of ear-shaped cartilage. In this way, the signal transaction between cells, cartilage matrix aggregation, and the nutrients transportation in the constructs could be achieved better. So in the few past years, some researchers of our lab turned to focus on this topic. And with the extrusion bioprinter, which allows bioprint heights work at the same time. In this way, multiple materials can be printed simultaneously, and both natural and artificial polymers were applied in our researchers for biofabrication of auricular cartilage. We developed a hydrogel system with native and cartilage components based on some biodegradable natural materials such as gelma, and use them as to print auricle constructs. The hydrogel provides a nice environment for chondrocytes. As shown in the video and figures, a very high resolution was achieved. The printed auricle by cartilage hydrogels has an excellent elasticity, which will mimic the elastic cartilage. Cells enjoy a friendly experience for living and differentiation. We implanted auricle constructs in nude model and rabbits. We can distinguish the ear shape on the back of mice clearly in this video, and staining results prove that the cartilage matrix has been secreted and the cartilage tissue was established. However, hydrogels alone cannot tolerate the pressure from the human skin. Though it is elastic enough, still need to be a little bit stiffer to maintain the 3D shape. Then we developed some artificial materials such as animal acid modified PU. The MPA was used to react with PU, and finally a waterborne PU was achieved. This PU bears excellent flexibility and can be printed by DM method during relatively low temperatures. It is around 50 to 70 degrees. The printed scaffolds have a high elongation and a break of nearly 200%. By contrast, another degradable material, PRA, just have a high elongation and a break of 6%. Its good elasticity and toughness can solve the problem of large brittleness and insufficient mechanical compliance of traditional degradable materials. And the comparative study on PLA degradation performance confirmed that these modified PU scaffolds had controllable degradation properties. Okay, so later, similar to Professor Kong from Korea, we developed hybrid bioprinting to get more satisfying results. At this point, we used our cell London cartilage hydrogels and the further modified PU simultaneously to meet the most requirements of cartilages. We also did some attempts on low temperature deposition with the PU and got charming outcomes. Besides, I also did some microsurgery related researches and mainly focused on perforator flaps. Microsurgery is a basic technique for a plastic surgeon. A perforator means a very small artery that can powerfully supply a wide skin flap. In my studies, we made some DIEP modes and selectively finish telomeres of certain vessels. And this is me who is doing the microsurgery. And then using a SPY system and ICG to monitor the fluorescence. In this way, we can observe the blood supply of a certain flap directly and predict the survival of the perforator flap. And based on my microsurgery background, I'm working on vascular conduits bioprinting projects with the collaboration of Dr. Sushila in Harvard Medical School and the supervision of Professor Yishrak Zhang. Using the extrusion printing system, Using a extrusion printing system, we successfully printed both single and double layer tubes vessels with satisfying mechanical properties and basic functions to meet the requirements of real vessels. We also printed cell London hydrogels to form vessels. It was proved that the cell enjoy high viabilities and stretching abilities in the vascular conduits. And at the end of this speech, please allow me to give a brief introduction of my hospital and my professors. The first one is my supervisor when I was an MD student. He is now the current president of our hospital. And he finished the first year reconstruction surgery with tissue engineered auricle. Nowadays, he really has 30 to 50 external ear related surgeries each week. Professor Cao Yulin in the middle is a scientist that fabricated the first case of the human-shaped tissue engineered cartilage, as we showed in the previous slides. He dedicated a lot to the field of tissue engineering. And the last man is my supervisor's supervisor. He created our hospital, who created the plastic surgery in China and then created Bada Chu ear reconstruction surgery. This great surgeon was extremely famous all over the world in his area. And then this is a general view of my hospital. It is the biggest plastic surgery hospital in the world. We have more than 20 departments, such as external center, cleft lab and planet center, and the breast center. We do acquired and congenital malformation reconstruction, scar revision, and aesthetic surgery, and many therapies related to plastic surgery and dermatology. All kinds of researches and clinical collaboration are welcomed. And if you have any idea, please contact me. It is my email. Okay, that's all for this.
Yeah, thank you, Dr. Wang, for your nice, interesting presentation and timely presentation. Yeah, if anyone has any question, please go ahead.
Any from floors? No?
Yes, so yeah, I have a quick question. Dr. Wang, what is the successful rate of engineered ear transplantation
That's the root mice, yeah, without immunology
in
reaction. And root
sorry,
mice.
can you please repeat it again?
Just the nude mice, very
Okay.
common specimen.
So you have not tried in human patients?
We have tried it as I shown in the previous slides. And the first is the mice, and then a big animal, and then about 5 to 6 years ago, we start from people.
Okay.
Yeah.
Yes, it's very exciting to see the
I have one question.
engineered tissue transplantation in human.
Thank you.
I have one question.
Okay.
Well, as you mentioned there, still we have some challenges for implantation due to some inflammation or maybe some degradation. I'm not sure about that, but I think this technology was developed in your hospital first time, right?
Yeah.
So is there any other collaborator for using your technology outside China or some other country?
Oh, okay. You mean the tissue engineer the ear implantation, right?
Yeah, that's right. Yeah, I'm just talking.
Actually, this is multi-center research. It was not only performed in my hospital. My hospital just performed the first case, and some other hospital in the China has also do the same work at this third case was performed in Shanghai, another hospital. But until now, no more hospitals outside China has done this before. So yeah.
Thank you.
Thanks for your question.
We have a question in Zoom from the audience.
Okay.
Can you comment more about the vasculature development inside the engineered ear transplant and also about the transplant rejection?
You mean the vascular?
Yeah. Do you need vasculature in engineered ear for its function? Yeah.
Okay, actually, cartilage doesn't need vessels. It was supplied by some other research, and vessels just exist around the ear. I mean, under the skin and some other fat tissue like this. So we don't need vessels. Yeah, but if you want to do this, maybe you can just try some other method to get a better nutrient supply, not only vessels. Yeah. Thanks for the question.
Yeah, and second, there is another question connected with the same one. So is there any rejection during transplantation? Like immunological rejection or any kind of?
No, because we just use the cells from the patient itself. And so there's no serious reaction, between them. But there is also some reaction generated from the materials, the general inflammation. Yeah, thanks.
Yeah.
Yeah, I have one question.
Okay, please.
Yeah, because basically we are all from Nepal, and most of our program are viewed by Nepal. In Nepal there are a lot of ear deformities. Those people who are living in remote rural areas, and they have ear deformities. And you have been working very excellent hospital with a very good technology. So how do you think that these technologies can be used in our country or the developing countries like Nepal? How it can be possible?
Okay, thanks for your question. So you mean the surgery method or tissue engineer, the Oracle?
Yeah.
Tissue engineer. Okay, okay. And for the previous, I mean the surgery, we have many collaboration with some other countries, especially in South Asia, and some professors for hospital often travel to this country to help them to finish these surgeries. As I know, also some doctors in Nepal did the excellent work about the macular and the ear reconstruction. But for tissue engineered auricle, now it's not very nice to do the wide clinical application. So there's still some gap between the research and the real application, I mean the worldwide application. So the first thing is that we have to solve the problems and then try to make it a little bit, try to simplify it and make it a common surgery and therapy to the world. I think at that stage, maybe we can achieve that in many countries, not only China and Europe, and also Nepal and South Asia and some other countries. Yeah. Also, there are a lot of excellent scientists in Nepal are doing the same work with us. Maybe we can have some further collaboration on this topic to make it a real thing in the near future. Thanks.
Yeah, thank you, Dr. Wang, for your nice and exciting presentation. It was a pleasure hearing you, your excellent work. Now moving on to our next presenter, Dr. Bises Khanal. He is a research scientist and chairperson of Nepal Applied Mathematics and Informatics Institute in Nepal. He has been a senior artificial intelligence scientist at Fuse Machines and a visiting researcher at King's College London. His area of research include visual perception, machine learning, and artificial intelligence. So today he is going to give a talk on artificial intelligence and medical imaging. Dr. Bises Khanal, please go ahead.
Hello, yeah, can you hear me?
Yes, please.
Okay, and can you see my screen?
Yes.
Yeah, you can see.
Okay. So thank you. First of all, thank you very much for inviting and giving me this opportunity. I am Bishesh, and I'll be talking about AI primarily in healthcare, especially for low-income countries like Nepal. Brief background, I'm not from medical science. I'm primarily an engineer by training, and then I did my master's in Europe, then moved on to UK for postdoc, and then I moved back to Nepal in 2019, and I'm currently doing research as a research scientist at NAMI. So what I will do is first I will quickly go through very brief introduction of what AI is, and then about AI in healthcare, then moving on into what we are doing, especially for global health and AI with examples in Nepal. So when we say AI artificial, I think that the term itself comes from in comparison to natural or human intelligence. And if we look at human intelligence or the intelligence in nature in general, we attribute to some sort of functions and activities of your brain. And there are low-level activities such as touch, taste, smell, hearing, balance, or even vision to some point where animals and humans share that. But then when we go to higher levels of intelligence like language, thinking, imagination, these are often attributed only to human beings. So that's what basically distinguish us from some other animals, right? And I think traditionally when researchers, philosophers, or people fantasize about emulating human brain or being able to do all of these activities, all of these, and but these days AI is not exactly used for this particular goal, or like large goal of really being able to emulate or do exactly like what humans can do. So it's more artificial general intelligence, and AI, or the artificial intelligence, is used for slightly different thing. And what that is if I have to describe it, I mean, it's some sort of tasks which is often attributed to intelligence. So that, and man-made agents, softwares, machines that can do those sorts of tasks, called as AI. We can take some examples. So if I type on the top, you can see where I just typed on Google Translate something in Nepali, and then it does two things. One, it detects that it is Nepali. It also tries to give some spell correction, and then it translates it into English, right? So translation, or detecting that this is Nepali and being able to understand what it says and then to translate it. So this is one form of AI, but that translator will not be able to do something else, such as on the bottom left figure, we see images of cars and vehicles are being detected, and then we even compute distances, how far they are. So this is yet another AI, but it's not again able to do the translation. So they are very specialized activities, specialized tasks that AI can do as for now, especially. And we have, you work with Facebook, or you've used Facebook. There's auto tagging features, advertisements personalized to you, recommendations in Netflix, Amazon, YouTube. All these are yet other examples of AI. And on the right, you see it's supposed to be video anyway. So it's a placenta. So it's on the ultrasound. It's detecting placenta and the fetal when doing fetal ultrasound. So these are examples of AI used in different applications. Now I will move to AI in healthcare and medical, especially in medical imaging informatics, because if we talk about healthcare in general, there are lots of different applications in many different areas within the healthcare one can work. But my focus of the talk and most of my work is on medical imaging, or imaging data especially. So this leads me to briefly talk about medical imaging informatics. So what does it really mean? It's primarily extracting clinically pertinent information. What for? It can be for early diagnosis. It could be for prognosis. It could be for surgery planning, therapeutics. It could be for robot-assisted surgery, or it could be for even basic understandings in biology, or in disease understanding. Right? We could use images, analyze, extract information. So that's the domain, and there are lots of different imaging modalities available. And what they do is they primarily look into the human body in different ways, mostly looking in the structure or function of different organs, different systems, biological systems. And AI is a hot topic in medicine, biomedicine right now. There's lots of papers being published in the last few years, spanning from, let's say, CT scan in brain hemorrhage, or to diabetic retinopathy, to, if you go on the bottom, then the cardiac diseases, to again CT scans and lung cancers, and mammograms, right? So there's lots of things being developed. But then I often get this question, people asking, is this really helpful for low middle income countries? Is it, or is it just for advanced economies in the healthcare systems there? Right? And actually, no, it's not just for advanced economy or in healthcare systems. There's a lot of things we can do in low resource constrained settings, but one needs to understand the local context and address right challenges. So there are opportunities to innovate in global health, and from the within the, I mean, the expertise within these countries, especially because we need less. So when I say democratizing innovation, it is in a sense that we need relatively smaller infrastructure compared to traditionally the amount of infrastructure we need for in medicine to do research, right? You need huge labs, lots of infrastructure. Now, if you have computers and you have human resource, you can do frontier research but you need funding. That's another part we will, but at least. All right, so what are we doing? So we'll take mostly the examples of what we are doing currently or planning to do in the context of global health. And so if I break down into the way I look into this problem or this field, especially in terms of global health lens, is I think we need to focus, and the greater focus is needed on neglected diseases that are important locally. And we need to focus also on technologies that will be affordable and accessible, because if it's very expensive, large number of people won't be able to really use that, especially in settings like ours where there's no global insurance policies, and in general most people have to pay on their own for accessing these cares. And the another important part, especially in the context of Nepal, is really empowering health professionals at all levels. We have all heard about success stories of female community health care workers or volunteers in especially improving the maternal and fetal health in Nepal. Nepal is taken as one of the examples in the global health for that scenario. There are medical officers, lab techniques, and mostly in primary health centers. So in the most rural areas of Nepal, majority of the areas, we have primary health centers, healthcare centers, health posts, and there are not many experts available over there, and not many advanced imaging facilities. But still, they are the, I mean, they serve large majority of the population, right? That's one important area where we can actually broaden the scope and the service coverage at that centers with AI. The other important part is when you have there's very few urban centers, few tertiary care centers, which has to cater to almost whole country. There is usually overburdened with patients, especially in government tertiary centers. So how can we really improve the quality of healthcare on those settings? That's another important area where we can work. I will give some examples. Most today, I will focus mostly not on the relatively expensive imaging devices like CT scans, MRI, but rather on the low-cost sides. And this would be ultrasound, X-rays, or even just smartphone cameras, right? And so I'll focus mostly on those three topics. So if you go to ultrasound, I say this is low cost, portable, and has high temporal resolution. So for example, if you want to get the beating heart, you can really quickly get the whole cycle of the heart, right? And there's no radiation, unlike X-rays or CT scans. So it's quite attractive. And these days, the size is getting smaller and smaller and coming in the smartphone size of the smartphone. But there are challenges, and the challenges mostly revolve around having limited field of view. So if you want to take, so these are the examples of fetal brain. Even for the small fetal brain, you of let's say 30 weeks, if it's around 30 or above 30, you cannot really get the whole brain at one go in 3D. So you need to take multiple images, which means the operator has to go around, maneuver the probe to get these images. So which means you need high level of expertise. One needs to understand what images you are taking. So this really limits the widespread applicability or uses of ultrasound, despite it being low cost and portable, right? Because in settings like ours, we struggle to send experts in most primary health centers, care centers, or to train and hold people there, because there are socioeconomic challenges related to that. So the whole idea is to be able to task shift and empower local medical officers or people who are already available there to be able to use these machines, and with assistance from AI-based technologies. So I'll give some examples of our work. So this was mostly when I was in Cat King's in London doing my postdoc with our colleagues, and what we are trying to do is so there are certain standard views or standard planes in brain that during the fetal diagnosis or routine fetal ultrasound one needs to do. So the idea is to automatically find the right plane within the 3D. So that way, instead of the expert being very accurate and finding the right target, you could have an AI finding it as soon as you are loosely around the region, around the area. So when you can do that, what that means is you will have more consistent detection amongst different operators, and you will need less expertise to do that. So this is standard plane detection. You can also combine. So as I said earlier, we have limited field of view. So if you go around maneuvering around the fetus, fetal head, if we can combine all those different images into one big 3D volume, that means you get better picture. You have you don't need to be a real expert to find the exactly right place. You can just combine all these images. So and what you see on the right here, on the bottom row, is the 3D, the best view obtained by an expert sonographer or expert operator who is finding the face, right, 3D face on the bottom row. While on the top, it is automatically combining all these different ultrasound images taken during the session. And it gives quite promising results. We can also go beyond that when we because in ultrasound, what we are doing is we are really trying to target right view, right image, obtain that, acquire those images, and then do further analysis. So if you can control the quality of these images by looking at the uncertainty or the how good your predictions are from the machine learning algorithms or AI algorithms. So if you can assess those, it can also help in whether to keep those images or to discard those images, right? So this is what we
Sorry,
are trying to do here.
just a
Yeah,
minute.
how long? Yeah.
Dr. Khanal, you have 5 more minutes for your talk.
Okay, thank you. So here the idea is to assess the uncertainty of the algorithms that predict the images, whether it's a right image, the right kind of images, or how confident it is, right? And then based on that, we do that. So we are working on. So this is this uncertainty estimation within the AI systems is also an interest theoretically, and we can apply this to many different tasks. So from now, fetal ultrasound, I'm moving to the cardiac ultrasound here that we are currently doing, and where we are segmenting, we're estimating the volume of different structures, cardiac structures, and with uncertainty, and using those uncertainty, you can discard bad images and then get better results. The another important challenge in AI is to have these, you need large amount of annotate labels. The experts need to annotate those, and how can we leverage the large amount of data that are unlabeled, that the experts haven't really annotated. So this is semi-supervised learning, what we call as semi-supervised learning, and we're working on those. Then I'll be quick because I think I don't have a lot of time. So in X-rays, what we are working on X-rays because it's again cheaper, portable, than CT scans, and it's quite useful for respiratory disease and orthopedics. So here's one example that we are working, we work is on scoliosis, with this estimating the spine curvature automatically, because for surgeons it takes a lot of time and it depends. So it's if you have 3 different surgeons, exact angle can be different. So there's a variability, and then it can affect the treatment planning. For the scoliosis, right? And this is COVID work that we're currently working and in preparation for submission, where you try to distinguish the pneumonia, common other bacterial or the other viral pneumonia from COVID, with but again focusing on X-rays instead of CTs. We are also working on, so if you do rural surgery, if you want to do rural surgery, there's mostly only CTs, X-rays available, CTs are not available, and then the X-rays don't give 3D images. So how do we combine multiple X-ray images to produce 3D images? We're trying to, so it's very preliminary work, but again, we need waiting for some funding for these sorts of work. Then the smartphone and low-cost ultrasound, we can do a lot of things from here. So there's pesticide estimation from paper analytic devices. So you have simple filter paper where you have the solutions for the, let's say the solution from the vegetable or sample and the reference, and you compare those. So this is how to use machine learning in these sorts of things. This is in collaboration with Kayas here in Kathmandu. So again, doing a microscopy, smartphone-based microscopy. So instead of bright field microscope, if we use smartphone microscope and be able to automatically detect giardia, cryptos, these diarrheal diseases. So that is again just starting with in collaboration with our collaborators. smartphone-based camera where we try to do gait analysis. So you estimate pose or motion of the people on different joints. So this can have applications on Parkinson's, dystonia, and other aging-related diseases that we're working with Naveen Koirala from NAMI and Yale, and Azad from ETH Zurich and NAMI. So there are a few other works I can talk about that during questions. I think the challenge is in Nepal, what I've realized is we don't have digital recording systems like electronic health record systems or PACS, and this is a big challenge. This must change. I mean, if you go to most of the hospitals, even in advanced hospitals, they rarely have images being stored from several months, right, and properly stored. Funding is, I think everyone knows that, and then the clinicians also, they are usually overburdened here, and they have very little time for research and innovation because of their clinical duties and not much incentive for this, and that makes it difficult to really collaborate with the clinicians also. So that's the challenges, and I would like to thank you. Thanks my collaborators and the audience.
Thank you, Dr.
Hello.
Bises.
Yeah, thank you.
Thank you, Dr. Bises, for your nice talk. Now the floor is over to audience if you have any question. Yeah, Dr. Bises, can you please talk briefly about the feasibility of this technology, specifically smartphone usability of smartphone microscopy for monitoring the function of heart, whether it be in vivo or in vitro?
So you mean smartphone microscopy for heart?
Monitoring heart function.
Okay. So I am again not a biologist or the clinician. So when we say smartphone based microscopic, so there's a smart microscope being attached to the smartphone micro camera, right? And then so this is usually, so you need a sample that one needs to take microscopic sample. I mean the sample where we can look into cells, and if there are cells or any kind of imaging appearance on under microscopy images that have implications to the cardiac function, then yes, we can use that, but one needs to take the sample out, right? So, but then having said that, so that's on the smartphone based microscopy, but I didn't show something else which we are considering to work on. One of my students had worked on that earlier, is on the sound, so cardiac sound. So instead of stethoscope, you could think of the digital, I mean, just taking from the digital microphone, the sound, and automatically analyzing this for arrhythmia or rheumatic heart disease. So this sounds, there I think there is a bigger applications and more straightforward perhaps.
Nice. Yeah, so we have one question from audience. He is saying if you have any comments or thoughts about the application of AI in COVID-19 diagnosis.
Yeah, thank you. So I showed one slide where we are working with our collaborators in Bangladesh. So here, so I think that so one biggest work we can do here is on prognosis. So here, so let's imagine, I mean, with the COVID, so as the numbers increase, so the hospital, I mean, already there's a big problem in Nepal that you don't have ICUs or these ventilators available for people, right? And then you have lots of symptomatic patients coming in. If we can predict early how bad the patients, I mean, how bad the prognosis will be. I mean, will it, the disease trajectory, do these patients need ventilators or ICU in 5 days, 6 days, or they can stay at home? So this can help a lot in clinical management. And for this, CT scan can be better, but CT is expensive and not practical in Nepal because it's you need to disinfect to avoid cross contamination. But even before that, it's expensive. So X-rays is being used currently by the clinicians to monitor these patients, and we could use AI for those aspects. But again, the challenge goes down to really being able to get those X-ray images, collaborate with clinicians in this fast-paced environment, especially during this scenario, which is challenging. But I think there is still good amount of work that we can do, and we are exploring that actually currently, talking and working with some clinicians.
Yeah, I have one question, Dr. Bises. as you are currently
Yeah.
working in Nepal, and then artificial intelligence is one of the biggest upcoming, career for the youngsters, and it is very good career because most of the things are going to be replaced by artificial intelligence. And regarding Nepal, okay, how Nepal is like dealing with the medical legal aspects of the artificial intelligence. Are you working with the Nepal government for the medical legal rights of this artificial intelligence in the medicine? So, is there anything you want to explain about?
Yeah, so I think that's an important point. So the two sides of this, I mean, of this question for me, I think. So one is at what stage I mean, there's a big career. I mean, within Nepal, if we want to innovate, there are, as I said earlier, these electronic health records or PAC systems that digitizing all these images properly. This hasn't happened yet in Nepal. If you go to most hospitals, this is not there. So that makes it extremely difficult to properly get this data and start working quickly unless you have a big amount of funding which can then get this infrastructure to get within the funding. Then so that means I think unless we have this pipeline ready, it will take some time to really have applied, I mean, to quickly innovate or within Nepal these systems and apply this in Nepal. I mean, but then there is another aspect where actually some companies from around the world are trying to test their AI systems in Nepal. I mean, they are saying, okay, we have developed the system and we want to apply this, but there is a lot of testing involved behind the scene when they come and actually. And here, I think it is not clear. The government, but I know Medical Council is thinking about these stuffs together with telemedicine and this scenario and how to or what kind of policies to develop for this and it's and not just the AI. I mean, there is these issues regarding approvals like FDA or what kind of approvals we need here in Nepal. Do we have our own approval monitoring structure or we follow others? So there's this one aspect. The other aspect is privacy. So privacy is extremely important in medical data. So how do we deal with the privacy issues within the medical data? So these two issues, I think, at least for the privacy and telemedicine, I know Medical Council is working on that. For the AI application in particular, I am not sure. I don't think they have already worked, but I am in communication or in contact with the people in Medical Council, and I know they are thinking, but I don't know when these things will actually fall in place or we will have policies developed. That in terms of timeline, I don't know.
Thank you. I have one question, actually not question, but as you said, the major part in here for the implementation of your images in digitalizing library in hospital, actually. You know, in Korea, most of the hospital, I mean, using a digital digital data in a hospital is very common. Starting from small hospital, not only hospital, even in a clinic, small clinic is also, they are using this digital data, including everything, X-ray, CT scan. Within a minutes after finishing their scan or X-ray, they just get the data accumulated in one place, and doctor can analyze immediately what we are actually expecting in Nepal as well, right? So, I mean,
yep.
If we want to do that, then from your perspective, can you make some kind of recommendation to the government, as I also can be some kind of helpful in communicating with the government or government from the perspective of technology that is being used in Korea. So here what I'm saying.
Okay, yeah, thank you. So I think so I mean, it's not the problem of technology. Actually, the digitizing and storing those images, it's in terms of technology, it's not that difficult. I think I mean, there are software tools, but I'm quite surprised that it's weirdly expensive. I mean, I don't know, at least some of the softwares being sold in Nepal, it's much more expensive than it should be, I think. I don't know why. So there's something else going on which is preventing these systems to be. So one could be, I think, the policies. So medical council could develop policies that ensures that this should be digitized. Because if you think of, let's say a patient comes today, and let's say I go to a hospital, I have my brain MRI taken, and after 6 months, if I go to the hospital, I think it's my right to be able to ask the doctor, so can you compare the last I mean the MRI and this one, because one needs to know to have proper, I mean, assessment, right? This sorts of, but then you cannot do that right now, and it's not because of technology. Because in terms of technology, there are technologies, or even the software developers in Nepal can build these sorts of things, or we can bring technology from elsewhere. It's not a big deal. But I think what's it's perhaps the economics are not feeling the need or importance of this digitization that is preventing from the people. And I'm in discussion with again the medical council or these people who have realized this sorts of issues, especially because of the COVID now, and the again, linked again with the telemedicine as well. So I think things are moving, but as I said, I mean, people are aware, they know this should be done, they are trying to work on this, but when will it happen, I don't know.
Thank you. Thank you for your
Thank you.
Please.
Yeah, thank you, Dr. Bises, for
Yeah.
your informative talk. It was great hearing this
Thank
kind
you.
of new technologies happening in our country. So now moving forward to our next speaker, Dr. Sagar Regmi. He's a postdoc fellow at Research Center for Applied Science and Technology, RECAST at TU. He received his PhD from School of Chemical and Biomedical Engineering, Nanyang Technology University in Singapore. His research interests include nanotechnology, biomaterials, single molecule biophysics, targeted drug delivery, and many more. He is going to give a talk today on nanotechnology in medical sciences. Dr. Sagar, is over
Thank
to you.
Susana ma'am, for your kind introduction. And firstly, I'd like to thank the organizer for providing this opportunity for presenting our research work in this platform. So today I'll be discussing about nanotechnology in medical science, but I'm particularly focusing the nanotechnology perspectives in generating the various cell lines like circulating tumor cell lines, as well as using a particular technology developed from nanotechnology basically to image these circulating tumor cells. So the title of my presentation today is a fluidic shear stress increases the anti-cancer effect of ROS generating drugs in circulating tumor cells. So giving a very, from a very fundamental basic, almost like 8.2 million people die every year through cancers. And this number is expected to increase by 70% in next 2 decades. Now there are, of course, like, more than 100 different kind of a cancer, and each cancer has a very different cause and like a different diagnosis and different treatment strategies. But most common cancers are lung cancer, liver cancer, stomach, colorectal, breast, and the oral esophageal cancers. If we see the statistics, almost like in 2008, 7.6 died with the cancers, and this number increases to 9.6 in 2018. So if you see this percentage of increment, almost 7.8% in between 2008 and, sorry, am I audible?
Yes.
Yeah. 7.6, almost like 7.8% increment between 2008 and 2012. And if you compare between 2015 and 18, it's a 9% of increment related to cancer death. So such a high number of cancer deaths come from the factor of a cancer metastasis. It is the cancer metastasis that is responsible for 90% of a human cancer death. There are 5 stages. At the lower stage, the death rate is relatively very low. For example, survival rate for a breast cancer at stage 0 and 1 is 99%, and a lung cancer is 55%. When a cancer turns into a very metastatic or a late stage, in stage 4 or 5, the survival rate significantly decreases. For example, in a breast cancer, it's 27%, and lung cancer is like a 4%. So basically today I'll be discussing about the cancer metastasis about at the late stage of metastatic cancer. So let us look in more detail about this steps involved in the cancer metastasis. There are various factors like a mutation in a p53 genes or various carcinogens that induces a primary tumor. So what happens is this primary tumor, the cell detach and it enters into the blood vessel through the process called intravasation. So the circulating tumor cells basically experiences a fluidic shear stress in the blood vessels. So there is a concept that this shear stress can kill the circulating tumor cells, and this circulating tumor cell basically exit and form a secondary tumor in a different parts of the body. So we understand these circulating tumor cells are considered as the origin of a metastasis. So starting from a breast, it can metastasize to the different region of the body to lung, brain, bone, and liver. So this is the complete process of a cancer metastasis, which is responsible for 90% of a cancer death. So we try to understand the mechanical force involved in these cancer metastasis. So there are two possibilities. These cancer cells basically can flow on the blood vessels or can get trapped depending upon the diameter of the cells and the diameter of the vessels. So it can experience a high velocity, in the capillaries regions as well as the low velocities in the vein region. So it has a different shear stress experience at the different parts of the blood vessel. For example, shear stress 5 dynes per centimeter square at the venous level, 15 at the arterial level at heartbeat of 70 beats per minute. If the heartbeat increases like 140 beats per minute, it experiences a shear stress of 30 dynes per centimeter square. And at a very intensive condition, it can reach shear stress 45 dynes per centimeter or shear stress 60 dynes per centimeter square. So basically today I want to target use the concept of shear stress and use the nanotechnology concept for generating a cell lines and imaging process. So basically many anti-cancer drugs are used in a chemotherapy, but very little is known about their efficacy, basically targeting the circulating tumor cells. So in this project, we basically went to understand the fluidic shear stress in whether that can affect the efficacy of these anti-cancer drugs in the market, available in the market. So this is still a problem because most of the people use a petri dish model and they try to validate in animal and in a clinical trial, but they are basically using a particular cell lines, but not the circulating tumor cells. And another problem with these circulating tumor cells are very rare, meaning like in a 1 ml of a whole blood, you can collect at least 1 to 10 CTC from cancer patients. And there are various techniques developed for this CTC isolation, but this system is not suitable for a drug development. So these are the current problem in the field. And so to solve this problem, we construct basically a microfluidic circulatory system which can generate a physiological shear stress, and we can study the effect of shear stress together with these anti-cancer drugs. So this is basically our microfluidic system that we have developed, which I will explain a little later. So basically talking about the hypothesis of this project, we know that fluidic shear stress basically produces the superoxide, which damages the mitochondria, and this destruction of a mitochondria lead to the apoptosis through the release of a caspase 3 enzyme. Or there is also a possibility that this superoxide production is eliminated by the different kind of antioxidant like a propyl gallate. Or there is another way that superoxide can be converted into the hydrogen peroxide through the enzyme MnSOD, manganese superoxide dismutase. Or this hydrogen peroxide then can be converted into hydroxyl ion, again damaging the mitochondria and apoptosis. This is a very well-known mechanism in this area. But we want to understand that whether these anti-cancer drugs, which can produce the reactive oxygen species, has a synergetic effect with the fluidic shear stress. So we try to give a hypothesis that this fluidic shear stress can increase the anti-cancer effect of an ROS generating drugs. So we try to understand whether shear stress affect the cytotoxic effect of anti-cancer drugs on breast cancer cell line. Then we want to understand whether there is a correlation between shear stress and the reactive oxygen for this targeted drug-treated cancer cells. Then we want to bring the concept of an antioxidant together with these anti-cancer drugs. And we wanted to optimize for a cancer CTC of a different origin. So we basically use the techniques of a FRET microscopy, MTT, NXNV, and PI, and DCFTA, DHE dye for detecting the cell apoptosis, viability, necrosis, and the cellular ROS. So I first like to explain about the concept of our microfluidic circulatory system. You have a flow rate controlling tube, R1. It is connected with the cell suspension. Basically, we keep our cells over here, and these cells will flow through this tube known as a circulatory tube of radius R2, and it can form a complete closed loops. So basically, we can calculate the shear stress using a Poiseuille's equation, which basically depends upon the flow rate, viscosity of a medium, and a radius of a blood vessels. And we are particularly interested shear stress 15 dynes per centimeter square because it is an arterial shear stress at a heartbeat of 70 beats per minute. So we basically construct these microfluidic devices. After that, we try to choose a different kind of anti-cancer drugs based on the level of an ROS. For example, Taxol, which has relatively less ROS production compared to the doxorubicin. So this is basically on the level of an increasing level of an ROS. So we tried around 16 drugs, but I have presented only the 4 effective drugs like a Taxol, etoposide, cisplatin, and a doxorubicin. So here comes the role of an nanotechnology. Basically, we generate these different cell line. First, let me explain about this MCF cell. This is a non-metastatic cancer cell, and this 231C3 is a metastatic breast cancer cell. So here we use a plasmid and transfect these cells, and we have a mechanism to detect the status of the cell. So meaning here is that the live cells are green in color, and when a cell die, it is a blue in color. So what basically it has is when you excite the light with 433 nanometer, there's a transfer of energy from syn fluorescent protein to the yellow fluorescent protein through the DVD sequence, and you get a color in a green spectrum. But when a cell undergo apoptosis, it releases a caspase-3, and this caspase-3 basically caught the sequence of a DVD, and there is no transfer of energy. So we get a color in a blue region to detect apoptosis. So after that, we went to do these various experiment, and you see here that shear stress 15 only has a relatively very less amount of a cell apoptosis. But when you use the completely a doxorubicin in adhesive or non-adhesive condition, also very less apoptotic cells. But you can see over here in this image, like when we have a shear stress 15 together with a doxorubicin, like a fluidic shear stress together with the doxorubicin, you can see a huge amount of an apoptotic cells. These are basically a quantified image. So after that, I tried into a relatively highly metastatic cell line, that is a 231 cell lines, and the results remain consistent. Like shear stress only has a very, you can see over here, like the shear stress has a very less amount of an apoptotic cells. When you have a doxorubicin, very few cells are dying. And even in the non-adhesive, very few cells are dying. But when you have a fluidic shear stress together with the drugs, you can see a relatively significant apoptotic cells. And these are basically a quantified image. So after we figure out that shear stress together with the drugs is effective, so we try to understand the mechanism. So we basically measure a level of an ROS using a dye called DCFTA, and we figure out that shear stress basically increase the level of an ROS in the circulating cancer cells. So this is basically a very simple experiment that we circulate the cells for a different time period, and we just measure the level of an fluorescent intensity for ROS. And we figure out that it saturates at the 3 hour of time point. So after that, this is completely a shear stress, no any anti-cancer drugs. So we come to know that 3 hours is enough for a shear stress to generate the ROS. So we basically use two different conditions, like static without any circulation in a microfluidic system. We basically treat the cells, breast cancer cells, with these different kind of drugs, and we measure the level of an ROS, and we figure out that doxorubicin and cisplatin are ROS generating drugs, whereas Taxol and etoposide are not. But when you have a shear stress 15, you can see a relatively, serious production of an ROS, but when there is a synergetic effect between SS15 and DOX and SS15 plus cisplatin, there is a relatively a higher significant production of an reactive oxygen species as indicated in this diagram. Here you can see DOX almost like 5-fold, but when you have SS15 plus DOX, it increases to almost 15-fold. But when you have a cisplatin, when you have a taxol and etoposide, there is not much of significance of a difference of the production of ROS compared to SS15 condition. So after that, we did exactly the same experiment, but this time we focus on a superoxide using a DHE dye, and we figure out the same finding that DOX and cisplatin is an ROS generating, and when we have SS15 DOX and SS15 plus cisplatin, they are very much production of an ROS, and they are basically inducing a cell apoptosis. So after that, we went to see the effect on cisplatin, another anti-cancer drugs, and we figure out that you can see the cells taken before circulation in our system. They are very healthy and very very good condition. But in adhesive condition, very few apoptotic cells. You can see in the non-adhesive condition. But when you have SS15 plus cisplatin, the synergetic effect produces a huge number, significant number of apoptotic cells in case of cisplatin drugs. Then after that, we tried another two different anti-cancer drugs, basically Taxol and etoposide. This is a phase image and this is a FRET image. You can see here, even when you have a no SS, no serous stress condition, or when we have a serous stress 15 condition together with the presence of a drug, there is not much of difference in the apoptotic rate. Here again with another drugs, etoposide, no SS plus SS15 presence with the same concentration of an etoposide has not much of an difference in the apoptotic rate. So these are the quantified data, and we tried MTT assay to see whether there is a certain, and the result remain consistent. And we make a conclusion that neither Taxol nor etoposide can cause a cell damage. After that, we are interested to correlate the effect with the antioxidant together with its anti-cancer drugs. So we basically use two different antioxidant, and acetylcysteine and propyl gallate, which are very much found in our vegetables and the fruits intake. So we combine these two different antioxidants. See, here is the before circulation, the cells, and we combine SS15 plus DOX. I repeat, SS15 is the serous stress generated at the arterial condition. That is a 15 dynes per centimeter square. And you can see that SS15 plus DOX kills a huge amount of cells, you can see over here. But when we have an antioxidant NAC and propyl gallate, the effect, we cannot observe the apoptotic cells. Similarly, over here also cisplatin, when you use, you see apoptotic, but when we just add the NAC, another antioxidant, and propyl gallate, we cannot observe these apoptotic cells over here. So this is basically a quantified drugs, and we make a conclusion that antioxidant reduces the stimulated anti-cancer effect of doxorubicin and cisplatin. So after that, we try to measure the level of an ROS together with its antioxidant, and it is a very well-known fact that in a static condition, no ROS production, SS15 produces the reactive oxygen species. But when we add the only SS15 together with the antioxidant NAC and propyl gallate, there is no production of an ROS, and the result is quantified here, and significant drop in the level of an ROS when we induce. When we add the NAC and propyl gallate. So we make a conclusion that NAC and propyl gallate reduces the ROS level of cells circulated under SS15 and 3-hour condition. So this is basically an optimized condition in our system. After that, we try to use another different approach for a different another cell line. So we basically try to use a lung cancer cell lines, A549, and we did exactly the same thing, but we didn't use a special engineered cell. Previously, all the cell line that I had shown was either a metastatic.
Excuse me, Dr.
Yes.
Sagar. Yeah, you have
Yes.
5 minutes more.
Okay, sure. So previously, I have shown all the results for an engineered breast metastatic or a non-metastatic cancer cell lines. So now we are trying to use a different model of a cell line. So basically we didn't engineer the cells, and we try to use an NXin-V and a propyl iodide to quantify the results. And we see, before, and we try to observe the apoptotic or a necrotic kind of a cell death. So here before circulation, no cell death was observed, but when you have SS15, only a mechanical force, basically this is a mechanical force, you see a relatively very less apoptotic and necrotic cells. SS15 dogs, you see significant apoptotic and necrotic cells in these both condition. But when you have etoposide and taxol, there is a different, it is very much similar to the SS15 condition. So then after that, we try to use another cell line, basically a cervical cancer cell line, that is a HeLa cells, and we understand that here is the results. You can see before circulation, very healthy and very happy cells. They are a very good status. SS15, very relatively few amount of an apoptotic cells. But when you have a SS15 together dogs and SS15 together cisplatin, you can see. I just zoom. Can you see this zoom image? I just try to enlarge. Is it visible on my screen? Yeah.
Yes.
Yeah, you can see, you can see the difference here, like, from the initial state, this is basically a initial state, and like, the apoptotic cells, the dead cells can, in case of an HeLa C3. So we come to a conclusion that doxorubicin and cisplatin can destroy this CTC derived from lung and the cervical cancer as well. So so far, I just saw the two different drugs, basically cisplatin and doxorubicin, which work under the mechanism of an direct generation of an ROS. But there are different other different drugs, and we need to examine whether we need to understand the mechanisms, whether they are killing the cells through the production of an ROS or other different. So this is a very much a suitable platform to such a research. So the key results that I figured out was fluidic shear stress increases the potency of the ROS generating drugs, doxorubicin and cisplatin, but had a little effect on the non-ROS generating drugs, taxol and etoposide. So when we have a co-treatment with the SS and ROS generating drugs, that basically elevate the increase the level of an reactive oxygen species in these circulating tumor cells. Then antioxidant, which basically lower the level of an reactive oxygen species, reduces the abolish the apoptotic effect of this doxorubicin and cisplatin. So the synergetic effect of killing of SS dogs or SS or with cisplatin were confirmed in circulated lung, breast, and the cervical cancer cells. So giving quite significance of this research, there is both taxol and doxorubicins are a first line of a chemotherapy agents used for breast cancer, but there is not much of a clear guidelines like which drugs should be used in a chemotherapy. So basically, based on our result, we figure out that doxorubicin is much superior compared to taxol. And another controversial research in the field is whether antioxidant supplements should be given to a cancer patient or not. So this is a very controversial. Some doctor prescribe antioxidants should be given, not given. Recently, Nepal Health Research Council, they have banned the use of an antioxidant supplements in different cancer hospital in Nepal. So based on our results, we have figured out that antioxidant counterattack with the anti-cancer effect of these ROS generating drugs, but there needs more research to identify the effect of these antioxidants together with these anti-cancer drugs to know whether these antioxidant supplements should be given to cancer patients or not. So this work was published in a breast cancer research and treatment. And I conducted this research work in Nanyang Technological University in Singapore with the guidance of Prof. Kathy Quyen Lu and Prof. Seren Lim. So I'm very much thankful for all our lab. And this is a small part of my PhD work. And I'm very much thankful for the Singapore International Graduate Award who supported my PhD study in Singapore. So I'll be, thank you so much, everyone, for your kind attention, and I'll be happy to take a question and answer. Yeah, thank you.
Thank you, Dr. Sagar, for your nice talk. Because of time limitation, we are not going to take any question. We have some question from audience. Dr. Sagar will reply those questions in chat.
Okay.
So,
Okay. Thank you so much. Yeah.
Yeah, we have another talk on deep learning by Srishti Khonda. She is a researcher at Department of Biotechnology, Thapar Institute of Engineering and Technology, Patiala in India. Today, she is going to give a talk on application of deep learning algorithms in classifying skin lesions using ISIC imaging dataset. Srishti Khonda, please go ahead.
Thank you so much for the introduction. My topic for today would be application of deep learning algorithms in classifying skin lesions using the ISIC image dataset, which is a capstone project which I performed in my bachelor's. I am not very advanced and skilled as many of professors and doctors here are, but I am privileged to be here. Thank you so much. So the background and problem statement for my project thesis was the skin that, as we all know, skin cancer is one of the most predominant types of cancers. And looking at the statistics, we know that the frequency doubles every 20 years. And also talking about cancer, melanoma is very deadly, but survival rates are high when detected early. So distinguishing between malignant and benign melanoma is very challenging, and we wish to do that with the ABCD rule, which is the CAD rule, computer-aided diagnosis. Accuracy without the computer-aided diagnosis is about 60 to 70%, but when CAD is applied, it gives us around more than 90% accuracy. So the main objective of my project was to classify skin lesions from different application of deep learning algorithms and to evaluate the CNN model accuracy. So as I mentioned earlier, 75 to 85% is without the combination of visual inspection and dermatoscopic images by dermatologist is with the combination of these. We aim to do it for around higher than 90%. The design process included importing essential libraries and the images after loading, then categorizing and normalization. Then the data was split into train and test. So what we do is we take the data and then we split it into two halves for training the data and then testing it, so that the accuracy is much higher. And then the model is built, and then the testing of the model is done. This is on the left hand side, we can see the image of skin lesions. The second image is of the segmented lesion, which we extract by feature extraction method. Then a learning model is applied in which the input layer is there, the hidden layers are there, and output layers are there, which have a different kind of parameters set, and the predictions that the computer-aided diagnosis gives us is whether a tumor was malignant or benign. So what exactly is going around inside the model? So first, the initialization of the images are done. Then the training and testing is done, as I mentioned earlier. The system here uses convolutional neural network algorithm, which is a deep learning algorithm. And when this is applied to the data set, the model is saved and predicted for the test data. So now the evaluation of the result is done with standard evaluation metrics like accuracy, negative predicted value, positive predicted value, sensitivity, and specificity. Moving on, these are the extraction layers. the feature extraction and relation between pixels whose product gives feature map. The layers include input layer, convolutional layer, pooling layer, another convolutional layer. And then there are different layers according to what algorithm you use. So it gives us a fully connected layer at the end. So at the bottom, we can see that there is a 6x6 matrix of the image that we use of the benign or the malignant cancer. To this, a 3x3 filter is applied, and we extract the features as I showed in the segmented lesion, and then the maximum pooling is done. The results which we obtained was that the 70% of the data was utilized as training set, and 10% was used as validating set. The rest of the 20 was used as the test sets. So this division is based on the principle of Pareto, which states that for many events, roughly 80% of the effects come from 20% of the causes. So as we see here, the graph shows us that the increase in learning caused by the both training and testing data sets. So now this was the accuracy which the 4 networks gave us: the CNN, convolutional neural network, AlexNet, ResNet 50, and the VGG16. We can see that the most accurate sensitivity, specificity, and negative predicted value, positive predicted value, and accuracy was given by our proposed convolutional neural networks. This is the snapshot of the code which we used in the R software. We used R programming language, which is a statistical software, to obtain these results. And on the right hand side, we have the snapshot of the model, and in the bottom, we have a snapshot of the training data set. Now we observe that the clinical image initially, this is basically the accuracy which the model gave us. So in 0.9993 of the total models, the data set, we obtained that the model predicted the basal cell carcinoma as the basal cell carcinoma 0.9993 times, and subsequently the other carcinomas in the list. So this is the confusion matrix that we applied, which gives us the precision, which is the true positive values upon the true positive plus false negative values. Similarly, the recall values, which is the negative predicted value, and then the accuracy, and then the F1 score. So the future aspect of this project was that the CAD, which is the computer-aided diagnosis, will be able to act from clinical diagnosis to biopsy, which makes it more desirable and useful. It also improves transfer learning and considering not only the image but also the patient demographics, which helps us to have more accurate data of what the patient actually has and not gain any false positive results. Another future aspect of ours was to integration of the medical information such as the vital signs, which is the temperature, oximeter reading, the PR levels, the routine blood testing, which may give us new insights in the biology or pathology of the disease. Thank you. That's it. Any questions would be highly entertaining.
Thank you, Srishti, for your nice talk. Any question from audience?
I think I don't see any.
Hi, I have a short question. Okay, can I ask?
Yeah.
Yes, please.
Okay. So you mentioned that one of the future interests would be to combine the imaging data with some other non-imaging data, right? So what kind of challenges do you think you might have when you try to combine those sorts of data? So what we call as multimodal or heterogeneous data source, so imaging with non-imaging data. So you used convolutional neural network, I guess, or was it fully connected network you used? And how would you change that Any idea regarding that?
I think the challenges would be what we face is we would not get very accurate data sets because we used only like 3003 data initially for validation sets. So I don't think that we will get a very accurate result when we use very large data set, but that is the challenge we might face. And I didn't quite get your second question. Can you please repeat?
I was saying if you try to get other kinds of data as input in addition to imaging data, so non-imaging data, how would you change your deep learning architecture? Or do you need to change it or not? Do you have any comments on that?
At this time, I don't.
Okay, no worries. Thanks. Okay, thank you.
So
Thank you.
thank you.
Yeah,
Thank
thank you,
you.
Srishti.
Thank you.
Okay, thank you everybody for a beautiful session. We have a very important and interesting topics that we have discussed, but because of the limited time, we don't have much time for this question and answer. So we are conducting through the written question and answer in the Zoom by the chat. So with this, we are going to end our session. Thank you, Dr. Sushila, and all the speakers for being with us, and all the audiences. Now it's time for conducting another session. We have a panel discussion. So for that, I'm handing over this session to Dr. Roshan. Thank you.
Thank you, Dr. Nitish. And thank you all the speakers once again. All the talks were very exciting and it energizes the researchers probably. Okay, let's move ahead with our last session. That is panel discussion on the topic biomedical technologies in COVID-19 diagnostics. So for this panel discussion, we'll have 3 panel members, and the session team will also be there for the panel discussion. So altogether there will be 8 members in the discussion. But of course, we will take the questions from audience through Zoom chat and also in the Facebook Live. So first, before heading with the panel discussion, I would like to introduce our panel members. So we have Mrs. Jyoti Acharya. So Mrs. Acharya is a joint medical technologist and the chief of Department of Microbiology at Nepal Public Health Laboratory, NPHL Teku. Mrs. Acharya received MSc in Medical Laboratory Technology with focus on microbiology and has extensive experience in antimicrobial resistance. She is also the member secretary of AMR, Surveillance Technical Committee, and a working group member of Global Antibiotic Resistance Nepal. Our next panelist is Dr. Sunita Gautam. Dr. Gautam is currently working as a research and development manager at Sikhar Biotech Private Limited based in Kathmandu. Dr. Gautam is also affiliated with Kathmandu University as a postdoctoral fellow with an international brain research organization returnee home fellowship. She completed master's and PhD in molecular biotechnology from Saitama University, Japan. After completing PhD, Dr. Gautam also worked as a postdoctoral researcher at Riken Center for Brain Sciences, Japan. And our next panelist is Dr. Ajit Karna, a veterinarian, a microbiologist, and a public health scientist, currently working as senior scientist at Center for Health and Disease Studies, Nepal. Dr. Karna obtained his veterinary medicine degree from Tribhuvan University, Rampur. He then completed master's in public health and international public health from University of New South Wales, Australia. And he received PhD in microbiology from Colorado State University and completed his postdoctoral training from New Mexico State University. So we will start the formal session now. So let's make it very interactive. त्यसले गर्दाखेरि हामी इङ्ग्लिस नेपाली दुईटैमा डिस्कस गरौँ so that it becomes more comfortable and interactive. Okay, so मैले first ज्योति म्यामसँग कुरा गर्न चाहेँ. Please एकदम हैन, let's be comfortable and discuss and make it more interactive. I request other participants in Zoom as well. If you have any questions or if you want to build up the session, please welcome for that. अनि ज्योति म्याम, हजुर त अब NPHL मा COVID-19 डायग्नोस्टिकमै लागिरहनु भएको छ हैन? अनि यसो हाम्रो अडियन्सहरूलाई हामीलाई पनि यसो बताइदिनु, what is the primary method that is being used in Nepal for diagnosis of COVID-19?
So thanks, Roshan, for the opportunity. Till date, we are only using the real-time reverse transcription PCR, that is RT-PCR method for the detection and diagnosis of COVID-19. So at present, we are only using the RT-PCR method.
सो हाम्रोमा चाहिँ अब अडियन्सहरू टेक्निकल नन टेक्निकल होइन सबैजना हुनुहुन्छ, नन साइन्टिफिक अडियन्सहरू पनि हुनुहुन्छ, जसलाई चाहिँ अब भनौँ न यो मोलिकुलर मेथड्सहरूमा त्यति आइडिया नहोला। तपाईँले चाहिँ सिम्पल्ली एउटा प्रिन्सिपल एज अ प्रिन्सिपल बताउनु पर्दाखेरि कसरी मोलिकुलर बायोलोजी पिसिआरले कसरी काम गर्छ, कसरी कोभिड-19लाई चाहिँ डायग्नोसिस गर्छ, त्यो सर्टमा भनिदिनुहुन्छ कि?
ओके, सो हामीले जुन यो भाइरल इन्फेक्सन छ सिंगल स्ट्रेन्डेड आरएनए भाइरसबाट कज हुने भएकोले हामीले इन्फेक्सियस भाइरस भन्दा पनि त्यो भाइरस भित्रको राइबोन्युक्लिक एसिड आरएनए मात्रै डिटेक्ट गरेर हुन्छौँ। द्याट्स वाइ आई वुड फर द क्लिनिशियन्स हु थिङ्क द्याट an RT-PCR positive would mean that the patient is infectious and producing viruses or not, it might not be indicative at times. So हामीले भाइरसमा धेरै प्रकारका कुराहरू हुन्छन्। सो उसको स्ट्रक्चरल प्रोटिन्सदेखि लिएर नन स्ट्रक्चरल प्रोटिनहरूको मिसमास त्यो एन्टिजेनहरू हामी भन्छौँ। सो एन्टिजेनहरू डिटेक्ट गरेर हुन्छौँ भेरियस पिसिआरहरू मेथडबाट। अनि त्यो एन्टिजेनहरूमध्ये कमनेस्ट वन्स वी स्टार्टेड विथ द ई एन्टिजेन द्याट इज द एन्भलपमा भएको भाइरसको, यो एन्भलप भाइरस भएकोले यसको एन्भलपमा भएको प्रोटिन जुन हुन्छ त्यसको जिनलाई डिटेक्ट गर्ने इपिसिआर ई जिन थियो। त्यसपछि आरडिआरपी भनेको आरएनए डिपेन्डेन्ट आरएनए पोलिमरेज, यो भनेको एउटा एन्जाइम, यही भाइरसभित्र चाहिने एउटा इम्पोर्टेन्ट एन्जाइम हो, त्यसको डिटेक्सनबाट सो कर्मेनको मेथडबाट यो गरिन्थ्यो सुरुमा। अनि सिडिसीले गरेको चाहिँ न्युक्लियोक एसिड, सो यसको हाम्रो ब्याक्टेरियाको न्युक्लियस जस्तै यसको पनि एउटा न्युक्लियस हुन्छ जसमा क्याप्सिड प्रोड्युस हुने भएको प्रोटिन्स हुने भएकोले न्युक्लियोक एसिड भन्छौँ। अनि त्यसको एन्जिनको N1, N2, N3 भनेर सिडिसीले सुरुमा निकाल्यो पिसिआर, तर पछि N3 को राम्रो सेन्सिटिभिटी स्पेसिफिसिटी नभएकोले त्यसको प्रोब डिजाइनमै प्राइमर डिजाइनमै इस्यु भएकोले त्यो उनीहरूले रिट्र्याक्ट गर्यो। सो सुरुमा वी स्टार्टेड विथ सिडिसीको त्यो मेथड। पछि हाम्रोमा अहिले अब यदि एउटा एन्टिजेन डिटेक्ट गर्यो भने फल्स नेगेटिभ हुने चान्स बढी हुन्छ। बिरामीमा त्यो रोगको कीटाणुहरू भए पनि हामीले ड्यु टु द टेक्निकलिटी डिटेक्ट गर्न सक्दैनौँ। सो पछि हामीले मल्टिप्लेक्स भनेको एउटै पिसिआर मेथडबाट हामीले 3 थरी एन्टिजेन पनि डिटेक्ट गर्न सक्यौँ। सो त्यसमा यसको माथि एन्भलप माथि पनि स्पाइकहरू हुन्छ जुन स्पाइकले आएर हाम्रो शरीरको सेलहरूलाई समात्ने हो। सो द वेयर इट कम्स एन्ड बाइन्ड्स अन द रिसेप्टर्स इन आवर एपिथेलियल सेल्स एयरवेको त्यो स्पाइक प्रोटिनको पनि एन्टिजेन डिटेक्ट गर्न सक्ने छन्। देन देयर आर अदर्स द्याट वी हेभ नट युज लाइक हेलिकेजहरू छन्, हिमोग्लोबिनहरूवाला एन्जाइमवाला छन्, त्यो हाम्रो देशमा युज गरिएन। त सुरुमा NERDRP थियो भने अहिले वी आर युजिङ मल्टिप्लेक्स। त्यसमा पनि अर्को कमन वान छ ओपन रिडिङ फ्रेम्सहरू हुन्छन् 1A र 1B यसको कम्बिनेसन भएकोले जसमध्ये त्यो RDRP मैले अघि कुरा गरेको एउटा नन स्ट्रक्चर त्यो एन्जाइमहरू पनि प्रोड्युस हुने भएकोले सो थरीका किटहरू आए। तर अन्तिममा अहिले हामी 3टा एन्टिजेन एकैपल्ट डिटेक्ट गर्न सक्ने आरटीपीसीआर टेस्ट गर्दैछौँ। त्यसमा के हुन्छ भने एउटा जिनलाई त्यो हाम्रो डिफ्रेन्ट एन्जाइमहरूको ककटेलहरू राखेर हामीले एम्प्लिफाई गरेर हुन्छौँ। सो पहिला त्यो जिन हाम्रो एउटा टार्गेट जिन लिन्छौँ, वी आर नट टेकिङ द होल जिन है, यो कुरा पनि बुझ्नुपर्छ। सो युजली वी आर टेकिङ अ कन्जर्भ टाइप अफ जिन जुन चाहिँ अरू कोरोना भाइरसहरूसँग नमिलोस्, यसको लागि स्पेसिफिक होस्। सो यदि तपाईँले ज्योति आचार्य भनेर खोज्नुभयो भने धेरै मान्छे भेट्नुहुन्छ होला। सो हाउ डु यु नो इट्स मी द ज्योति आचार्य? सो यु निड टु ह्याभ समथिङ भेरी स्पेसिफिक टु मी। सो त्यस्तै प्रकारका कुराहरू यसमा हामीले वी टेक अ डिफ्रेन्ट मिक्सचर अनि त्यसपछि त्यही कन्जर्भ रिजनलाई कुनै प्रकारको प्राइमर प्रोबहरू बनाएर हामी जोड्छौँ अनि त्यसलाई हिट गर्यो भने त्यसले सुरुमा हामीले किनभने यो आरएनए भाइरस हो यसलाई डिएनएमा परिणत गर्नुपर्छ सो आरएनएबाट डिएनए बनाउँदा द्याट्स वेयर द रिभर्स ट्रान्सक्रिप्सन वर्ड कम्स इन सो रिभर्स ट्रान्सक्रिप्सन मेथडलाई केही समय टेम्परेचरमा डिफ्रेन्ट टेम्परेचरमा राख्नुपर्छ अनि त्यसपछि प्रोबहरू आएर जोडिन्छन् यदि मिल्ने छ भने स्याम्पलमा यदि यो भाइरस आरएनए छ भने त्यो प्रोब आएर त्यहाँ जोडिन्छ एन्ड देन द डिफ्रेन्ट एन्जाइम्स व्हेन दे are amplifying, annealing and getting disentangled, and that time के हुन्छ भने जुन हाम्रो प्रोबहरू छ तिनीहरूले फ्लोरोसेन्स देखाउँछ अनि त्यो फ्लोरोसेन्स हामीले रियल टाइममै हेर्न पाइराखेकोले यो होल मेथडको नाम चाहिँ रियल टाइम आरटीपीसीआर हुन गएको छ सो ओभरअल इन द जिस्ट वी आर डुइङ द्याट ओन्ली हामीले जिन मात्रै डिटेक्ट गरेर यो बिरामीमा आरएनए देखिएको छ कि छैन अहिलेसम्म त्यही डिटेक्ट गरिरहेको छौँ
भनेपछि यो आरटीपीसीआर भनेर यदि कोही बिरामीमा सानो मात्रामा पनि त्यो भाइरस छ भने चाहिँ त्यसैलाई ठुलो मल्टिपल कपिज बनाएर तपाईले चाहिँ फ्लोरोसेन्सको थ्रु कति मात्रा छ भने देखिन्छ र धेरै भयो भने पोजिटिभ थोरै भयो भने त्यही अनुसारले चाहिँ लेभल छुट्टिने भयो
द्याट विल अल्सो भेरी अब त्यो पनि हाम्रो कुन प्रकारको हामीले किट युज गरिरहेको छौँ कसैको लिमिट अफ डिटेक्सन एक कपी पनि थ्योरेटिकल भनेको छ कसैले 10 भनेको हुन्छ भने यहाँ हजारौं लिमिट अफ डिटेक्सन वाला पनि किट उपलब्ध भए सो इट विल अल्सो डिपेन्ड अन द लिमिट अफ डिटेक्सन अफ द किट
हजुर अब एक्चुअली म डाक्टर सुनिता गौतमसँग जान्न चाहे ज्योति म्यामले भन्नुभयो आरटीपीसीआर चाहिँ हाम्रो नेपालमा युज भइरहेछ हैन सबै अब अलमोस्ट धेरै संसारमा पनि यही चाहिँ एज अ डायग्नोस्टिक एसेको रूपमा युज भइरहेछ के यो मात्रै गोल्ड स्ट्यान्डर्ड एसे हो कोभिड-19 डायग्नोस गर्नलाई कि अरु पनि एसेजहरु अवेलेबल छ तपाईको एज अ मोलिकुलर बायोलोजिस्ट तपाईको विचार के छ यसमा
धन्यवाद रोशन सर फर द अपर्च्युनिटी अहिलेको अवस्थामा एज अफ नाउ भन्दाखेरि चाहिँ गोल्ड स्ट्यान्डर्ड अफ डायग्नोसिस अफ कोभिड-19 इज एसआरटीपीसीआर किनभने नि अब यो आरटीपीसीआर किनभने यो कोभिड-19 कज गर्ने नोभेल कोरोना भाइरस सार्स कोभ 2 जब हुवाङबाट बाहिर भनौँ न फर्स्टमा हुवाङमा डिटेक्ट भयो त्यसपछि अब यसको सिम्पटम्सहरू चाहिँ माइल्ड हुने अनि अरू जस्तो इन्फ्लुएन्जाको जस्तो भनौँ न फ्लूको जस्तो सिम्पटम्सहरू देखिने यताउता हुँदाखेरि ठ्याक्कै यसको डिटेक्सन के भन्दाखेरि जब यसको सिक्वेन्स जेनोमिक सिक्वेन्स पत्ता लाग्यो त्यसपछि चाहिँ अरू भाइरल एन्टिजेनहरूको डिटेक्सन जस्तै यसलाई पनि आरटीपीसीआरबाट डिटेक्ट गर्ने भनेर अब थुप्रै ट्राईहरू भयो अनि त्यसपछि यसको होल जेनोम सिक्वेन्स थाहा भइसकेपछि अघि ज्योति म्यामले भने जस्तै एकदमै यसको आरएनए एज अ जेनोमिक मटेरियल हुने भयो त्यो आरएनएलाई आरटीपीसीआरको रिभर्स ट्रान्सक्रिप्सनबाट सिडिएनए बनाएर अनि पोलिमरेज चेन रियाक्सनले अब जस्तो 1 कपी 2 कपी अथवा 10 कपी अफ भाइरस छ भने त्यसलाई एम्प्लिफाई गर्दै यसमा अब पेसेन्टमा यो नोभेल कोरोना भाइरस छ कि छैन भनेर टेस्ट गरिने नै अब गोल्ड स्ट्यान्डर्ड भयो अनि त्यसपछि चाहिँ ज्योति म्यामले भने जस्तै अब यो कोरोना भाइरसको थुप्रै स्ट्रक्चरल जिनहरू र अरू जस्तै ओपन रिडिङ फ्रेम जिनहरू छ अनि त्यसलाई मल्टिप्लेक्समा राखेर नट ओन्ली अ सिंगल जिन डिफ्रेन्ट जिनहरू को पीसीआर गर्दाखेरि पोजिटिभ देखिएपछि चाहिँ यो एकदमै स्पेसिफिक हुने भयो किनभने आरएनए डिजाइन गर्दा यसरी डिजाइन गरिएको हुन्छ कि जस्तै सार्सको अरू पनि सिमिलर टाइपको कोरोना भाइरसहरू जस्तै 2003 को सार्सको तिनीहरूसँग हाइली सिमिलर छ तर प्राइमर बनाउँदाखेरि यसरी बनाइएको हुन्छ कि इट इज ओन्ली प्रेजेन्ट फर द नोभेल कोरोना भाइरस SARS-CoV-2 सो एकदमै केयरफुल्ली डिजाइन गरेको प्राइमरहरू र एकदमै केयरफुल्ली बनाएको आरटीपीसीआर किटहरूले चाहिँ यसको यो जुन कोभिड-19 को पोजिटिभ जुन भाइरस छ त्यसलाई स्पेसिफिकली र एकदमै हाई सेन्सिटिभिटी डिटेक्ट गर्ने भएकोले अहिलेको अवस्थामा गोल्ड स्ट्यान्डर्डको रूपमा आरटीपीसीआरलाई प्रयोग गरिएको छ तर अब यसको सर्टेन लिमिटेसनहरू छन् जस्तै अब आरटीपीसीआरको मेन लिमिटेसन भनेको कस्तो यसलाई एकदमै सोफिस्टिकेटेड ल्याब चाहियो तपाईलाई बीएसएल लेभल 2 किनकि एकदमै हाइली कन्टाजियस भाइरस भयो अनि अर्को कुरा चाहिँ एकदमै तपाईलाई एकदम ट्रेन्ड भनौँ न मोलिकुलर बायोलोजी अथवा माइक्रोबायोलोजी चाहियो जसलाई आरटीपीसीआर एकदमै राम्रोसँग गर्न आउँछ किनकि तपाईले सानो मात्र हेलचक्राइ गर्नुभयो स्याम्पल कलेक्सनदेखि लिएर आरटीपीसीआर को स्याम्पल प्रिपरेसनमा सानो मात्रै मिस्टेकले गर्दा तपाईँको एकदमै फल्स पोजिटिभ अथवा एकदमै फल्स नेगेटिभ रिजल्ट आउने हुन्छ। त्यही भएर एकदमै वेल ट्रेन्ड हामीलाई म्यान पावर चाहियो। अनि अर्को कुरा चाहिँ आरटीपीसीआर किट पनि आफैमा एकदमै महँगो छ र आरएनए भाइरस एक्सट्र्याक्सन गर्नको लागि जुन किट आरएनए भाइरस किट एक्सट्र्याक्सन जुन चाहिन्छ त्यो पनि अति महँगो छ। अनि भिटिएम पनि चाहियो। त्यो हरेक कुराले गर्दाखेरि अब आरटीपीसीआर गोल्ड स्ट्यान्डर्ड भए पनि जस्तै नेपाल जस्तो डेभलपिङ कन्ट्री र अझै दूर दराजमा जस्तै भनौँ न कर्णाली प्रदेशमा को त्यहाँको ठाउँमा त्यहाँको गाउँमा गएर हामीले आरटीपीसीआर अहिले सेटअप गरेर त्यहाँ पेसेन्टहरूको डायग्नोसिस हुन सकेको छैन। त्यो हुँदाखेरि हामीलाई चाहिँ अब अल्टरनेटिभ आरटीपीसीआर को अल्टरनेटिभ डायग्नोस्टिक टेस्टहरू चाहिएको छ जुन चाहिँ एकदमै ऱ्यापिडली डेभलप कन्ट्रीहरूमा डिफ्रेन्ट अप्सनहरू आइरहेको छन्। र अब नेपालको केसमा चाहिँ अहिलेसम्म आरटीपीसीआर छ, तर अब अरू अल्टरनेटिभ केस जस्तै एन्टिजेन बेस्ड डिटेक्सन किट ऱ्यापिड के भन्छ यो लेटरल फ्लो असैबाट अथवा एलाइजा बेस्ड डिटेक्सन किट अनि सेरोलोजिकल डिटेक्सन किट भनेर सेरोलोजिकल चाहिँ नट फर द डायग्नोसिस बट फर द सर्भेलेन्स इपिडेमियोलोजिकल स्टडी त्यस्तो खालको। र अहिले रिसेन्टली चाहिँ यो क्रिस्पर जेनोम एडिटिङ टेक्निक भनेर भन्छ, त्यसको डिटेलमा म पछि भन्न होला। अब
त्यो बारे हामी पछि फेरि डिस्कस गरौँला।
हजुर, अनि त्यो हुँदाखेरि थुप्रै खालको अहिले एफडीएबाट इमर्जेन्सी युज अथोराइजेसन पाएको अल्टरनेटिभ किटहरू पनि आएको छ र होपफुल्ली नेपालमा पनि त्यो किटहरू ल्याएर फर्स्टमा इन्टरनल भ्यालिडेसन, किनभने अलरेडी एफडीएबाट इयुए पाइसकेको भएपछि अब किटमा शङ्का गर्नुपर्ने अवस्था भएन होइन। तर पनि नेपालको कन्टेस्टमा यहाँको टेक्निकल स्टाफहरूले ह्यान्डलिङ गरेर कतिको रिप्रोड्युसिबल रिजल्ट आउँछ आउँदैन त्यो भेरिफाई गरिसकेपछि डेफिनेटली फ्युचरमा चाहिँ आरटीपीसीआर को आई डोन्ट से टोटली अल्टरनेटिभ or the replacement of the RT-PCR, but as a complement of RT-PCR चाहिँ नियर फ्युचरमा नेपालमा आउँछ भन्नेमा चाहिँ म विश्वास गर्छु।
हजुर, थ्याङ्क यू डक्टर। Actually, Dr. Ajit जान्न चाहेँ। Dr. Ajit has been involved in यो monitoring COVID-19 situation होइन, पहिला चाहिँ आउट अफ भ्याली पनि, अनि अहिले हाम्रो ज्योति म्यामले पनि भन्नुभयो, डक्टर सुनिताले पनि भन्नुभयो आरटीपीसीआर को पोजिटिभ कुराहरू र कसरी त्यसलाई चाहिँ युज भइरहेछ नेपालमा। के छ तपाईँको विचारमा यो आरटीपीसीआर युज गरेर हामीले जति टेस्टिङ गर्नुपर्ने क्यापेसिटी छ त्यो भइरहेछ कि छैन? जस्तो काठमाडौँमा त त्यहाँ प्राइभेट ल्याब्सहरू पनि छन् र हाम्रो एनपीएचएलले पनि त्यतिकै सकेसम्म ज्यादा टेस्टहरू गरिरहेको छ डेली। तर जस्तो डक्टर सुनिताले भन्नुभयो अघि लिमिटेसन छ होइन, इन्फ्रास्ट्रक्चर चाहियो, ह्युमन रिसोर्सेस चाहियो। कस्तो छ आउट अफ भ्याली चाहिँ यसको सिचुएसन कस्तो छ?
Thank you, Roshanji, thank you NRN for allowing me this opportunity to talk to you guys. यसमा चाहिँ कस्तो छ भने अब म्याक्स सबै ग्राउन्ड वर्क चाहिँ सुनिताजी र ज्योति म्यामले भनिसक्नु भयो होइन, लिमिटेसन कहाँ छ, के गर्न सकिन्छ, के गर्न सकिँदैन। सो थ्याङ्क्स फर सेटिङ अप मी फर द्याट स्टेज होइन। र बाहिर म कोभिड सर्भिलेन्सको लागि सुरुदेखि नै बाहिर काठमाडौँभन्दा बाहिर काठमाडौँमा पनि जे होस् घुमे भनौँ होइन। र त्यो घुम्ने क्रममा चाहिँ मैले राम्रो राम्रो एक्सपिरियन्स पनि बेस्ट प्र्याक्टिसेसहरू पनि भेटेँ र कति ठाउँमा इम्प्रुभमेन्ट गर्नुपर्ने ठाउँहरू पनि भेटेँ होइन। र आरटीपीसीआर डेफिनेटली गोल्ड स्ट्यान्डर्ड राम्रो छ यदि त्यो लेभल अफ क्यापासिटी डेभलप भएको छ भने होइन। तर कतिपय जिल्लाहरूमा त ल्याब पनि छैन होइन, ल्याब छैन र त्यो जिल्लाबाट भनौँ न त्यो जिल्लामा कुनै एउटा जिल्लामा एउटा मात्र ल्याब छ भने नजिकको अरू 2-3 वटा जिल्लाले उसैलाई पठाउँछ स्याम्पल होइन। त्यो सबै ठाउँमा छैन बाहिर। इभन सुनिता म्यामले अघि भन्नुभयो कर्णाली प्रदेशमा होइन गाह्रो छ होइन। तपाईँ भनौँ 4वटा ल्याब छ त्यहाँ कर्णाली प्रदेशमा जसले चाहिँ कोभिडको टेस्ट गर्छ। त्यहाँ 10 वटा जिल्ला छ, 4वटा ल्याब छ होइन। त्यो प्रेसर टेक्न सक्छ कि सक्दैन होइन। उसले के गर्नुपर्छ भने पछि अरूको प्रदेशमा अथवा अरू ठाउँमा स्याम्पल पठाउनु पर्ने आवश्यकता देखिन्छ। र ल्याब भए पनि त्यहाँनिर के छ भने अब आरटीपीसीआर मेसिन त छ होइन, तर अब म्यानुअली एक्सट्र्याक्ट गरिरहेको छ होइन। भनेपछि त्यो जुन टाइम टु रिजल्ट भन्ने चाहिँ अलिकति त्यस्तो कन्डिसनमा 8 घण्टा अथवा 9 घण्टा पनि लाग्न सक्छ। अब अटोमेटिक एक्सट्र्याक्सन भए पनि अब फुल्ली भन्छन् हैन, तर पनि त्यो फुल्ली त के भन्ने मान्छेले आफैले राख्नु नै पर्ने हुन्छ। त्यसमा चाहिँ सेमी अटोमेटिक भन्नु पर्यो हैन। र त्यो भए पनि रिजल्ट चाहिँ यस्तै हाराहारी 4-5 घण्टामा आउने गरिरहेछ। त्यही भएर आरटीपीसीआर गोल्ड स्ट्यान्डर्ड राम्रो छ हैन, र वाइडली युज टेक्नोलोजी हो, अलरेडी प्रुभन मेथड पनि हो। त्यसको लागि त एक किसिमको स्टेज छ हाम्रो देशमा, डिफ्रेन्ट ठाउँमा ल्याबको क्यापासिटी अलिकति डेभलप गर्नुपर्ने, मान्छेहरू जुन काम गरिरहेछन् त्यो उहाँहरू चाहिँ आफै पनि इन्फेक्सन प्रिभेन्सन कन्ट्रोलमा ध्यान दिएर अनि एसओपीहरू फलो गर्ने त्यो चाहिँ गर्नुपर्ने हुन्छ। र ट्रेन म्यान पावरको समस्याले गर्दाखेरि आरटीपीसीआरै ल्याब भए पनि त्यो इन्टरप्रिटेसनमा चाहिँ गल्ती हुन सक्ने सम्भावना बढी हुन्छ।
राइट। अनि जस्तो डाक्टर देवी बस्नेत, डाक्टर इन्दिरा तिवारी साउथ कोरियामा हुनुहुन्छ। अब साउथ कोरिया त वान अफ द एक्जाम्पलरी देशै भयो होइन, कसरी मिटिगेट गर्यो कोभिड-19 लाई। त्यहाँ चाहिँ कस्तो रहेछ, के इन्फर्मेसन छ हजुरसँग त्यहाँको डायग्नोस्टिक टुलहरू बारे, अथवा चाहिँ कसरी गरिरहेछ, अथवा यही कुरामा तपाईहरु के थप्न चाहनुहुन्छ?
कोरियामा चाहिँ डायग्नोस्टिक कुरा मात्रै गर्ने हो भने अघि म्यामहरूले भन्नुभयो नि, अहिले फर्स्टमा त त्यही नै हो, यहाँको चाहिँ आरटीपीसीआर नै हो। अघि अजितजीले भने जस्तै यो आरटीपीसीआर पनि तपाईँको हाउ इफिसियन्ट अनि हाउ इफिसियन्टली इज डन होइन। अनि त्यसपछि त्योभन्दा पनि इम्पोर्टेन्ट कुरा के छ भनेदेखि तपाईँको कुन किट युज गर्नुहुन्छ र त्यो किट कुनमा अप्टिमाइजेसन गरेको छ, कुन मेसिनमा अप्टिमाइज गरेको छ भन्ने कुरा एकदमै इम्पोर्टेन्ट हुन्छ। अनि एज अघि म्यामले भने जस्तै, ज्योति म्यामले भने जस्तै यो चाहिँ स्टेप बाई स्टेप चाहिँ के अरे त्यो बटल नेकहरू हुन्छ होइन। तपाईँको चाहिँ भाइरस आइसोलेसन गर्नेदेखि लिएर होइन, एमआरएनए आइसोलेसन गर्नेदेखि लिएर त्यसलाई ट्रान्सफर गर्ने विभिन्न स्टेप्सहरू बटल नेकहरू हुन्छ। त्यसैले आरटीपीसीआर मात्रै भन्दा पनि त्यसका चाहिँ स्टेप्सहरू हामीले इफिसियन्टली ह्यान्डल गरेको छ कि छैन भन्ने इम्पोर्टेन्ट भयो एउटा। अनि अर्को कुरा चाहिँ जस्तो टाइम अफ टेस्ट, कहिले चाहिँ त्यसलाई चाहिँ टेस्ट लिएको छ अनि त्यसको चाहिँ नम्बर अफ कपीहरू कति छ तपाईँको स्याम्पल्समा इज भेरी इम्पोर्टेन्ट। त्यो डिपेन्ड्स अन द किट्स। मलाई जहाँसम्म याद छ हामीले एकचोटि कोरियाबाट पठाएका थियौँ, सेजिनको किट पठाएका थियौँ, जो चाहिँ अघि ज्योति म्यामले भन्नुभएको थियो। यसले चाहिँ सिमल्टेनियसली 3वटा ओआरएफहरू जिनहरू चाहिँ यसले मल्टिप्लाई गर्छ, जसले गर्दाखेरि चाहिँ दुईटै खालको डिटेक्सन प्लस कन्फर्मेसन टेस्ट एकैचोटि हुन्छ। अब मलाई थाहा छैन नेपालमा अलिअलि मैले त्यो बिचमा कम्युनिकेट गर्दाखेरि चाहिँ दुईचोटि टेस्ट गरिन्थ्यो, एउटा चाहिँ डिटेक्सन टेस्ट गर्ने अनि त्यसपछि फेरि त्यही स्याम्पललाई चाहिँ अर्कोचोटि पिसिआर रन गरेर चाहिँ कन्फर्मेटिभ टेस्ट गरेर पनि गरेको भन्ने सुनेको थिएँ सुरुमा। अहिले रिसेन्टली मेबी अलरेडी चेन्ज एभ्रिथिङ होइन, मलाई थाहा छैन। अब मेन चाहिँ त्यही नै हो। अब अहिले जब यो चाहिँ सुरुको केसहरूमा भयो, अब जब अहिले चाहिँ म्याक्सिमम नम्बरहरू बढ्न थालिसक्यो, त्यसपछि तपाईँको चाहिँ जिन एम्प्लिफिकेसन भन्दा पनि एन्टिजेन डिटेक्सन एन्टिबडी युज गरेर एन्टिजेन डिटेक्सन सिस्टमहरू पनि युज गरेको छ। अब जो मेरो विचारमा अहिले यो अलिकति हाई मेनी पपुलेसनहरूमा बढी देखिसकेपछि ऱ्यापिड टेस्टहरूलाई हामी त्यो पनि भन्छ क्या होइन, ऱ्यापिड टेस्ट। त्यहाँ दुईवटा कुरा छ जस्तो लाग्छ मलाई, एउटा चाहिँ एन्टिबडी टेस्ट गर्ने, अर्को चाहिँ एन्टिजेन टेस्ट गर्ने, एन्टिबडी युज गरेर एन्टिजेन टेस्ट गर्ने खालको त्यो टेस्ट छ। तर कोरियाको केसमा चाहिँ एकदम एक्जाम्पलरी नै हो यो कन्ट्री। यसमा विभिन्न स्टेप्सहरू छ, जस्तो तपाईँको यसको मेन पार्ट चाहिँ के भनेदेखि त्यसलाई एक्सपान्ड हुन नदिने टेक्नोलोजी मतलब मेथोडोलोजी। एक दुईजनालाई जहाँ डिटेक्ट भयो त्यसको चाहिँ ट्रेसिङ गर्ने एकदमै राम्रो यहाँको चाहिँ मेथड छ। यसमा चाहिँ यिनीहरूले हेल्थको लागि चाहिँ यो प्राइभेसीलाई कन्सिडर गर्दैन, पब्लिक हेल्थ इज सबभन्दा फर्स्ट प्रायोरिटी। त्यसैले जस्तो कोही मान्छेलाई त्यस्तो खालको चाहिँ डिटेक्सन भएको छ भने त्यो चाहिँ कम्प्लिटली फलोड हुन्छ, नम्बर 1। नम्बर 2 यहाँ एकदमै हाइली के भन्छ लिगल्ली चाहिँ बढी स्ट्रिक्ट छ। मैले चाहिँ मास्क जस्तो उनीहरूले अनाउन्स गरिसकेपछि मास्क लगाएर हिँड्नु हुँदैन भनिसकेपछि मास्क लगाएर हिँडेन भने त्यसलाई तपाईँको दण्डहरू दिएको छ होइन, त्यो चाहिँ स्ट्रिक्टली लागु हुन्छ। त्यसैले यहाँनिर अलिकति सबै डिसिप्लिन पनि छ अनि स्ट्रिक्ट लजहरू पनि भएको कारणले गर्दा यो चाहिँ एक्सपान्ड हुने काम कम भयो। अनि डिटेक्सन सिस्टम त तपाईहरुलाई थाहा छँदैछ कोरिया इज वान अफ द बेस्ट इन टेक्नोलोजी उनीहरुले चाहिँ फेरि त्यो टेक्नोलोजी मात्रै हैन यिनीहरुको चाहिँ मल्टिपल कम्पनीहरुको चाहिँ उनीहरुको डेभलपमेन्ट सिस्टमहरु छ तपाईको यहाँबाट एक्सपोर्ट पनि गरेको छ त्यसैले मेरो विचारमा यो उनीहरुको हाई टेक हैन एफिसिएन्ट वर्किङ स्टेप्स त्यसपछि रिपिटेडली टेस्टिङ स्टेप्स अनि अर्को कुरा भन्नुपर्दा चाहिँ यो कन्ट्रोल्ड एक्सपान्डिङ यी सबै कुराले चाहिँ कोरियामा कन्ट्रोल भएको छ बीचमा यति हुँदाहुँदै पनि अस्ति बीचमा अलिकति बढेको थियो तर स्टिल दिस इज फाइन त्यति धेरै भनौँ न अहिलेसम्म म आफै यहाँ पनि काम गर्ने भएको भएर हामीले अहिले कहिले पनि हाम्रो अफिस बन्द गरेनौँ हैन तर प्रत्येक अफिसमा यदि स्ट्रिक्ट रुल्सहरु छ मोनिटरिङ हुन्छ त्यो गभर्नमेन्टले दिएको रुल्सहरु लागू गरेको छ कि छैन भनेर उनीहरुको टोटली मोनिटरिङ हुन्छ त्यसैले स्टिल वी आर फाइन हियर
हजुर थ्यांक यू डाक्टर बस्ने डाक्टर इन्दिरा के थप्न चाहनुहुन्छ यसमा
एज डाक्टर देवी दाइले भनिसक्नु भएको थियो होइन यहाँ चाहिँ हरेक चिजको टेक्नोलोजी प्लस यो बायो इन्फर्मेटिक्स सबै कुराहरू हजुरको कतै अब भनौँ न कुनै ठाउँमा कोही पेसेन्ट चाहिँ डिटेक्ट भएको छ भने हामीलाई इमर्जेन्सी अलर्ट आउँछ फोनमा होइन कुन ठाउँमा भएको थियो कुन उसमा भएको थियो भनेर त्यो सबै डिटेल इन्फर्मेसन आउँथ्यो अहिले चाहिँ के भएको छ भने हजुरहरू रेस्टुरेन्टमा जानुहुन्छ भने खाना खाइसकेपछि हजुरले आफ्नो रेकर्ड दिएर आउनुहुन्छ इन केस त्यहाँ कसैलाई कोरोना भएको छ भने हजुरलाई अलर्ट आउँछ हजुरले चाहिँ त्यसपछि गएर टेस्ट गर्नुपर्ने हुन्छ हिजो कि अस्ति भर्खरै के भएको छ भने एकजना कोरियनले कन्ट्याक्ट ट्रेसिङमा चाहिँ यताउता गडबडी गरेको हुनाले उसलाई 6 महिनाको सजाय दिएको छ होइन इभन कोरियनलाई नै नेपालीहरूले के अरे एकजना भियतनामीलाई चाहिँ रिपोर्ट पनि गरिसकेको छ यहाँ त्यसले गर्दाखेरि यहाँ चाहिँ एकदमै फलोअप चाहिँ भइरहेछ हामी ल्याबहरू जाँदाखेरि पनि हामीलाई पनि सधैँ चेक गरेर होइन हामी त्यसरी गइरहेका छौँ अनि अर्को कुरा के छ भने मैले यसो फेसबुकमा लाइभमा हेर्छु कुनै साथीहरूले अमेरिकातिर क्यानाडा अस्ट्रेलियातिर उहाँहरू पार्कमा जाँदा मैले मास्कको त्यति युटिलाइज गरेको देखिनँ तर यहाँ देवदाइले भने जस्तै गरी यहाँ चाहिँ कम्पल्सन छ मान्छे घुम्न गइरहेछ तर मास्क लगाएर चाहिँ हिँडिरहेका छन् कि गभर्मेन्टको रुल्स एन्ड रेगुलेसन्स चाहिँ एकदम राम्रो गरेको छ अनि बिचमा कोरियाले कसरी कन्ट्रोल गर्यो भन्दाखेरि यिनीहरूको चाहिँ स्याम्पलिङ हजुरको ड्राइभ थ्रुमा जहाँ जहाँ ड्राइभ थ्रुहरू चाहिँ त्यहाँनिर चाहिँ उनीहरूले स्याम्पल कलेक्सन गर्यो होइन स्याम्पल कलेक्सन गर्यो अनि विदिन 30 मिनट चाहिँ नियर बाई ल्याबमा चाहिँ यिनीहरूले स्याम्पल प्रोसेसिङ गरे होइन त्यही हुनाले चाहिँ यहाँ एकदमै फास्टमा चाहिँ टेस्टहरू हुने अनि यहाँ चाहिँ के पनि भयो भने 24 आवर्स हजुरको ल्याब इज रनिङ त्यसले गर्दाखेरि हजुरको यति धेरै टेस्टहरू भयो जति मान्छेहरू पोजिटिभ आए उनीहरूको चाहिँ सबै इन्फर्मेसन चाहिँ हजुरको मोबाइलमा आइरहेको छ कुन ठाउँमा कुन मान्छे कतिखेर कुन रेस्टुरेन्टमा गयो त्यो यो हरेक मिनटको चाहिँ उनीहरूले इन्फर्मेसन फ्लो गरिरहेका छन् कि त्यसले गर्दा मान्छेहरू अलर्ट भएर त्यो रेस्टुरेन्टमा चाहिँ यो एरियाको त्यो ठाउँमा चाहिँ त्यो उसमा मान्छेहरू इन्फेक्टेड भएको रहेछ भनेर त्यसलाई सिल गरे मान्छे त्यहाँ जाँदैन गएको मान्छे पनि अनि यहाँ गभर्मेन्टले एलोकेटेड हस्पिटल्सहरूमा गएर त्यहाँ टेस्ट गरेको छ कि त्यसले गर्दाखेरि यहाँ एकदमै सजिलो भएको हो हामीले चाहिँ हाम्रै हस्पिटल पनि एउटा गभर्मेन्ट एलोकेटेड हस्पिटल भएको हुनाले हामीले नयाँ नजिकबाट चाहिँ निहाल्ने अवसर पाएको हुनाले चाहिँ यो कुराहरू थाहा पाएको हो
थ्याङ्क यू फर सेयरिङ इन्फर्मेसन अब डाक्टर नितेश पनि दोहामा हुनुहुन्छ कतारमा त्यहाँ चाहिँ के छ अब हजुर त आफै फिजिसियन हुनुहुन्छ पेसेन्ट डेली पेसेन्ट हेर्नुहुन्छ कस्तो छ त्यहाँको स्थिति त्यहाँको डायग्नोस्टिक टेक्नोलोजीहरू के छ केही फरक छ हाम्रो देशमा भन्दा
यस्तो हो अब गर्ने त संसारभरि सेम नै हो आरटीपीसीआर नै हो हैन तर अब कस्तो हुन्छ भने कतिपय कुराहरू अब यहाँ धेरैलाई थाहा होला फल्टी टेक्निक्सहरूबाट पनि फल्स नेगेटिभहरू आउने हुन्छ अब त्यो टेक्निक्सहरूमा चाहिँ यिनीहरूले अलिकति बढी जोड दिएको थियो सुरुदेखि नै हैन अनि त्यो ट्रेनिङ दिने स्टाफहरूलाई रेगुलरली अनि यहाँको चाहिँ कस्तो भने मोनिटरिङ चाहिँ सेन्ट्रल तपाईको जतिसुकै जहाँसुकैबाट पनि तपाईले स्याम्पल लग्नु भयो भने चाहिँ सेन्ट्रल ल्याबमा पठाउनु पर्यो तपाईले स्याम्पल कलेक्सन चाहिँ जहाँ पनि स्वाब कलेक्सन जसलाई पनि गर्न दिनु भयो प्राइभेटमा गर्न पाउने भयो तर रिपोर्टिङ चाहिँ तपाईको हाम्रो सेन्ट्रल ल्याबोरेटरीबाट मात्रै हुने भयो त्यही जाने भयो अल्टिमेटली सबै स्याम्पल त्यहाँ जाने भयो र त्यहाँबाट रिपोर्ट आउने भयो हैन अनि त्यस्तो गर्दाखेरि चाहिँ यहाँ पनि हाम्रो यो मिडल इस्टमा भनौँ कतारमा चाहिँ सबभन्दा बेस्ट पार्ट भनेको चाहिँ जस्तो एउटा ट्रान्जिसन टाइम हुन्छ नि जस्तै अब तपाईलाई सिम्पटम्स भयो अनि तपाई गएर हस्पिटलमा स्वाब दिनुभयो अनि त्यसपछि तपाई घर आउनु पाउनु भयो हैन घर आउनु भयो अथवा तपाई वेट गर्दाखेरि तपाईलाई मोनिटर गरिएको हुन्छ कि त्यो टाइममा पनि तपाईले चाहिँ अरूलाई सार्न पाउनु भएन किनभने तपाईको इमिडिएटली तपाई सस्पेक्टेड भयो भने हामीहरुसित एउटा मोबाइल एप्लिकेसन हुन्छ जुन चाहिँ यहाँको हाम्रो आइडी कार्डसँग लिंक भएको हुन्छ आइडी कार्डसँग लिंक भएको मोबाइल एप्लिकेसनमा तपाई टेस्ट गर्न गएपछि त्यसमा 3-4 वटा कलर कोडिङ हुन्छ अनि त्यो कलर कोडिङमा यु क्यानट गो इन्टु द मार्केट क्राउडेड प्लेसेसहरु तपाई टेस्ट गर्न जानु भने सपोज तपाईको येल्लोमा राखिदिन्छ तपाई सस्पिशियस भयो तपाईको रिजल्ट आओस् चाहे नआओस् हैन अनि भोलिपल्ट भनौं 24 आवर्स अब किनभने यत्रो धेरै स्याम्पल जाँदाखेरि अब जतिसुकै गरे पनि एटलिस्ट 24 आवर्स त रिपोर्ट दिन लाग्छ त्यो दिँदाखेरि चाहिँ 24 आवर्स पछि यदि तपाईको नेगेटिभ भयो भने तपाईको ग्रीनमा गयो सिग्नल हैन and then you can move around यदि तपाईको पोजिटिभ आयो भने रेड भयो र तपाईलाई रेडमा दिनु भन्दा पहिले उनीहरुले फोन गरेर तपाईलाई प्रोपर आइसोलेसन कहाँनेर लिएर जाने हो उनीहरुले सुरुमै प्लानिङ गरेर लिएर जान्छ कि त्यो टेक्नोलोजी चाहिँ हामीले नेपालमा पनि सुरुदेखि नै हामीले एनआरएन को थ्रुबाट पनि किनभने अहिलेको जमानामा स्मार्टफोन सबैसित हुन्छ हैन किनभने यो बिचको ग्यापले पनि अब जस्तै सिम्प्टोमेटिक पेशेन्टहरुको बिचमा यो जुन ग्यापमा चाहिँ टेस्ट गर्न दियो अनि फेरि उ घरमा जान्छ मजाले सबसित बसेर गफ गर्छ अनि भोलिपल्ट रिपोर्ट आउँदाखेरि चाहिँ पोजिटिभ हुँदा अरूलाई धेरैलाई सारिसकेको हुन सक्छ भन्ने चाहिँ एउटा हाम्रो पहिलेदेखिको एनआरएनमा हाम्रो भनाइ उठाइरहेको हो त्यो हिसाबले चाहिँ यहाँ अलिकति त्यो राम्रो छ र अर्को पार्ट भने चाहिँ अब 1,20,000 जति पोजिटिभ हुँदाखेरि पनि अब भनौं न लकडाउन पनि एकदम धेरै खासै भएन यहाँनेरि सबैलाई एवेयरनेसको प्रोग्रामहरु एकदम राम्रै गर्यो हैन र सेन्ट्रल मोनिटरिङ सेन्ट्रल ल्याबोरेटरी अनि त्यसपछि टेस्टिङहरु सबै फ्री भएपछि अब मान्छेहरुलाई भनौं न अलिकति सजिलो त यहाँ त भयो अब तर मलाई थाहा छैन नेपालमा चाहिँ सुरुमा किन एकदम धेरै नेगेटिभ आयो अब मैले सुन्दाखेरि चाहिँ सुरुमा चाहिँ नेपालमा र्यापिड डायग्नोस्टिक टेस्ट पनि एप्रुभ गरेको थियो जसले चाहिँ इनिशियल इन्फेक्सनहरुलाई मिस गरेको हुन सक्छ त्यसले गर्दा पनि अहिले एकैचोटी यो धेरै धेरै मात्रामा देखेको हुन सक्छ तर अहिले यहाँनेर हाम्रोमा चाहिँ अब त्यो र्यापिड डायग्नोस्टिक टेस्ट भन्नाले एन्टिबडी जुन हेर्छौं नि त्यसमा पनि आइजीएम र आइजीजी बथ अब त्यसमा आइजीएम मात्रै पनि हेर्ने आइजीएम भने चाहिँ अर्ली इन्फेक्सनको भयो आइजीजी भने चाहिँ लेटको भयो हैन त्यो दुईटै कम्बाइन गरेर यो किट्सहरु अवेलेबल छ यहाँनेरि दुईटै पोजिटिभ हुँदाखेरि चाहिँ यो सर्भाइलेन्समा चाहिँ जस्तै अब स्कुलमा भयो अनि यो फ्याक्ट्रिजहरुमा इन्डस्ट्रिजहरुमा चाहिँ अब त्यसलाई अहिलेलाई चाहिँ एप्रुभल दियो किनभने वी आर इन स्टेबल फेज नाउ अहिले चाहिँ हाम्रो पोजिटिभ आएको छैन अब त्यसलाई चाहिँ हामीले चाहिँ अब यहाँको सरकारले चाहिँ अब वाइड स्प्रेड चाहिँ सर्भाइलेन्स गर्दैछ हैन अब के गर्ने कसो गर्ने भनेर प्लानिङको लागि आई थिंक दैट इज द बेस्ट पार्ट दैट वी आर हियर एन्ड मलाई लाग्छ नेपाल सरकारले पनि त्यो गरेदेखि राम्रो हुन्थ्यो जस्तो लाग्छ स्मार्ट टेक्नोलोजीहरु आएर हैन
हजुर त्यो मोस्टली चाहिँ फोकस त अब कन्ट्र्याक्टेसन त गर्नै पर्यो अनि एक्चुअली म फेरि ज्योति म्यामकोमा आउन चाहे अघि डाक्टर अजितले डाक्टर सुनतालाई पनि भन्नुभयो यो आरटीपीसीआर चाहिँ मल्टी स्टेप्स प्रोसेस दैट रिक्वायर्स लाइक ट्रेन ह्युमन रिसोर्सेस हैन हु क्यान इन्टरप्रिट इट करेक्टली नेपालमा त मोलिकुलर बायोलोजिस्टहरु त्यति अझ पनि छैन कमनली कस्तो च्यालेन्जेस फेस गर्नुभयो तपाईहरुले जब यो कोभिड-19 सर्ज भयो एनपीएचएल मा भनौं न ह्युमन रिसोर्सको कमी होला टेक्नोलोजीको कमी होला कसरी तपाईहरुले कोप अप गर्नुभयो त्यो बेला चाहिँ के सम्झिनुहुन्छ
अब यस्तो त्यसबेला हाम्रोमा बाइ चान्स त्यो डब्ल्युएचओ बाट एकजना जोनाथन भन्ने आउनु भएको थियो जसले आफ्नो थीसिस इन्फ्लुएन्जामा गरे पनि उहाँ ठ्याक्कै आउनुभयो अनि हाम्रोमा एकजना माइक्रोबायोलोजिस्ट हुनुहुन्थ्यो एनआइसी मा डाक्टर रञ्जित अनि उहाँ दुईजनाले स्टार्ट गर्यो सुरुमा अनि एनआइसी कै हाम्रो स्टाफहरु जुनियर जुन अब आरटीपीसीआर सँग दे वर फेमिलियर सो दे युटिलाइज दोज पर्सनल अनि पछि बिकज फर्स्टली एज डाक्टर नितेश already told कि we were getting very less positives and we were only following those CDC and top Tim mobile kits and later when we saw the first case and after that another case so we then planned and all of the lab people who had experience in PCR just मैले पनि bacteriology सम्बन्धी PCR गरेको छु virus सम्बन्धी नगरे पनि PCR को procedure त सेमै हो so in our country molecular biologists are there but within the government sector we have very less number of people who have exposure to molecular जस्तै हाम्रो विष्णु उपाध्याय सर हुनुहुन्छ उहाँले इन्फ्लुएन्जा को 2009 को पेन्डेमिक को बेलामा धेरै काम गर्नुभयो and some people are there in the government sector so त्यस बेला हामीले के गर्यौं त we looked at people who were already trained internationally or nationally in PCR उनीहरुले हामीले लिड गर्न लगाएर अरु जुन हाम्रो मास्टर्स को र टेक्नोलोजिस्टहरु छन् सबैलाई मोबिलाइज गर्यौं so we did it in steps पहिला यो कोर टिमले काम गर्छ, त्यसपछि विथ द सर्च वी एड अन मोर पिपल। अनि पहिला त हामीले म्यानुअल एक्सट्र्याक्सन नै गरिरहेको थियौँ, बट वेन द स्याम्पल्स स्टार्टेड कमिङ फ्रम अल ओभर द कन्ट्री, अनि त्यसपछि हामीले अटोमेटेड एक्सट्र्याक्सन मगाउनु पर्ने भयो, किनभने जति काम गर्दा पनि 24 घण्टा काम गर्दा पनि तपाईँले 900 भन्दा बढी काम गर्न सकिराख्नु भएको छैन, बिकज द मेथड टुक लाइक एक्सट्र्याक्सनको लागि नि 2.5 घण्टा लागिदिने। अनि त्यसपछि अब अटोमेटेड आएपछि पनि स्टिल विथ द प्रेजेन्ट म्यान पावर वी आर फिलिङ कन्स्ट्रेन्ट्स कि हामीलाई टार्गेट दिएछ 3500 को, विच इज नट फिजिबल विथ द प्रेजेन्ट नम्बर अफ मसिन्स। तर अब हाम्रोमा आरटीपीसीआरको मसिन पनि थपिएको छ। सो यस, होपफुली in the near future we'll be increasing the throughput. So turnaround time को कुरा हो, RT-PCR मा तपाईले 4 देखि 6 घण्टा त मिनिमम लाग्छ, whether you use automated or manual extraction। किनभने इन्टरप्रिटेसन को कुरा पनि आयो, इन्टरप्रिटेसन पनि हामीले सीधै we don't have that type of interpretive software that it will directly go to the software, so you manually interpret and you manually enter the results, and then it has to go through another validation, कसले verify गर्ने हो। So the overall procedure does take some time, and yes, it was difficult, but all of the team present in our NPHL, जसले कसैले HIV मा काम गरिरहेको थियो, कसैले हेपेटाइटिस मा काम गरिरहेको थियो, कसैले ब्याक्टेरिया मा गरिरहेको थियो, सबैजना हामी we did have some masters in microbiology, everybody was mobilized, and some extra people were also hired on a short term basis, अनि हामी सबैजनाले मिलेर नि गरेको।
ज्योति म्याम, एउटा क्वेशन म राखौँ रोशन? हाम्रो देशमा कतिवटा टेस्टिङ सेन्टर्सहरू छन् ओभर द कन्ट्री, एन्ड सबै गभर्नमेन्ट सेन्टरहरूले नै टेस्ट गर्छ कि कुनै प्राइभेट सेन्टर्सहरू पनि छ?
अहिले त प्राइभेटमा पनि भइसक्यो, आई डोन्ट हेभ एक्ज्याक्ट, तर अराउन्ड 50 नै पुग्न आँटिसक्यो, अनि त्यसमा प्राइभेटमा छन्। The lesser number is in private, more is in government only. Government में हमको
40 government and 18 private.
40 or 18.
40 government.
So more in the government side and lesser in the private side.
ज्योति म्याम, मैले चाहिँ एउटा एकदम क्युरियस भएर सोध्न खोजेको, हामीले सबैले पिसिआर मात्रै भन्यो होइन, द बेसिक इज रिएजेन्ट होइन। अब त्यस्तो पेन्डेमिकको बेला हजुरहरूले, किनभने अरूले आफू कहाँ नभइकन त अरूलाई त दिँदैन होइन, त्यस्तो बेला सबैले आफ्नो टेस्ट गरिरहेको थिए भने हाउ डिड यु म्यानेज फर द रिएजेन्ट? पिसिआरको लागि मात्रै कुरा भयो, पिसिआरको लागि चाहिने रिएजेन्ट्सहरू हामीलाई प्राइमर प्रोबहरू चाहियो, पोजिटिभ कन्ट्रोल चाहियो होइन, अब केही न केही त पोजिटिभ त चाहियो। हाउ डिड यु म्यानेज फर द रिएजेन्ट्स?
सुरुमा when we had lesser number of cases, the WHO र CDC बाट आयो किटहरू, तर it was only done at NPHL। जब हामीले अरू ठाउँमा गभर्नमेन्टमा साइटहरू बढाउँदै गयो, त्यसबेला व्यवस्थापनले जिम्मा लियो। हाम्रो डिपार्टमेन्ट अफ हेल्थ सर्भिसेसको म्यानेजमेन्ट डिभिजनको आपूर्ति भन्ने डिभिजन छ, उनीहरूले अब स्पेसिफिकेसन हाम्रोबाटै बनाउन लगाएर, टेक्निकल स्पेसिफिकेसन बनाउन लगाएर कति ठाउँबाट उनीहरूले किन्यो पनि, अनि कतिवटा डोनेसनबाट पनि आयो। अघि डाक्टर देवीले भन्नुभएको थियो के अरे, सिजिनको किट हामीले सुरुमा डोनेसनबाटै पाएको, एन्ड इट वाज वान अफ द बेस्ट that we have used till date। अनि अहिले हामी ट्यागपेथको युज गरिरहेको छौँ, विच इज अल्सो भेरी गुड, अनि त्यसमा पनि 3टै एन्टिजेन युज गरिरहेको छौँ। सो एट प्रेजेन्ट इट्स मोस्टली वी आर बाइङ द किट्स एन्ड बट वी आर अल्सो गेटिङ फ्रम डोनेसन, सो इट्स मिसम्याचै भइरहेछ। अनि जिम्मा डिस्ट्रिब्युट गर्ने किन्ने जिम्मा चाहिँ व्यवस्थापनको भए पनि, बट बिकज वी हेभ मोर इन्टरेक्सन विथ ल्याब, सो दे आर प्रोभाइडिङ अस एन्ड इट इज डिस्ट्रिब्युटेड बाइ अस, सो इट्स लाइक a technical thing।
ज्योति म्याम, अर्को क्वेसन है, यो कोरोनाको पेन्डेमिकले हजुरले भन्नुभयो हामीसँग टोटल 58 ल्याबहरू छ देशभरि, भन्नुको अर्थ यदि अबको 4-5 वर्षमा फेरि यस्तै केही आउँछ जस्तो इन्फ्लुएन्जा भाइरसको आउँछ होइन भने, आर वी रेडी टु ह्यान्डल अल दोज इस्युज, पेन्डेमिक इस्युज बाई आवर सेल्फ होइन? हामीले आउटसोर्सिङ त गर्नु पर्दैन नि हाम्रो टेस्टहरूको लागि। अब भने हाम्रो नेपाल यसले चाहिँ यो कोरोनाले दिएको चाहिँ हामीलाई अवसरमा हामीले राम्रो मोलिकुलर ल्याबहरू इस्टाब्लिस गर्न सक्यौँ र हामीले ट्रेन्ड म्यान पावर पनि बनायौँ। अब आउने हामी कहाँ जति पनि यस्ता अवस्था आउँछ, हामी चाहिँ आफैमा स्वावलम्बन हुन सक्छौँ भन्ने कुराहरू पनि हामीले पायौँ होइन त?
एकदम हजुरले भने जस्तै, जसरी 2009 को पेन्डेमिकले एटलिस्ट एनपिएचएलमा पहिलादेखि इन्फ्रास्ट्रक्चर थियो, हामीले यहाँ केही सुरु गर्नु परेन त्यसले एउटा कुरा सिकाएको थियो तर त्यसबेला चाहिँ हामीले के गरेको थियौँ बिकज कम्पेरिटिभली द नम्बर्स वर लेस we could do it in one place अनि एक अथवा दुई ठाउँमा गरेर सिद्धियो तर अहिले कोरोनाको केसमा बिकज अफ द इन्फेक्टिभिटी यु गेटिङ loads of cases त्यसैले धेरै ठाउँमा बनाइयो in the future what will happen is and at least NPHL is more prepared because we have already planned कि कतिवटा कुरा जुन हामी पहिला रेयर होला भनेर सोच्थ्यौं त्यो NPHL लाई चाहिँ स्टडी बनाएर राख्ने at least we will be at the back कि कुनै नयाँ कुरा आयो भने we will be able to detect जस्तै अरू कन्ट्रीजहरुले चाइनाले सुरुमै उनीहरुसँग प्रविधि नभएको भए त कसरी डिटेक्ट गर्थ्यो सो त्यस्तै हाम्रो NPHL ले पनि स्टडी बनाउने तर जुन बाहिर 58 वटा ल्याब जुन बनिसकेको छ I don't think all of those would be continuously working but at least हरेक प्रोभिन्समा दुई तीनटा ल्याब चाहिँ पछिसम्म पनि रहिरहने भन्ने आशा गरेको छु because at present people have realized that we need human resources in the ल्याबमा यदि अब अहिलेको पेन्डेमिकबाट पनि यदि हाम्रो माथिका निकायहरूले सिकेनन् भने ल्याबमा मोलिकुलर बायोलोजिस्ट माइक्रोबायोलोजिस्टहरू चाहिन्छ भनेर सिकेनन् भने अगेन we will be facing another pandemic in another decade and same हालत हुन्छ तर अहिलेकै हिसाबले जुन इन्फ्रास्ट्रक्चर भइसक्यो तर मान्छे पनि रहिरह्यो भने I think we are better prepared. We have learned a lot but regarding the management part and the contact tracing and some things we are still not very good at but at least the technical part lab को तर्फबाट भन्नु पर्यो भने we are very much prepared and have learned a lot.
त्यसैमा म अलि एड गर्न चाहन्छु हैन ल्याब इन्फ्रास्ट्रक्चर हामीसँग 58 वटा छ हैन कति कन्टिन्यू गर्ला कति कन्टिन्यू नगर्ला हैन तर मान्छेहरु हैन मान्छेमा हामीले इन्भेस्ट गर्छौं भने त्यो मान्छे अब जहाँ जुनसुकै ल्याबमा गए पनि अथवा नयाँ ल्याब खोले पनि त्यो टेक्निकल वर्कफोर्सले चाहिँ त्यो धान्न सक्ने अवस्था हुन्छ तर अहिले च्यालेन्जिङ के छ भने हामी भनौं फेडरल स्ट्रक्चर गयौं तीन तहको गभर्नमेन्ट भइसकेको छ नेपालमा हैन संघीय प्रादेशिक स्थानीय तह कोअर्डिनेसनको पार्ट पनि हुन्छ एउटा हैन जब हामी बिग बल पार्कमा हेर्छौं भने नि त्यो ठुलठुलो कुराहरु छ तर कोअर्डिनेसन मेकानिजम त्यो उलाई चाहिँ कसरी अलिकति सशक्तिकरण गर्ने हैन स्थानीय तहलाई त्यो पार्टमा हामीले पनि फोकस गर्न सक्यो भने इभन कुनै पनि त्यस्तो प्रिपेयर्डनेस अथवा रेस्पोन्सको कुराहरु आउँछ फ्युचर पेन्डेमिक्सहरु त्यसमा चाहिँ हामीले एफिसिएन्टली त नभनौं हैन वी डोन्ट नो what scale and what nature it will come but will be able to handle that manageably हैन त्यो
मेरो एउटा फाइनल क्वेशन है अहिले नेपालमा साथीहरुसित कुरा गर्दाखेरि हैन दे आर भेरी नेगेटिभ के नेपालको टेस्ट के छ र भनेर हाम्रो यो टिम वर्कलाई चाहिँ एकदमै नराम्रो पारेको छ हैन व्हाट आर यु डुइङ कि हामीले नेपालले पनि यस्तो डेभलप गरिरहेछ भनेर हाम्रो जो पब्लिकलाई चाहिँ छ नि कन्फिडेन्समा लिएर आउन हजुरहरुको टिमले चाहिँ के गरिरहनु भएको छ किनभने हाम्रो टेस्टलाई के भन्छ नेपालको टेस्ट त त्यस्तै त हो नि भनेर के त्यस्तै त हो नि भनेर यत्रो जो भित्रपट्टिको पर्दा भित्रको वर्कलाई चाहिँ हाम्रो चाहिँ एकदम नेग्लेक्ट भएको फिल छ नि हजुरहरुले गरेको कामलाई चाहिँ भित्री कामलाई कसरी प्रिपेयर गर्नुभयो अनि जनरल पब्लिकले चाहिँ हाम्रो एनपीएचएल को रिजल्ट चाहिँ रिलायबल छ है भनेर चाहिँ बिलिभ गराउने त्यो कन्फिडेन्समा लिएर आउने काम चाहिँ कस्तो भइरहेको छ किनभने साथीहरुसित कसैसित कुरा गरे भने दे आर नट गुड के रेस्पोन्डिङ राम्रोसँग गर्दैको मैले चाहिँ पाएको छैन
अनि मेरो विचारमा यसमा म सर्ट हामी यो टेक्नोलोजीमा कन्सन्ट्रेट भएको भएर अलिकति डाइभर्ट नहो हैन यसमा चाहिँ सबभन्दा पोजिटिभ कुरा भनेकै एउटा ल्याबबाट अहिले 58 वटा ल्याब भएको छ नेपालभरि दैट्स द पोजिटिभ थिंग्स दैट वी हैव टु टेक हैन म यसमा चाहिँ एक अतिथिसँग कुरा गर्न चाहे जस्तो तपाईले जस्तो यो आरटीपीसीआर त एकदम कम्प्लिकेटेड मल्टिपल स्टेप्स इन्भल्भ हैन अब मल्टिपल भनौं न ट्रेन म्यान पावर तपाई चाहिँ अब आफै ल्याम्प टेक्नोलोजीमा चाहिँ अप्टिमाइजेसन र भ्यालिडेसन गर्नलाई चाहिँ तपाईले स्टडी पनि सुरु गर्नुभएको छ यो ल्याम्प टेक्नोलोजी भनेको के हो र यसले चाहिँ हाम्रो डेभलपिङ कन्ट्रीज नेपाल जस्तो डेभलपिङ कन्ट्रीजमा चाहिँ कसरी हेल्प गर्छ
धन्यवाद रोशनजी अब यस्तो हो कि आरटीपीसीआर एक्सपेन्सिभ सोफिस्टिकेटेड ल्याब चाहिने हैन र त्यसले गर्दाखेरि मान्छे जहिले पनि अल्टरनेटिभ खोज्न थाल्यो हैन अब अल्टरनेटिभ मेथड्सहरु त्यही उसमा चाहिँ ल्याम्प भनेको नयाँ टुल पनि होइन यो कोभिडले गर्दा निस्केको कुरा होइन हैन धेरै वर्ष भइसकेको छ ल्याम्प पनि आएको हैन खास त ल्याम्प भनेको लुप एम्प्लिफिकेसन आइसोथर्मल एम्प्लिफिकेसन एसए हो कि त्यो त्यसमा चाहिँ कस्तो हुन्छ भन्दाखेरि तपाईको अलिकति त्यो सोफिस्टिकेटेड ल्याब नै चाहिँदैन तपाईलाई त्यो भनौँ न एकदमै ट्रेन म्यान पावर पनि चाहिँदैन लो कस्ट हो स्केलेबल पनि छ हैन पोइन्ट अफ केयरमा तपाईँ एप्लाई गर्न सक्नुहुन्छ यसमा चाहिँ हुन्छ के भन्दाखेरि 4 to 6 प्राइमर सेट्सहरू हुन्छ हैन र जस्तै पिसिआर एसेहरूमा तपाईँको एनिलिङ हुने एम्प्लिफिकेसन हुने त्यसपछि रिभर्स ट्रान्सक्रिप्सनले कम्प्लिमेन्ट्री डिएनए बनाउने अथवा डबल स्ट्यान्डर्ड डिएनए बनाउने त्यही नै आखिर एन्ड प्रोडक्ट त्यही आउँछ तर विदिन अ सिंगल ट्युब तपाईँको त्यो प्रोसेसलाई चाहिँ अझै एफिसिएन्टली विदिन लेस टाइम हैन एउटै टेम्परेचरमा त्यही भएर आइसोथर्मल भने हो एउटै टेम्परेचरमा त्यो एकुमुलेट गरिदिन्छ डिएनए प्रोडक्टहरू र त्यो डिएनए प्रोडक्ट जब एकुमुलेट हुन्छ त्यसलाई चाहिँ हामी कुनै कलरिङ डाईहरू युज गरेर अनि हामीले रिड गर्छौँ आफ्नो त्यो पनि चाहिँदैन माइक्रोस्कोप पनि चाहिँदैन त्यसमा चाहिँ हाम्रो आफ्नै आँखाले पनि छुटाउन सक्छौँ यो स्याम्पल पोजिटिभ अथवा यो स्याम्पल नेगेटिभ यो भने अनि फेरि यो कस्तो छ भने तपाईँ एउटा टुल बक्स बनाउनु भयो एउटा बक्समा राख्नुभयो सबै रिएजेन्टहरू तपाईँ जानुभयो तपाईँसँग एउटा कुनै हिट ब्लक छ हैन त्यो अब बजारमा 10-15 हजार रुपियाँ या अथवा त्यो पनि त्यसमा पनि कममै त्यो पाउन सकिन्छ त्यो मात्रै एउटा त्यस्तो इक्विपमेन्ट हुन्छ त्यसमा तपाईँले हिट गर्ने 60 के अरे 60 मिनटको 96 डिग्री सेल्सियससम्म म्याक्सिमम हैन अनि त्यो गर्दाखेरि तपाईँले चाहिँ कलर चेन्ज देख्नुहुन्छ र भनौँ न कस्तो बेला युज गर्ने त हैन अब अभियसली अहिले त डायग्नोस्टिक्समा गएको छैन कस्तो बेला युज गर्ने त त्यसलाई भने अब भनौँ न जहाँनिर तपाईँको एन्टिजेन आरटी ऱ्यापिड स्क्रिनिङ टेस्ट पनि आएको अवस्था छैन र आयो भए पनि त्यसको चाहिँ सेन्सिटिभिटी रेस्पोसिभिटी त्यत्रो राम्रो भएको अवस्था नभएको कारणले गर्दाखेरि यसलाई चाहिँ जस्तै यो पोइन्ट अफ इन्ट्री अथवा ल्यान्ड क्रसिङहरूमा युज गर्न सकियो होइन तपाईँको यो के अरे हस्पिटल सेटिङ्सहरूमा होइन पोइन्ट अफ केयरहरूमा युज गर्न सकियो र अरू कुरा चाहिँ यसमा हामीले अर्को के पनि हेरिरहेको छौँ भने सलाइभा युज गरेर होइन अब मैले आफैले एक्सपिरियन्स गर्दाखेरि त्यो स्वाबभित्र जाँदाखेरि आफूलाई एकदमै अनइजी लाग्ने अथवा नाकमा जाँदाखेरि पनि एकदमै अनइजी लाग्ने कतिजनाको त नाकबाट त्यो स्वाब निकाल्दाखेरि पनि अड्केको होइन त्यो धेरै डिस्कम्फोर्टहरू भएको कारणले गर्दाखेरि सलाइभा होइन अब युज गरेर कुनै राम्रो कम्पेरिटिभ मेथड अथवा कम्प्लिमेन्ट्री मेथडहरू बनाउन सकिन्छ भनेर खास यो चाहिँ नास्टले ग्रान्ट दिएको थियो यो उसको लागि काम गर्नलाई र त्यसमा चाहिँ हामी काम गरिरहेका छौँ अहिले
नाइस थ्याङ्क यू
अझै चाहिँ यो ल्याब बेस्ड टेस्टिङ अफ कोभिड चाहिँ इन्ट्रेस्टिङ लाग्यो तर हाम्रो नयाँ श्रोताहरूको लागि यो के हो त ल्याब बेस्ड टेस्टिङ भनेको कसरी काम गर्छ यसले यसको बेसिक्स बारे अलिकति भनिदिनु न
ए हुन्छ अब यसमा अब यो अलिकति जार्गन युज नै हुन्छ यो यसमा ट्याक्कै नेपाली त मलाई भन्न आउँदैन होइन तर के छ भने यसमा 4 देखि 6 वटा प्राइमर सेट्सहरू हुन्छ होइन जस्तै तपाईँले आरटिपिसिआर कसै तपाईँ युज गर्नुभएको छ आरटिपिसिआर गर्नुभएको छ भने प्राइमर सेट्स युज गर्नुहुन्छ त्यो भनेको प्राइमर भनेको के भन्दाखेरि अघि ज्योति म्यामले भनिदिनु म ज्योति हो भने चाहिँ मलाई कसरी थाहा हुन्छ भने स्पेसिफिक मतिरको सिक्वेन्स हुनुपर्छ भनेर होइन त्यही भएर त्यो 4 देखि 6 वटा सिक्वेन्स जुन चाहिँ स्पेसिफिकली त्यो भाइरसको कम्प्लिमेन्ट्री हुनु पर्यो र त्यही अनुसारले तपाईँ प्राइमर डिजाइन गर्नुहुन्छ र प्राइमर डिजाइन गरिसकेपछि त्यही अब आफैले पनि बनाउने अथवा बाहिरबाट ल्याउने र त्यो ल्याम्प एसेमा 4 देखि 6 वटा प्राइमर मध्येको एउटा प्राइमर भनेको इनर प्राइमर होइन त्यो इनर प्राइमरले के गर्छ भने डिएनए पोलिमरेजसँग बाइन्ड गरेर त्यो उसमा चाहिँ हाम्रो आरएनए सिक्वेन्समा अनि एक्सटेन्ड गर्छ होइन त्यो एक्सटेन्ड जब सिक्वेन्स एक्सटेन्ड हुन थाल्छ नि त्यतिखेर अरू प्राइमरहरू जुन 6 वटा प्राइमर मैले भने अथवा 4 वटा प्राइमर अरू प्राइमर आएर त्यहाँ लुप हाइब्रिडाइजेसन त्यो लुप बनाउँछ एउटा हाइब्रिडाइजेसन लुप बनाइदिन्छ हो त्यो लुप पछि एक्सटेन्ड अनिलिङ हुँदै जाँदाखेरि तपाईँको डबल स्ट्यान्डर्ड डिएनए बन्न जान्छ र त्यो एउटा ट्युबमा जब डबल स्ट्यान्डर्ड डिएनए एकुमुलेट हुँदै जान्छ र हाम्रो त्यही ट्युबमै पनि हाम्रो हुन्छ डाईहरू हुन्छ होइन जस्तै हाइड्रोक्सी न्याफ्थोल ब्लू डाई युज गर्छ कसैले अरू डाई युज गर्छ त्यो भनेको कलरिङ एजेन्ट हो त्यो कलरिङ एजेन्टले जब तपाईँको पिएच त्यसमा इन्क्रिज हुन थाल्छ होइन अनि त्यो चेन्ज हुन थाल्छ त्यसको कलर भनेपछि त्यो कलर चेन्ज देखिने बित्तिकै यो चाहिँ पोजिटिभ अथवा यो चाहिँ नेगेटिभ स्पेसिमेन थियो भनेर चाहिँ हामीले त्यो पत्ता लगाउन सक्छ अभियसली त्यहाँनिर कन्ट्रोल चाहिन्छ होइन पोजिटिभ कन्ट्रोल नेगेटिभ कन्ट्रोल त्यो हुँदाखेरि तपाईँलाई चाहिँ त्यो क्लियर डिरेक्सन दिन्छ आफ्नै आँखाको अगाडि विदिन सर्ट
भनेपछि यो बेसिकली आरटिपिसिआर नै भयो
एक किसिमको आर्टिफिसियल भयो तर सबै कुरा एकैचोटि हालिदिने खिचडी बनाइदिने हजुर
ओके
थ्याङ्क यू डक्टर कर्ण यसपछि म डक्टर सुनितासँग अलिकति उहाँको पनि इन्ट्रेस्ट र अहिले लास्ट विक हामीले चाहिँ क्रिस्परमा हाम्रो नोबेल प्राइज पनि पाउनुभयो होइन क्रिस्पर डिस्कभर गर्ने साइन्टिस्टहरूले यो अहिले क्रिस्पर टेक्नोलोजी युज गरेर पनि कोभिड-19 को चाहिँ EUA एप्रुभल आएको छ क्यारे कुनै कुनै टेस्टहरू यो चाहिँ कतिको फिजिबल छ यसले चाहिँ आरटिपिसिआरलाई एज अ कम्प्लिमेन्ट्री टेस्ट पनि गर्न मिल्छ भन्ने कुरो भएको छ यसको बारेमा अलिकति बताइदिनुहुन्छ यो टेक्नोलोजी कस्तो हो कसरी युज गरिन्छ भनेर
हजुर अब यो क्रिस्पर भन्दाखेरि चाहिँ फर्स्टमा म अलिकति यसको ब्याकग्राउन्ड भन्न चाहन्छु किनभने धेरैलाई थाहा नहुन पनि सक्छ अब क्रिस्पर भनेको क्लस्टर्ड रेगुलरली इन्टरस्पेस्ड सर्ट प्यालिन्ड्रोमिक सिक्वेन्स भन्छ त्यो ब्याक्टेरियाको जेनोममा हुने सर्ट प्यालिन्ड्रोमिक सिक्वेन्सहरू जसले चाहिँ ब्याक्टेरियाको एडप्टिभ इम्युन सिस्टम हो ब्याक्टेरियाले भाइरस अथवा प्याथोजेनबाट आफूलाई प्रोटेक्ट गर्नको लागि भएको त्यो सिस्टम अनि पछि चाहिँ जेनिफर डोर्ना र एमएनयू उहाँहरूले त्यो जुन ब्याक्टेरियाको इम्युन सिस्टमलाई पत्ता लगाएर क्रिस्पर क्यास भनेको क्रिस्पर एसोसिएटेड सिस्टम इन्डोन्युक्लियस भयो जुन चाहिँ आरएनए डिपेन्डेन्ट पनि हुनसक्छ डिएनए डिपेन्डेन्ट पनि हुनसक्छ अनि त्यस्तो जुन के भन्छ एकदम ग्राउन्ड ब्रेकिङ वर्क ब्याक्टेरियाको इम्युन सिस्टमलाई उहाँहरूले चाहिँ जेनोम एडिटिङको लागि पत्ता लगाउनु भयो अनि त्यसपछि जस्तै 2012 को अराउन्ड सेप्टेम्बरमा साइन्समा उहाँहरूको फर्स्ट पेपर पब्लिस भइसकेपछि त्यसको अराउन्ड 6 मन्थ्स पछि फेरि अब उहाँहरू पछि अब फेङजाङ एमआइटी को उहाँहरूले फेरि अर्को पेपर कसरी भने त्यही क्रिस्पर क्यासलाई युज गरेर म्यामेलियन सेलमा आफूले चाहेको जिनलाई भनेको ठाउँमा कट गर्ने कपी पेस्ट गर्ने त्यस्तो खालको इम्प्लिमेन्ट गर्नुभयो अनि त्यसपछि अल अराउन्ड द वर्ल्ड विदिन अ सर्ट टाइम क्रिस्परलाई चाहिँ एकदमै अहिलेको अब रुटिन पिसिआर जस्तै अब एकदमै युज गर्न थालियो र 2017-18 मा यो सेरलक भन्ने टेक्नोलोजी चाहिँ फेङजाङको ल्याबबाट उहाँहरूले बनाउनु भयो त्यो सेरलक भनेको चाहिँ कस्तो भने स्पेसिफिक हाई सेन्सिटिभिटी एन्जाइमेटिक रिपोर्टर अनलकिङ भन्ने टेक्निक उहाँहरूले क्रिस्पर क्यास 9 युज गरेर बनाउनु भयो त्यसको चाहिँ उहाँहरूले डेमोन्स्ट्रेसन मेनली जिका भाइरस डेङ्गु भाइरसहरूको आरएनएलाई डिटेक्ट गर्न सक्छ कि सक्दैन भनेर बनाउनु भयो अनि त्यो टेक्निकलाई युज गरेर अहिले अब यो कोभिड पेन्डेमिकको बेलामा उहाँहरूले फेरि भए हामीले यो अब कोभिडमा पनि इम्प्लिमेन्ट गर्न सक्छौँ कि सक्दैनौँ भनेर लागि पर्नुभयो र कोभिडको केसमा पनि उहाँहरूले सेरलक अनि त्यसपछि जुन सेरलक टेक्निकलाई युज गरेर उहाँहरूले कम्पनी बनाउनु भयो त्यही कम्पनी मार्फत उहाँहरूले किट बनाएर अहिले अब एफडिएबाट युए पाएको छ र यो किटले कसरी काम गर्छ भन्दाखेरि क्रिस्पर सार्स-कोभ-2 किट भनेर भनेको छ त्यसमा चाहिँ ल्याम्प मेथड जुन अघि डाक्टर रञ्जित जीले भन्नुभयो ल्याम्पलाई युज गर्छ अब ल्याम्पको डिटेलमा उहाँले भन्नुभयो म त्यहाँ डिटेलमा जानु परेन अनि ल्याम्प प्लस क्रिस्पर युज गर्छ र यसको फाइदा चाहिँ कस्तो भने अब ल्याम्पले आइसोथर्मल एम्प्लिफिकेसनबाट जस्तै पिसिआरमा तपाईँको टेम्परेचर डिफ्रेन्ट टेम्परेचर ग्रेडियन्टमा एम्प्लिफिकेसन गर्छ भने ल्याबमा चाहिँ आइसोथर्मल कन्स्टेन्ट हिटमा तपाईँको डिफ्रेन्ट सेट अफ प्राइमरले तपाईँको जेनेटिक मटेरियललाई एम्प्लिफाई गरेर अनि एज अ रिपोर्टरले डिटेक्ट गर्ने कलर चेन्ज भएर त्यो केसमा अब कहिलेकाहीँ फल्स पोजिटिभ रिजल्ट अथवा फल्स नेगेटिभ रिजल्ट हुने सम्भावना पनि हुन्छ अनि त्यसमा क्यास 9 वा क्यास 13 एन्जाइम राखिन्छ त्यो चाहिँ कस्तो भने जस्तै सार्स-कोभ-2 को केसमा जेनेटिक मटेरियल आरएनए भएको भएर हामीले चाहिँ आरएनएलाई डिटेक्ट गर्ने आरएनए को जेनोमिक डिएनएलाई डाइजेस्ट गर्ने क्यास एन्जाइम चाहियो त्यो भनेको चाहिँ क्यास 13A वा क्यास 12A भनिन्छ त्यो उहाँहरूले प्रयोग गर्नुभयो र त्यसले कसरी त्यो आरएनए एन्टिजेनलाई डिटेक्ट गर्छ SARS-CoV-2 को केसमा भन्दाखेरि उहाँहरूको त्यो रियाक्सन मिक्सचरमा गाइड आरएनए भनेर हुन्छ। त्यो गाइड आरएनए भनेको चाहिँ कस्तो हुन्छ भने जस्तै तपाईँलाई उदाहरण दिँदा अमेरिकाको एउटा भनौँ न टुरिस्ट नेपालमा आयो, उहाँलाई पशुपतिनाथ मन्दिरमा जान हेर्न मन लागेको छ भने कि त तपाईँले एउटा गाइड लगिदिनु पर्यो, जबकि गाइडले यसरी यसरी तपाईँ पशुपति टेम्पलमा पुग्नुहुन्छ भने सँगै लग्नुहुन्छ अनि पशुपतिमा जानुहुन्छ। अर्को कुरा नेभिगेसन, तपाईँले गिभन एड्रेस राख्नुभयो भने त्यो एड्रेसले ठ्याक्क तपाईँको जस्तै स्ट्रिट होम नम्बर सबै भयो भने पुर्याइदिन्छ। त्यस्तै जस्तो यो गाइड आरएनए भनेको 20 बेस पेयर अफ सिक्वेन्स हुन्छ त्यो स्पेसिफिक SARS-CoV-2 वा जिका वा डेङ्गु, अनि त्यो राखिसकेपछि त्यसले क्यास एन्जाइमलाई गाइड गरेर जुन स्पेसिफिक ठाउँमा त्यो 20 बेस पेयर हुन्छ त्यही ठाउँमा Cas9लाई लगेर छोडिदिन्छ। अनि त्यसपछि Cas9 अथवा Cas9 को केसमा डिएनएलाई डाइजेस्ट गर्छ। Cas13 जुन अहिले कोभिडको केस छ त्यसमा चाहिँ SARS-CoV-2 को जेनोमलाई कट गर्छ। अनि कट गरिसकेपछि त्यो एन्जाइम त्यहाँ रिपोर्टर हुन्छ, अनि त्यो रिपोर्टर Cas13 सँग अट्याच त्यो डिटेल्स हुन्छ नि, डिटेल्स भइसकेपछि त्यसले कलर एमिट गर्छ। र त्यो कलर एमिट गरेको डिटेक्सन चाहिँ तपाईँले इदर तपाईँको प्लेट रिडर जस्तै एलाइजा प्लेट रिडरहरूबाट युज गर्न सक्नुहुन्छ, वा लेटरल डिपी स्टिकहरू पनि अहिले अभाइलेबल छ, त्यसरी युज गर्न सक्नुहुन्छ। बेसिकली यसरी एकदमै हाइली सेन्सिटिभ र उहाँहरूले क्लेम गरे अनुसार सेरलकले त 1 कपी अफ भाइरसलाई डिटेक्ट गर्ने भनेर छ। र यो अहिलेको कोभिड-19 को टेस्ट किटमा चाहिँ भाइरल लोड उहाँहरूले अराउन्ड 3 म करेक्ट भएसम्म मलाई थाहा भएसम्म भनेको मोर स्पेसिफिक इभन मोर सेन्सिटिभ टु आरटिपिसिआर, अझ भन्दाखेरि चाहिँ फेम्टोमोलरको रेन्जमा यसले डिटेक्ट गर्न सक्छ भन्ने छ। अनि अब यसको टेस्ट अमेरिकामा कस्तो छ नि, नयाँ टेक्निक भयो, उहाँहरूले स्टिल यसलाई चाहिँ जस्तै त्यो सेरलक कम्पनीलाई फलो गर्दाखेरि पोइन्ट अफ केयरको रूपमा अहिले चाहिँ यसले टोटल 60 मिनट्समा रिजल्ट दिन्छ। यदि तपाईँले उहाँहरूले चाहिँ यसलाई अब विदिन 15 to 20 मिनट्समा ल्याउने तरिकाले ट्राई गरिराख्नु भएको छ। र अब नेपालको केसमा यसलाई ल्याउन सक्यो भने चाहिँ आरटिपिसिआर जुन अब अहिले आरटिपिसिआर को भन्दा एकदम सर्ट टाइममा, जस्तै यता 6 घन्टा लाग्छ भने क्रिस्पर को टेक्निक युज गरेर 1 आवरमा चाहिँ तपाईँको रिपोर्ट आउँछ। त्यो टर्नओभर टाइम चाहिँ एकदमै धेरै हुन्छ। अनि अब फिजिबिलिटीको कुरा गर्दाखेरि तपाईँलाई बायोसेफ्टी लेभल 2 सुरुमा जस्तै ल्याम्पकोमा जस्तो हो त्यस्तै हो। अनि त्यो बाहेक अरू सफिस्टिकेटेड टेक्निकहरू जस्तै अहिले यो युए पाएको केसमा चाहिँ तपाईँलाई प्लेट रिडर चाहिन्छ, त्यो हुँदा अलिकति तपाईँले एक्स्ट्रा पैसा तिरेर किन्नु पर्यो। तर डिप स्टिक जुन उहाँहरूले चाहिँ डिप पनि यो एफडीए युए पाइसकेपछि अहिले बनाइसक्नु भएको छ, रिजल्ट पनि पब्लिस भइसकेको छ। तर युए अप्रुभल नभइसकेको अवस्थामा चाहिँ यसमा डिप स्टिक मेथडबाट गर्दाखेरि तपाईँलाई एकदमै सस्तो पर्ने हुन्छ। त्यही भएर यो डिप स्टिक मेथड यदि नेपालमा भनौँ न इन फ्युचरमा नेपालमा यस्तो नयाँ खालको डायग्नोस्टिक टुलहरू सधैँ आरटिपिसिआरमा मात्रै भर परेर भन्दा पनि अब अहिलेको नयाँ इनोभेटिभ टेक्निकहरू पनि इम्प्लिमेन्ट गर्ने साइड बाई साइड एज अ कम्प्लिमेन्ट वा एज अ सब्स्टिच्युट, जस्तो लेस रिसोर्स भएको ठाउँहरूमा जहाँ आरटिपिसिआर सम्भव छैन त्यस्तो ठाउँमा ट्राई गर्ने हो भने वान अफ द अल्टरनेटिभ मेथड हुन सक्छ। र हामीले डिफ्रेन्ट जस्तै ल्याम्पको लागि चाहिने प्राइमरहरू अलरेडी पब्लिस छ, किटहरू अलरेडी अभाइलेबल छ। Cas13 एन्जाइम तपाईँले एक्जोजिनस एक्सप्रेसन गरेर आफैले ल्याम्पमा पनि प्युरिफाई गर्न सक्नुहुन्छ। पार्ट छुट्टै छुट्टै ठाउँहरूबाट किनेर आफूले होल किट सिस्टम आफैले बनाएर पनि गर्न सकिने अवस्थामा छ यो।
हस्, थ्याङ्क यू डाक्टर गौतम। कसैको केही क्वेसन थियो अहिले? थिएन है? ओके, अब हामीसँग चाहिँ यो प्यानल डिस्कसनलाई 1 आवर एलोकेट थियो। अब अफकोर्स अहिले नयाँ डेभलपमेन्ट नयाँ टेक्नोलोजी गर्दाखेरि धेरै नै एसेजहरू डेभलप भइरहेको छ। अब सबै हामीले कुरा गरेर साध्य पनि हुँदैन है। अब एटलिस्ट हामीले चाहिँ ल्याम्पबारे अलिकति बुझ्यौँ, सर्लोकबारे अलिकति बुझ्यौँ, ज्योति म्यामले चाहिँ आरटिपिसिआरको डिटेल बताइदिनु भयो, नेपालमा के भइरहेछ भन्ने कुरो पनि हामीलाई चाहिँ बताइदिनु भयो। त्यसको लागि फेरि एकचोटि सबैलाई धन्यवाद। र डाक्टर अजित, डाक्टर सुनिता नेपालमै हुनुहुन्छ। आई थिङ्क इफ दे मस्ट बी, आइम स्योर दे आर ह्याप्पी टु हेल्प द गभर्मेन्ट इफ निडेड for anyways, I know, with the optimization validation of newer technologies. hoping that we would like to conclude this panel discussion section here. And once again, thank you very much, Dr. Ajit, Dr. Sunita, Dr. Jyoti Acharya, and Dr. Indira Tiwari. Thank you for participation. र म अब यो सेसन चाहिँ डक्टर देवी बस्नेतलाई ह्यान्ड ओभर गर्न चाहन्छु टु कन्क्लुड दिस सेसन. थ्याङ्क यू भेरी मच.
Thank you. Namaste. All ठीक छ?
हजुर.
Thank you very much. Actually, हामी सँग हाम्रो एकजना NRN को रिप्रेजेन्टेटिभ आउने कुरा थियो. चुटी हेर्नु चाहिँ हुन्छ कोही? मैले देखिनँ यहाँनिर. देखिनँ. ओके, नो प्रब्लम. अहिले अर्को प्रोग्राम पनि सुरु भइरहेको छ. अहिले यो NRN डे आज NRN डे भएको भएर अर्को प्रोग्राम भयो. सबै साथीहरू त्यता जानु भएको छ हैन. म अहिले यो सेसनलाई कन्क्लुड गर्नु पर्दाखेरि चाहिँ सर्वप्रथम त तपाईहरु सबैजनालाई जस्तो आफ्नो टाइम दिएर यहाँ आइदिनु भयो हैन. अनि विशेष गरिकन हामीले जुन कुरा चाहेका थियौं यो एडभान्स टेक्नोलोजी नेपालमा मतलब वर्ल्डमा एडभान्स टेक्नोलोजी विशेष गरिकन चाहिँ हेल्थ र डिजिज क्युरको लागि कस्तो खालको भइरहेको छ र त्यसको चाहिँ इम्पोर्टेन्स के हो पर्टिकुलरली त्यसको बारेमा हामी छलफल गर्न चाहेका थियौं. मेरो विचारमा हामीले धेरै कुराहरुमा टच गर्यौं. विशेष गरिकन हामीले यो क्यान्सरको बारेमा हैन 3D इमेजिङ देखि लिएर हाम्रो चाहिँ डिटेक्सन सिस्टम देखि लिएर अनि जस्तो यो रोबोटिक्सहरुको बारेमा हाम्रो न्यानो टेक्नोलोजीको बारेमा हामी छलफल गर्यौं. यो चाहिँ डेभलपिङ हुँदै जाने भएको भएर यी इन्फर्मेसनको सँगसँगै हामीले नेपालमा विशेष गरिकन यो आजको हाम्रो कन्क्लुजन के गर्नुपर्ने हुन्छ भने हामी डायस्पोरामा नेपाली डायस्पोरामा इभन एडभान्स टेक्नोलोजी लर्न गरेर एचिभ गरेर बसिरहेको हाम्रो डायस्पोराको ह्युमन रिसोर्सेस छ हैन. एउटा पोइन्ट त्यो हामीले चाहिँ पाएका छौं. त्यसमा पनि अहिले तपाईको एकदमै रिनाउन्ड युनिभर्सिटीमा बसेर एकदमै राम्रोसँग रिसर्च गरेर बसिरहनु भएको छ र कति धेरै जना साथीहरु कमर्सियलाइजतिर जानु भएको छ. जस्तो अहिले तपाईहरु यही एक्जाम्पल नै हुनुहुन्छ. कति नेपालमा फर्किएर के गर्न थाल्नु भएको छ. वास्तवमा यो राष्ट्रको क्षेत्रीय अनि यो ग्लोबलले चाहिँ हामी धेरै जना प्रोफेसनलहरुलाई र जो चाहिँ प्रोफेसनल हुन खोजिरहनु भएको छ र भइसक्नु भएको छ उहाँहरुलाई चाहिँ किसिमको हौसला अनि वर्किङ टुगेदरको एउटा इन्भाइरोन्मेन्ट क्रिएट गरेको छ भन्ने मैले महसुस गरेको छु. हाम्रो यो सेसन मात्र हैन, मैले हिजोदेखि अलमोस्ट सबै सेसनहरु फलो गरेको छु र तपाईहरुले पनि हेर्न सक्नुहुन्छ. हाम्रो सबै रेकर्डेडहरु छ हैन. हामी कुनै पनि टाइममा फ्री टाइममा हेर्न सकिन्छ र यसले चाहिँ अझ अर्को इम्पोर्टेन्ट कुरा के भने जस्तो यहाँ सुनिता म्याम हुनुहुन्छ, अस्ति भर्खर नेपाल जानुभएको छ, त्यहाँको इन्भाइरोन्मेन्ट बुझ्नु भएको छ हैन. त्यो यो किस्पर के भन्छ किस्पर 690 भन्छ है 19 त्यो किस्पर त्यो टेक्नोलोजी चाहिँ
अथवा क्रिस्पर टेक्नोलोजी
त्यो टेक्नोलोजी चाहिँ रिसेन्ट टेक्नोलोजी हो. हाम्रो मेरो आफ्नो कम्पनीमा पनि हामीले रिसेन्टली यो सेल इन्जिनियरिङ विशेषमा मैले अहिले गरिरहेको काम चाहिँ एन्टिबडी इन्जिनियरिङबाट अलिकति लेट स्टेजमा त्यो सेल इन्जिनियरिङ गर्दैछौँ. हामी त्यही टेक्नोलोजी युज गर्दैछौँ भर्खर नै हैन. नयाँ टेक्नोलोजी त्यो चाहिँ अलिक प्याटेन्टेड भएको भएर त्यस्तै खालको अरू केही छ कि भनेर हामी खोज्दैछौँ. आई मिन हाम्रो यो खालको रिसर्चको जुन नेटवर्किङ छ त्यो चाहिँ कन्टिन्यू हामीले गरिरहनुपर्छ हैन. त्यसले गर्दाखेरि हामीले हाम्रो देशमा बसेर वा देश बाहिर बसेर अरूसँग कोअर्डिनेसन गरेर सिक्नुभन्दा पनि हामी आफ्नै नेपाली कम्युनिटी जुन छ अहिले हामीसँग जुन जम्मा भएका छौँ यो बिचमा हामीले डिस्कस गऱ्यौँ भने अझ फ्रुटफुल हैन अनि इजी टु कम्युनिकेट इजी टु एक्सपान्ड हुन्छ जस्तो मलाई लाग्छ. अहिले म चाहिँ खास यो विषयलाई चाहिँ कन्क्लुड गर्दाखेरि यो हाम्रो एउटा ग्रुप बनेको छ. तपाईहरुले यो जति पनि प्रेजेन्टेसनहरु गर्नुभएको छ सबै हाम्रो आर्किभमा बस्छ. अनि यो चाहिँ अलमोस्ट 300 पेजको तपाईहरुले पाउनु भएको छैन 300 पेजको हाम्रो एउटा डकुमेन्ट तयार गरेका छौं हामी. जति पनि अहिले प्रोफेसनल साथीहरु हुनुहुन्छ उहाँहरुको सबैको पेपरहरु एब्स्ट्र्याक्टहरु अनि इभन उहाँको कन्ट्याक्टहरु देखिएर सबै हामीले डकुमेन्ट गरेका छौं. त्यो वेबसाइटमा पाउन सक्नुहुन्छ. विशेष गरी यो हाम्रो सेसनमा चाहिँ हामीले जुन अर्न गरेको टेक्नोलोजी छ नेपालमा हामीले के गर्नुपर्ने छ भने अघि हाम्रो के अरे उहाँ साथीले भन्नुभएको थियो नेपालमा चाहिँ डिजिटलाइजेसन भएको छैन। खासमा हामीलाई थाहा छ बाहिर हामी गर्न सक्छौँ, तर नेपालमा चाहिँ त्यो लेभलमा जुन लेभलमा चाहिँ बुझ्नुपर्ने यो डिजिटलाइजेसन सिग्निफिकेन्ट के हो भन्ने कुरा बुझेर अगाडि इम्प्लिमेन्ट गर्नुपर्ने हो, त्यसको बारेमा त्यति डिस्कस भएको मैले पाएको छैन। सायद अब अहिले हुन्छ होला, हाम्रो धेरै डिस्कसनहरूबाट सायद केही आइडिया आउला होइन। त्यो म्याडमहरू पनि हुनुहुन्छ, उहाँले पनि यो कुराहरू चाहिँ सन्देश लिइरहनुहुन्छ होला। यी म्याक्सिमम् हामीले डिस्कसन गर्नुपर्छ हाम्रो यी साइन्टिफिक रिसर्च, साइन्टिफिक आउटपुटहरूको बारेमा कुन हामीले इम्प्लिमेन्ट गर्दाखेरि चाहिँ राम्रो हुन्छ, कुन नराम्रो हुन्छ, यसको साइड इफेक्टहरू के हो भन्ने कुरा चाहिँ तपाईँ हामी जस्तो प्रोफेसनल मान्छेहरूबाट डिस्कसन आएको आउटपुटलाई लिएर गभर्मेन्टमा इम्प्लिमेन्ट गर्नुपर्छ जस्तो मलाई लाग्छ। र यही विषयहरूमा हामीले छलफल गऱ्यौँ आज, जस्तो विशेष गरिकन कोभिडको कुराहरू गऱ्यौँ, डायग्नोस्टिकको कुराहरू गऱ्यौँ। यसमा अब मलाई डिटेल म अहिले कमेन्ट गर्न त मैले चाहिनँ। अघि पनि मैले भनेको थिएँ मलाई त्यति धेरै त थाहा थिएन, तर यो हामीले यहाँ कोरियाबाट जुन किट पठाउनको लागि कोअर्डिनेट गरेको थियौँ त्यो मैले नै गरेको थिएँ। त्यति बेला मैले धेरैजना नेपालमा हाम्रो साथीहरूसँग कुरा पनि भयो, अफिसरसँग कुरा भयो, उषा म्यामसँग पनि मैले पर्सनली कुरा गरेको थिएँ। त्यहाँको त्यति बेला त अलिकति हुनै थियो, सुरु भएको भएर। अब अहिले धेरै भइसकेको छ मेरो विचारमा, राम्रो गर्नुभएको छ। तर के हो भने यी यस्ता विषयहरू डिस्कस गर्दाखेरि, डिसिजन गर्दाखेरि, साइन्टिफिक कुराको डिसिजन गर्दाखेरि एटलिस्ट साइन्टिस्टहरूसँग बसेर त्यसको इम्प्याक्ट के हुनसक्छ, कुन राम्रो कुन नराम्रो छ भनेर डिस्कस गर्ने खालको इन्भाइरोमेन्ट चाहिँ नेपालमा हामीले बनाउनुपर्छ। त्यो कुरा चाहिँ हामी सबैले महसुस गरेको हुनुपर्छ जस्तो लाग्छ मलाई। सायद त्यो चाहिँ यदि भएन भने पनि हामीले त्यो चाहिँ गराउनुपर्छ। मैले नेपालको केही मेरो साथीहरूसँग विभिन्न ठाउँहरूको प्रोफेसनल साथीहरूसँग कुरा गरेको थिएँ। यो इन्भाइरोमेन्ट बनाउन म अलिकति आउट अफ अर्डर गएको छु। यो मतलब मैले कन्क्लुजन गरिसकेँ भन्नुको खास चाहिँ यो मैले के भन्न खोजिरहेको छु भनेदेखि हामीले यो विभिन्न खालको सेमिनार अनि यो कन्भेन्सन अनि त्यस्तो यत्रो भेला गरेर टाइम लिएर किन गरेको भने यो एनआरएनको क्याम्पेन पनि हो। पहिलो कुरो हामी बाहिर बसेको साथीहरूको जुन यो साइन्टिफिक फोर्स छ, पोटेन्सियालिटी छ, त्यो पोटेन्सियललाई चाहिँ नेपालमा भएको साथीहरूसँग कोअर्डिनेट गर्नु पर्यो, नम्बर 1। नम्बर 2 नेपालमै भएको हाम्रो प्रोफेसनल साथीहरूले पनि नेपाल गभर्मेन्टलाई के खालको एउटा पुस गर्नु पर्यो भनेदेखि तपाईँहरूले गरेको डिसिजनमा हाम्रो साइन्टिफिक नलेज पनि चाहिन्छ। र अहिलेसम्म मेरो विचारमा अहिलेसम्म त्यस्तो धेरै अन्डरस्ट्यान्डिङ भएको छैन, नेपाल गभर्मेन्टले बुझेको छैन। तर त्यो काम हामी गर्न सक्छौँ। कसरी गर्छौँ भने मैले एउटा सल्लाह गरेको थिएँ, प्रत्येक युनिभर्सिटीमा प्रत्येक प्रोभिन्समा तपाईँ हामीले चाहिँ साइन्स एन्ड रिसर्च एन्ड डेभलपमेन्टको बारेमा चाहिँ छलफल गराउनु पर्छ, चलाउनु पर्यो। सबै ठाउँमा प्रोभिन्समा चलाउनु पर्यो, बुझाउनु पर्यो होइन। यो यसको भ्यालु यस्तो भनेर बुझाइसकेपछि पक्कै पनि धेरै टाइम नलाग्छ, छिटै नै हामी यो काममा चाहिँ हामी गर्न सक्छौँ। यो हाम्रो बाहिरबाट पनि एनआरएनको साथीहरूले पनि पुस गरिरहनु भएको छ, कनेक्सन भइरहेको छ। त्यसैले यसलाई हामी अगाडि बढाउँ। र फाइनल्ली चाहिँ मैले यो हाम्रो एनआरएनको साथीहरू यहाँ आउनुहुन्छ भनेको थिएँ, उहाँहरूले मलाई अहिले भर्खर मेसेज पठाउनु भयो, तपाईँले नै साथीहरूलाई चाहिँ एनआरएनको तर्फबाट पनि गर्नु होला भन्नुभएको छ। खास यो अर्थको चाहिँ म एनआरएनको यहाँ कोरियाको आइसिसी प्रतिनिधि पनि हो। त्यसैले म विशेष गरिकन यो साइन्टिफिक कम्युनिटीमा एज अ एनआरएन क्याम्पेनर नै छु भन्दा पनि हुन्छ। त्यसैले तपाईँहरूसँग तपाईँहरूको स्पेसल टाइमहरू मैले लिएँ। मलाई लाग्छ हाम्रो डिस्कसनबाट धेरै साथीहरूले चाहिँ यो के नलेजहरू पाउनुभयो। विशेष गरी मैले धेरै कुरा सिकेँ होइन। मैले गरेको कुराहरू तपाईँहरूबाट अलिकति तपाईँको आफ्नो एक्सपर्टिजहरू सेयर गर्नुभयो, धेरै साथीहरूले सिक्नुभयो। अब फाइनल्ली म चाहिँ सबैजना हाम्रो स्पिकरहरूलाई धेरै धेरै धन्यवाद दिन चाहन्छु। विशेष गरी हाम्रो डाक्टर वाङ, उहाँले चाहिँ राति कति राति भए पनि उहाँहरू आएर हाम्रो चाहिँ प्रोग्राममा आफ्नो कुराहरू सेयर गर्नुभयो। वास्तवमा द्याट्स अ भेरी इन्ट्रेस्टिङ अनि एकदम इन्फ्लुएन्सिङ टेक्नोलोजी हो, त्यो नेपालमा पनि चाहिन्छ। अघि हामी कुरा गरेका थियौँ त्यस्तै नेपालमै काम गरिरहनु भएको हाम्रो साथीहरू हुनुहुन्छ। जस्तो मैले नामै लिनु पर्दाखेरि चाहिँ विशेषजी भयो होइन, डाक्टर असिमजी, उहाँहरू दे आर फ्यान्टास्टिक। आई थिङ्क मलाई थाहा थिएन त्यो कुरा त्यस्तो राम्रो काम गर्दै हुँदो रहेछ भन्ने कुरो। त्यो एकदमै वन्डरफुल काम हो। उहाँहरूले यहाँ केही कुराहरू राख्नुभएको छ, जस्तो यो इम्प्लिमेन्टेसनको पार्टमा, फाइनेन्सियलको पार्टहरूमा चाहिँ अलिकति अझै पनि के अरे ल्याक छ भन्नुभएको छ। अनि यी कुराहरू चाहिँ हामीले एनआरएनको नेटवर्कमा हामीले डिस्कसन गऱ्यौँ भने सायद हामी काम रिसर्च एक्सचेन्ज मात्रै होइन, हामी फाइनेन्सियल सपोर्टिङ पनि भएर काम गर्न सक्छौँ जस्तो मलाई लाग्छ। अस्ति हाम्रो यो रिजनल कन्फरेन्समा हामीले एउटा सानो सेसन राखेको थियौँ, सायद सुन्ता म्याम हुनुहुन्थ्यो है। त्यो सेसनमा चाहिँ हामीले के चाहेको थियो त्यसबाट हाम्रो आउटपुट के भयो भने नेपालमा न्युली अर्न मतलब इस्टाब्लिस भएका इन्स्टिट्युट वा कम्पनीहरूले कुन स्ट्याटसमा काम गरिरहनु भएको छ, कसरी सस्टेन भइरहनु भएको छ भन्ने बारे हामी विशेष गरेका थियौँ। त्यस हामीसँग धेरै डिस्कसन भएको छ। हामी रिकमेन्ड मतलब एउटा डकुमेन्ट बनाएका छौँ। हामी फर्दर यो 2018 को भन्दा फरक किसिमबाट अहिलेको यो ग्लोबल कन्भेन्सनबाट आएका आउटपुटलाई इम्प्लिमेन्टेसनतिर अगाडि जाने हामीले प्लानिङ गरेका छौँ। मलाई आशा छ तपाईँहरूसँग फेरि पनि वी विल बी किपिङ टच भेरी सुन। मैले चाहिँ NRN को तर्फबाट तपाईँहरूलाई मैले भन्नु पर्दाखेरि त्यो मैले भन्न चाहेँ। फाइनल्ली थ्याङ्क यू भेरी मच। विशेष गरी हाम्रो रोशनजी, सुशीलाजी, इन्दिराजी अनि हाम्रो नितेशजीहरूलाई म राति फोन गरेर उहाँको चाहिँ क्लिनिकबाट पनि बाहिर निकालेर गफ गर्दै उहाँहरूले चाहिँ टाइम लिएको थिएँ। सबै एटलिस्ट अजितजी, म्याम, सबैजनालाई मैले चाहिँ धन्यवाद दिन चाहेँ। विशेष गरी हाम्रो अनुपमा, डाक्टर अनुपमा, सी इज नाउ इन नेपाल एन्ड सी इज रियली वन्डरफुल मेकिङ दिस काइन्ड अफ इभेन्ट सक्सेसफुल। यो मात्रै होइन, हाम्रो रिजनलमा पनि सी इज डुइङ कोअर्डिनेटिङ वान सेसन होइन। इन्दिरा बहिनी, she has a very special experience of being in Nepal and she come back now in Korea. So this is a wonderful experience and sharing. Thank you very much. Thank you.
Thank you.
देवीजी, अब रुम नम्बर 1 मा जाउँ है।
हजुर, तपाई आउनु भएको छ?
म तपाईलाई निमन्त्रणा दिन आएको यता।
मैले तपाईँले देख्न मलाई खबर गर्नुभएको थियो। सरी, महासचिवजी हुनुहुन्छ डाक्टर हेमराज शर्मा। he is, as you know, he is chair of the so please मैले कन्क्लुड नगरिकन तपाईँ आइदिनु भएको छ। मैले थ्यांक यू।
होइन, म खास चाहिँ हामी रुम 1 मा थियौँ, तपाईहरु के गर्दै हुनुहुन्छ हेर्न आएको। सबै रुम 1 मा जाउँ। अब रुम 1 को लिंक छ नि यहाँहरुसँग हैन?
लिंक म शेयर गर्छु। केमा शेयर गरौँ?
Thank you सबैजनालाई। Thank you so much for the opportunity.
Thank you फेरि एकचोटी सबैलाई।
Thanks for
Thank you.
Thank you.
सानिमा बैंकको विदेशी मुद्रा बचत खाता। विदेशी मुद्राको बचतमा उच्चतम ब्याज दर। अमेरिकी डलरमा वार्षिक 4%, ग्रेट ब्रिटेन पाउन्डमा 3% र युरोमा वार्षिक 3% ब्याज दर प्राप्त गर्न सक्नुहुनेछ। सानिमा बैंकको खाता अनलाइनबाट पनि खोल्न सकिन्छ। अनलाइन खाता खोल्नका लागि eaccount.sanimabank.com/account मा लगइन गर्नुहोला। थप जानकारीको लागि हाम्रो भाइबर नम्बर 9860923783 मा सम्पर्क गर्नुहोला।
सम्पूर्ण बैंकिङ सेवाका लागि सानिमा बैंक सबल अनि विश्वसनीय बैंक।